4-2 वाला फोल्डर है, 4-2 के नाम से मैंने आज की डेट का बनाया, वो कॉपी कर लो, ठीक है। Yeah, sir. Perfect. Sumer, recording started. Okay. Yeah, good. Beginning. Hi. Do we have 9 o'clock already? We're good. Yeah, we are 9. Okay. So, Bismillahirrahmanirrahim. Assalamu alaikum, everyone. And thank you so much for joining us. Assalamu alaikum and welcome to all the participants online. I know we have an international audience online with many, many people who have joined and who will continue to join. So just like to give a very, very warm welcome. I hope it's a dry welcome too, because it's been quite wet here in Karachi. We've been, it's always Karachi, Karachi rains always are welcome for people because it doesn't rain very much in Karachi, especially in winter, but I think it's been a bit disruptive. So we hope that some of you participants who had planned to come and join us in person will at least join us online. But hopefully, as the day dries up, most of you will be able to join us here in Lecture Hall 3 at the AKU Auditorium. So thank you very much for joining us in our first genetics seminar organized collectively by the Pakistan Society of Medical Genetics in conjunction with many other institutions, which we will introduce over time. And we're so happy that there are so many people who have the love for genetics and have dedicated their careers and their studies and their lives to helping people with genetic disorders. So without further ado, I will introduce our first speaker, Dr. Syed Ajaz Ahmed, who will actually introduce the Pakistan Society of Medical Genetics and the efforts that have been made over the past few years to really democratize genetics knowledge in Pakistan and how to make sure that every medical professional is up to date with genetics and how they can use that information to help their patients. So Dr. Ajaz is a graduate of Dow Medical College. He's a partner physician at Kaiser Permanente in California and is on faculty at the University of California, Riverside. He has board certifications in pathology, clinical genetics, and molecular genetics pathology. He's one of the founding members of the Pakistani Society of Medical Genetics and Genomics. He volunteers his time at the Telehealth Genetics Clinic at the National Institute of Child Health, NICH in Karachi, and the South City Hospital in Karachi. So thank you so much, Dr. Ajaz. You've traveled all the way from the US to be with us. Please come and join us. Thank you.
Assalamu alaikum. Yeah, I've come from US. Walaikum salam to banta acha wala, right? There you go. Thank you. Thank you for the kind introduction, Salman, and thank you all for being here. I know it's a Sunday and, it's a holiday weekend for you guys. So for you to be here is a testament of your interest in genetics, and I really appreciate that. I stand here on behalf of a team who spent nearly 5 months organizing the symposium, and they did it over the weekends, and we had plenty of meetings. So it's I'm just being here as a representative of that team, and I think this is the first of its kind symposium in genetics, and I was talking to Dr. Usman, and it seems like it's probably the first genetic symposium in Pakistan of its kind. So you all are part of this historical event. So welcome to this. And should any of you attending today pursue a path in clinical genetics, being a geneticist or a genetic counselor, or even just incorporating genetics in your clinical practice, I think we would have achieved a goal, even one of you, if you do that. So that would be the best thing. And first and foremost, I would like to really thank Dr. Salman Kirmani, for his dedication and collaboration. And he played a significant role in bringing this symposium to life. And he really handled most of the heavyweight lifting. So thank you so much for all of this. We really appreciate it. I just wanted to just acknowledge our committee and our committee members, Dr. Salman Kirmani, of course, and Dr. Rizwan Naeem, Dr. Jamal Raza, Myla, Ashfaq, Aisha, Furqan. You will hear from them shortly. And Dr. Misbah, Hanif, Zoha Ajaz, Dr. Saqib Mahmood, and myself. This was the organizing committee, and we have our collaborating organizations, Aga Khan University Hospital and Aga Khan University, Pakistan Society of Medical Genetics, and Institute of Child Health and Neonatology, National Institute of Child Health, and Noor International University. For Aga Khan team, I would like to acknowledge the conference secretariat for their hard work, for all of you being here and all of this going live online. Manzoor, Irshad, Arif Mehboob Ali, and Khalid Chunara, and of course the whole audiovisual team here, and students for Pediatric of Life. I really want to offer my sincere gratitude and thanks to all the speakers who voluntarily spent so much time, and Dr. Usman Rashid for flying from Lahore over here, and Dr. Rizwan Naeem flying from all the way from the US. So it's a great team, and hopefully you will enjoy these talks from these amazing speakers that we have here. My special acknowledgement to all our poster presenters who submitted their posters and brought them over here, and to those who are setting up their stalls, Al Falah, Karachi Down Syndrome Program, and Patients Welfare Association. This all would not be possible, and we would not be here if it was not because of our sponsor. They have a booth there. It's Roche Pharmaceuticals, who are sponsored for big. So a big thank to our sponsors. I will take this opportunity to introduce Pakistan Society of Medical Genetics and Genomics to you, just a brief introduction, and go from there. This is our website, Pakistan Society of Medical Genetics. And just to give you a background, COVID was really bad times for all of us. There was a lot of hardship for a lot of people. But there was lockdown, and one good thing that came out of it was time for us to reflect on ourselves and ask ourselves questions as to what we want to get out of life. And for me, as they say, that in the heart of hardships blooms the flower of opportunities. So for me, it was something that was I always wanted to give back to the community. There's some guilt factor when you train here and you go to medical school here and then you go abroad and you stay there. You always want to contribute back and give back to the society. So I really wanted to start the medical genetics or bring genetics to Pakistan. I mean, Salman is here, and just to be part of that organization. So I picked up the phone one fine morning and called my teacher, my mentor, my friend, Dr. Rizwan Naeem, who's here, and told him that this is something that we should do. And he was very, very supportive. And he's a guy who has all the contacts if you ever want anybody. So he picked up the phone, called Dr. Jamal Raza, and he's a visionary guy. He said, yes, definitely, we should start this process. And then he recruited Misbah, but this would not have been complete without the genetic counselors. So we were able to find, I searched up Myla Ashfaq that you will hear from. And she contacted Aisha. She said Aisha is very much interested in doing this. So it just all formed together, and we started our clinics and all that, and we realized that we needed a platform. So we, decided to form the society, the Pakistan Society of Medical Genetics. And our mission is to enable the provision of affordable medical genetic services to the people of Pakistan and to create awareness of genetic disorders through education, community outreach, and research. So this is part of the education and the outreach. So we are all part of this. It is now a registered organization in the state of California with a 501c status. That means all the donations that we can receive, and we'll start doing fundraising in the US soon, those will be tax exempted. So it's a big process to get through this, and we're lucky that we were able to get this. We have a board that comprises of 7 members: Dr. Jamal Raza, Dr. Salman Kirmani, Dr. Saqib Mahmood, Dr. Rizwan, Naeem, Myla, Ashfaq, Aisha, Furqan, and myself. I would like to really acknowledge our web designer, Shazia Ali, who really has voluntarily spent her time to put everything and all our lectures and everything, and constantly updates our website. And our social media manager, Fatima, who does a wonderful job at posting everything on Instagram and Facebook. Right now, our membership stands at about 100, more than 100 strong. So within 2 years, that's a big accomplishment. Just thinking that, people are interested in genetics. This is a free membership. So those of you who are here and who are not part of this group, you can just sign on psmg.org and then just become a member. All you will get is all the emails and good information about genetics. And as we go forward, we will add more stuff to it. We have a lecture series on it, which Myla and Aisha will talk about. So with this, I will introduce you to Myla and Aisha, who really are the heart and soul of this organization. They have done a lot of legwork in getting everything done, and I mean, you can't thank them enough for what they've done. So they will talk to us about the accomplishments that we have achieved so far as an organization. So I'll just, you can hear them, and then we can start our lectures. Thank you.
Assalamu alaikum and welcome to the first of its kind Genetic Symposium, which is a joint collaboration between the Pakistani Society of Medical Genetics and Genomics, Al Khan University Hospital, the National Institute of Child Health, and the Sindh Institute of Child Health and Neonatology. My name is Myla Ashfaq. I'm a genetic counselor and associate professor at the University of Texas Health Science Center in Houston. I'm also one of the co-founders of the Pakistani Society of Medical Genetics and Genomics. I'm here with Aisha Furqan. Hi, Aisha. How are you?
Hi, Myla. I am a little nervous, but mostly excited. We have put in a lot of work for this symposium and hope that it is a learning experience for everyone. A bit about myself. So I am an oncology genetic counselor at Stanford Healthcare in Palo Alto, California. And I'm also one of the co-founders of the Pakistani Society of Medical Genetics, and we'll refer to it as PSMG for short. And we're a small but mighty organization that 4 Pakistani genetics experts actually started in the summer of 2020. It is comprised of course, you and I, and our geneticist, Dr. Rizwan, Naeem, and Dr. Syed Ajaz.
Yes, and a lot has happened since the summer of 2020, and we've come a long way. So before we talk more about PSMG, Aisha, can you tell the audience a bit about the profession of genetic counseling?
Yeah, great question. Yeah. So first, let's be sure to give it up for all the doctors that have been doing genetic counseling for all these years with very limited resources, and they do it well. And secondly, in majority of the countries that offer genetic counseling as a separate career path than traditional medicine, genetic counselors are master's degree holders with special training in both medical genetics and in psychosocial counseling. Much like medicine in general, genetic counseling is a practice that is present at all stages of a patient's life and their journey. Genetic counselors are embedded in various different subspecialties. Like you'll find them in pediatrics, prenatal, fertility, cardiology, oncology, neurology, et cetera, et cetera. Think of it this way. All organs contain DNA, so all are subject to having their very own genetic counselors.
Nice. Well said, Aisha. So what was the purpose behind starting the society?
Yeah, so the stated mission of the PSMG Society is to enable the provision of affordable medical genetics services to the people of Pakistan, and really to create awareness of genetic disorders through education, community outreach, and research. And ever since 2013, which was 10 years ago, when you actually published your groundbreaking research that showed that 100% consensus among Pakistani physicians in favor of referral to genetic counseling for common conditions like Down syndrome, beta thalassemia, and hereditary cancer. So then I asked myself, well, what can I do to improve the state of genetic counseling in my country? And just in a recent publication that we had, we found that 660 geneticists and 2,500 genetic counselors are required to meet this need of genetic counseling in the Pakistani population of 220 million. And so, finally, that moment arrived when you called me and said, hey, what about this idea of starting a professional society where like-minded individuals can come together to promote this culturally sensitive, scientifically sound, and academically oriented genetic counseling. And this is where we do some shameless plug of what we've accomplished in the past. So take a minute to share those with our audience, if you will.
Yeah, definitely, Aisha. It was actually that paper that helped Dr. Ajaz Ahmed find me, and then I reached out to you. So it all worked out, and PSMG was formed. But one of our initial projects was to plan a needs assessment webinar. And we did this in collaboration with Jinasin Medical University Alumni Association of North America, JSMU-ANA for short, and the National Institute of Child Health, Al Khan University Hospital. And in this webinar, we interviewed experts, both from the United States and from Pakistan, in a wide range of healthcare specialties and also patient advocacy groups. The objective was to identify gaps in the provision of medical genetic services as perceived by healthcare providers and the general public. The webinar took place on Zoom on December 6, 2020, and we were able to publish the findings of the webinar in a peer-reviewed journal. This webinar, the discussion that we had was very useful because it provided us with a lot of insights into the current state of medical genetics in Pakistan. And that also helped us streamline the goals for our organization. And there are many goals, but one of which is the need for increased education in genetics in the country. So while our long-term goal is to have a master's degree program for genetic counseling in Pakistan, in the interim, we organized a monthly lecture series, and that was in collaboration with JSMU-ANA, in which different genetic topics were discussed, such as prenatal genetics, Down syndrome, cardiac genetics, cancer genetics, so on. So we have a total of 17 lectures, and I definitely want to thank all of the specialists and the healthcare providers and the advocacy group members that helped and spoke in these different lectures. And I also want to thank the audience that attended and participated in the discussions. So you can find all of the lectures on our website, psmg.org, and they are free to view. So that was the education aspect. Do you think, Aisha, you can tell us a little bit about the training of genetics clinic coordinators and the clinics that you and Dr. Ajaz have been doing?
Yeah, pride and joy. I'd say we've had so much fun and learned so much from these clinics. So yeah, I'd be happy to. So we're really inspired by how Dr. Kirmani had conducted a training program for Fizza Akbar. And we thought of training multiple clinical coordinators that could work as genetic counseling assistants while getting trained on the concepts of medical genetics and genetic counseling techniques. And so since there were no formal genetic counseling training programs in Pakistan when we started, and no designated medical genetics clinics in any government or nonprofit hospitals, so first we had to establish a genetics clinic where we could see patients via telehealth, which would then require a clinic coordinator who could observe the genetics team from the US and then learn those tips and tricks of the trade. But I'm going to be quite frank with you. I think that the learning has really been a two-way street. At NICH, we work with Dr. Mohsina Ibrahim, Dr. Arif Prakash, and Dr. Jamal Raza. And we also work with clinic coordinators, Dr. Misbah Hanif and Zoha Ajaz. And since the early 2021, when we started that clinic at NICH, we have seen multiple cases of children with skeletal dysplasia, inborn error of metabolism, disorder of sexual development, as well as chromosomal disorders that are so rare in other populations. And so we learned a lot, not just the medical side, but also the psychosocial side. And at South City Hospital, we work with Dr. Shama Munim. And again, we saw individuals with histories of multiple miscarriages or neonatal deaths, ultrasound abnormalities, and then, of course, previously known genetic disorders in older siblings. And then while we don't see patients at NICVD, we are working with Dr. Mohammed Mohsen and Dr. Azam Shafqat as consultants. So if they have any cases that require genetic counseling attention, or they want to know more about any variants that are identified, they send those cases to us, and we see them on a consultation basis. So all of these local teams have been tremendously helpful in our understanding of the psychosocial aspects and genetic testing and counseling bits. But we're very fortunate to have recruited Dr. Misbah Hanif and Zoha Ajaz, both of whom are scholars of molecular genetics, and we're very eager to learn about these genetic counseling tips and tricks. So you'll hear more about the telehealth genetics clinic from Misbah's talk later on in the evening, but I just wanted to say thank you to those local teams that have been so instrumental in our learning. And just a little bit more about the training bit. So we conducted 1 or 2 genetic counseling sessions with some of our US-based centers of excellence. So there's one in Delaware for skeletal dysplasias at Nemer Hospital in Delaware. And we did a genetic counseling session with the genetic counselor here who had expertise in skeletal dysplasia, and our genetic counseling coordinator was able to observe that session as well to see what that looked like. And then genetic counselors from all over the world contact us via PSMG website, and they need assistance helping find resources for testing at-risk relatives that live in Pakistan. So we've been very fortunate to have partners in different parts of the country that were able to facilitate testing for at least some of these individuals.
That's really outstanding work, Aisha. And now that you mentioned the facilitation of testing, can you tell us more about some of the genetic testing collaborations that our team has established?
Yeah, so we have built some bridges with 1 or 2 labs that are US based, and we connect them. They're commercial laboratories, so we won't be naming names, but there are some labs that we have built connections with at NICH and at South City Hospital. We have been working towards building a bridge between Pakistani labs as well as US-based genomics lab. And so far, we haven't really been very successful in finding affordable testing. And largely due to the lack of expert personnel, large gaps in variant curation process of the NGS testing. One idea that has been floating around is to offer send-out testing for affected members of the family, which is going to cost the big bucks, but then offer site-specific analysis to patients' relatives in Pakistan at a much more reduced cost. Another idea that has been floating around is to find a partner lab that can do next-gen sequencing in Pakistan and upload that data through a cloud service that is in a foreign country. And then they can, with their expertise and databases and machine learning tools, they can do the variant interpretation and generate the reports. And of course, this would be the most cost-effective way and will reduce not only just the cost of shipping and sending samples outside the country, but also variant analysis, we think. So yeah, lots more to go. We're eager to continue to do our work, and we're steadfast, slow and steady. And so we're looking forward to what the future holds. So Myla, can you tell the audience a little bit about how they can get a hold of us and find out more about the society?
Yes, so the easiest way to contact us is from our website, which is psmg.org. That website has been maintained and is updated by Shazia Ali. And I just want to say a special thanks to Shazia, because like Shazia and all of the other members of PSMG, me and you, we are all doing this work on a volunteer basis. So thank you, Shazia, for donating your time with the website and keeping that up to date. So overall, PSMG is an organization that's continuing to grow, and we are welcoming new members. So if you would like to be a member of PSMG, then please visit the website, and there is a membership form which you can complete and submit. We're also present on social media. Our Instagram is GC in Pakistan. And for that, we'd like to thank Mahnur Fatma, who updates and runs our Instagram handle. Again, she donates her time to do that. So in summation, I would say that, we have achieved a lot as an organization, and we continue to have lofty goals for 2024 and the future. We plan on continuing our educational efforts with the goal of increasing the presence of genetic experts in Pakistan, to help with the provision of equitable genetic testing and medical genetic services, to increase the awareness of genetic conditions in the general population, and to continue to build and strengthen collaborations with institutions, individuals, and organizations that currently have boots on the ground. So hopefully we're going to achieve all of that, and hopefully we'll continue to have the support that we've been receiving from everyone in Pakistan and internationally. So thank you so much for your attention, and we really hope you enjoy the symposium. So Khuda Hafiz from me.
Okay.
Thank you, Dr. Ahmed, for your talk, and thank you, Ms. Aisha Furqan and Myla Ashfaq for joining us. And we're looking forward to your talk in the afternoon, Dr. Ahmed. Following that, we are very privileged to have another distinguished expert in this field with US board certifications in pediatrics, pediatric endocrinology, and medical genetics. Dr. Salman Kirmani is an associate professor and chair of the Division of Women and Child Health at Aga Khan University. He graduated from Dow University and went on to train and work as a faculty at Mayo Clinic before returning to Pakistan. He's dedicated to improving access to diagnostic therapeutics and preventative interventions for patients and families with genetic disorders, and building local capacity by training students, residents, fellows, and genetic counselors. Please join me in welcoming our keynote speaker, Dr. Salman Kirmani.
Thank you so much, Maha. So let me introduce Maha. So Maha is she's a fourth year medical student here at AKU, and she is the convener for Pediatrics for Life, which is the Student Interest Association for Pediatrics. And as we all know, once you're in pediatrics for life, right? So thank you very much, Maha, for that and for the kind introduction. And thanks to everyone. Loved Myla and Aisha's talk, and I'm so happy to see such a wonderful audience here in the hall with us, and I'm sure the online audience is enjoying themselves too. Shout out to our pediatric residents who are here in their busy schedule and their on-call schedules. They've dedicated their time out to come here. Our medical students, our stellar medical students who are here, and not just AKU medical students, I think medical students from other areas too. Our nurses are here, our genetic counseling team, and really wonderful to see so many people in the audience. So my job is to whet the appetite for the rest of the day. So I'm going to hopefully give you an introduction to the wonderful world of medical genetics, because medical genetics specifically revolves around patient care. That's really the bottom line that we're doing, because genetics is a huge, huge field, right? And I remember even at Mayo Clinic, when my badge said medical genetics in the elevator, other doctors would ask me, so which chromosome are you working on, or which gene are you? And I had a stethoscope around my neck, and I said, I see patients just like you. I order the test, I tell patients what to do. So, this is medical genetics, this is really what we do. And medical genetics doesn't just mean people who see patients. These are people who also sit in the labs and interpret the results, so just like our radiologists may not interact with the patient personally, but they are the ones who actually help you take better care of your patients. So this is the team of medical genetics, which includes the clinical people who see the patients, as well as the laboratory geneticists and the genetic counselors who kind of, bring it together so that the patient gets the best information to make the best decisions for themselves and their families. So no disclosures for me. And I'm going to really introduce everyone to precision medicine, because this is something that really, really is at the heart of medical genetics, right? You want to get to personalized, care. And then I'm going to introduce the term genomic medicine, because this is an evolution in terms, genetics is an evolving field. It's an ever-changing field. By the end of the conference, we'll probably have a new term that some people use. And there's lots of challenges and opportunities, especially in Pakistan. And that's why, I was a happy geneticist, in the US, but it is just so wonderful to be able to practice that knowledge over here, because number one, the hunger for knowledge is so much. People, the students just gravitate towards anything close to genetics because, it's such an attractive field in its own right. But also the patients, there's so much need, the number of patients that need to be seen, the number of diseases that are there, as Aisha mentioned, and just the desire to do something about it. One of the things that people told me before I moved from the US was, what are you going to do over there? People don't even have money for regular care. So how are you going to, this is a boutique or a niche field. And what they didn't realize is that people travel from far and away to get an answer for their genetic condition. And they know that you may not have the cure, but they want better care. They want a better understanding. And that is where we all come in, and better understanding leads to better care. Without understanding, you can never care for your patient. So that's very, very important. So, precision medicine is, really hit the target where it needs to be hit, right? And it's very interesting that we all think, this term individualized medicine, precision medicine, it's been around for at least a decade, if not more, couple of decades, right? But are we truly hitting the target or not is the question, right? So I think we need to kind of have a paradigm shift to the way we see our patients. So, in my student lectures, I always make fun of the endocrinologists, because I am a pediatric endocrinologist, and I do feel I have a little bit of right to make fun of the adult endocrinologists. Maybe some of them are not in the audience today. But, you go to a diabetes clinic, and I tell them, you can enter the clinic at 9 in the morning, and then you can close your eyes, and at 5 in the evening, the same conversation will be going on about the same drugs, about the same non-compliance, about the same tests, and it goes over and over and over again. So what are we doing with our patients? We're throwing the dice at them. We're hoping that some will benefit and some may not benefit, and some may actually have side effects, which we are causing, and we've been doing that for decades, and that has to change. So then has come the era of stratified medicine, where you use biomarkers, you use population demographics, you use some information that has been collected very meticulously over the few years to maybe stratify risks and say, okay, this is the higher risk population. This particular ethnic group requires this medication because they do better. And you kind of, try to fine tune your care, but it's not quite precision medicine. So what is precision medicine, right? So precision medicine is where each individual patient gets the right care at the right time, right? And that's where you're not just throwing the dice anymore. You are studying all the factors, and the background obviously is genetics. So the genomics of the patients really, really make a big deal, but it even goes beyond genomics. This is called the multi-omic approach to care, where the proteomics, the metabolomics, et cetera, and so forth are important. And then all of the medical information that the patient has gathered over the years, you use big data and machine learning to put that all together so that all the background genomic data, the multi-omic data, the biological data of the patient combines with the patient data from the electronic record, and then you truly do the right thing for the right patient at the right time. And we're hoping that the access to all of this multi-omic things becomes routine. I remember when I was a trainee in medical genetics, people told me that, in a few years, people will be walking into a general store, a pharmacy, they will spit in a cup at the entry point, and by the time they're at the checkout counter, they will get a personalized set of drugs and a prescription list based on their genomics. Hasn't quite happened yet, but it's certainly within the realms of imagination. So then we come to, okay, so what is the difference between genetics and genomics? Why differentiate the two? So, in general, genetics is the study of monogenic conditions, right? Things that, we teach you, things that you see in clinic, cystic fibrosis, thalassemia, etc., where one single gene, causes a single problem or a set of problems. But genomics is more broad than that. And that's why the American College of Medical Genetics changed their name to Medical Genetics and Genomics. And the PSMG is also the Pakistani Society for Medical Genetics and Genomics, because this is the study of the entire organism's genetic makeup. And it includes how genes talk to each other, how genes talk to the environment. And it includes not only the monogenic disorders, but also the complex disorders, where a lot, that's where most patients suffer. They don't suffer from monogenic disorders, they suffer from complex disorders. So it's really important. And I truly believe that it's actually the monogenic disorders that will help us take better care of the patients with complex disorders. And certainly cancer care has benefited from all the benefits from these. So, this is the central dogma. Everyone knows is you can't have a genetics conference without showing this diagram, right? So this is how everything unravels. This is how we're organized. This is the oldest whole genome test, started in the late 1950s, right? This is whole genome, right? It is everything, but it's not high resolution, right? And you can see that, we learned how to look at the genome as an organized set of instructions, the encyclopedia set of life, or the cookbook for life, and so forth. And it's important to this day, we start with the chromosomes and then we go deeper. And then we say, okay, what does a gene do? What is a gene, right? And it's been defined by these terms. And, it is the fundamental unit of heredity that passes on from generation to generation. Very interesting, when the Human Genome Project was being initiated, people thought they'd be looking for 100,000 genes. And the reason was because there was 100,000 proteins. So we said, one gene, one protein. Well, that theory, was obviously shut down because we learned about alternative splicing and how one gene can actually make multiple protein products, right? So the consensus now is that there's somewhere in the ballpark of 20 to 23,000 genes, 25,000 genes. And there's actually controversy about certain factors as to, do we really call this a gene? Does this encode a protein or not? Does it regulate another gene? What does it do? So there are genes that don't make proteins, but encode RNAs. And control, genetic transcription. So that's very, very important to know. And it's also surprising when I tell most of my trainees and even practicing clinicians that most of our DNA is non-coding, and in the old days, in the dark ages, we used to call them junk DNA. Well, it's not junk DNA, because if it was junk DNA, evolution would have gotten rid of it millions and millions of years ago. The reason why we've hung on to all this DNA is it must be doing something useful. So around 1 to 1.5% of your DNA is the exome, which is the coding region, and 98.5 is non-coding. And we're learning more and more about the non-coding, region and how important it is. And, genetic variation is part and parcel. Everyone sitting in the audience today, everyone online today, we know there are genetic differences between us, right? So it's really important to start with how identical we are. So 99.9% of our DNA is exactly identical. It's really that 0.1% that's different between each one of us that makes us unique. Some of these DNA differences are part of normal human variation and really don't make much of a difference. Whereas others directly cause very severe, very bad disease. And, it's really, really important to see these two extremes, right? But between these two extremes is where most of where human disease lies. The diabetes clinic, the general cancer clinics, the hypertension clinics, they're full of these patients who have polygenic diseases, who have multifactorial diseases, where there are basically genetic influences and environmental influences all coming together. when what we say in genetic disease, the monogenic disorders are the extreme example of what a variation can do. So the delta F508 pathogenic variant can cause a very severe phenotype because of its consequence. But there may be milder variants in the CFTR gene that maybe don't cause the full-blown disease, but maybe cause some mild susceptibility to some of the features, and we're understanding all of that. And we have to realize that just because the genetic sequence shows something different from, what our reference genome is, doesn't mean it's normal, because it really boils down to who's the reference, and it turns out that the first human genome was a person of Northern European descent. That can never be the reference genome for us, right? And for decades, we used Western data as the reference genome, and we still continue to do so. And that's where our population ends up with so many uncertain variants of uncertain significance, because we don't know in our population what those variations are doing. So the way to look at it is on a continuum, that, there are variants that will have a high effect size, and that effect size will be recognizable as a disease, as a phenotype. And there will be variants that will be so common in the population that they may have such a low effect size that you may not even notice it. And it's this, this sort of continuum, and all of us carry some high effect size variants and a lot of common variants. They're common, that's why they're all around, which have a low effect size. And maybe most of human disease lies somewhere on this spectrum, where there are minor variants that are not very common, but not very rare either. And it's the combination of these variants that predisposes us to cancer and to coronary artery disease and to hypertension and so forth. So, this is what I meant when I said that learning about monogenic, the rare genetic disorders, will better prepare you to take care of the huge slew of patients that actually needs your help. So it's really, really important. So do not ignore your rare disease patients. The reason why we call them orphan diseases is because these patients are truly orphanized. No one likes to take care of them. No one knows how to take care of them. And one of the goals of the conference today is to make all of you much, much more comfortable in taking care of these individuals. So I like this slide because it sort of summarizes all that I've said in numbers, right? The 6 billion letters of DNA, packaged nicely into these 22,000 genes on the 46 chromosomes can fit, in a thumb drive. I mean, it can really be, this is your data, right? And this is an old slide. And I'm happy to say every time I present the talk, the number goes down. And even last night I was tempted, is a whole genome really $500? It's probably lower than that somewhere in China. Somewhere someone's working on it, and it's for probably $300 or $200. And people are talking about the $100 genome, which is truly going to make it happen. So it's really good. And this is the fun part. And that's where the medical application of all this knowledge comes in, right? So one thing which what PSMG was talking about, what we all do, is to take care of these patients with Mendelian disorders. And we say Mendelian to honor Gregor Mendel, who really started the field. But there's so many other things that can happen, pharmacogenomics, where if you understand how certain genetic variants make you a fast metabolizer or a slow metabolizer of a drug, you can actually predict whether that person needs a higher dose of a drug or a lower dose of a drug. So this is used routinely in, when warfarin is prescribed, for example, and you want to know whether, this person is going to be susceptible to bleeding or not. This is used routinely in psychotropic medication prescriptions because the therapeutic window is so narrow that you don't want to give too much or too little. So pharmacogenomics has really, really changed the way we do this. And it's not a high cost test. It's a pretty low cost test because it's known variations that will do this. And it's becoming much, much more routine in cancer care now to study pharmacogenomics and to tailor chemotherapy dosing and drugs accordingly. It's because of the advances in genomics that drugs like imatinib or tyrosine kinase inhibitors were built, because once you understood the genetic mechanism, you knew that instead of carpet bombing the entire tumor, you could home in on which cells had the pathway that was ramped up or ramped down, depending on the variant. And then you could target that. So, lots and lots of new treatments in cancer have started because people started studying the tumor tissue. They started studying, because cancer at the cellular level is a genetic disease, right? And Dr. Usman will educate us much more on that in his talk. So if you understand that genetic mechanism, you can treat cancer better. And there's so many more exciting technologies that I'll also mention later. And then, it comes to the common diseases. If you understand what's going on here, you will be able to do such a much, much better job of taking care of your patients with the common diseases. So in diabetes care, we're realizing now that people with certain polymorphisms may have that higher susceptibility, and you may need to treat them differently. And, with other common diseases, this has started happening too. And this is where active areas of research are happening. So a lot of you people who are thinking of careers in internal medicine or oncology or pathology, you don't always have to study rare diseases. The common diseases and the genetics of common diseases will be way of the future. The next 50 years, we'll see a lot of progress over there. And then comes the art of predictive genomics. If at a young age, before the disease sets in, the genomic information can guide you as to who will develop cancer later or who will develop diabetes later, maybe your interventions will be much, much more focused and much better. And maybe the preventative actions will help the disease from actually showing up. So this helps us transition into the genetics clinic. What happens in a genetics clinic, right? So a lot of times the geneticists were initially accused of just being diagnosticians. Okay, you can pat yourself on the back, you diagnose a rare disease, you guys are very happy. What about the patient? The issue is everything, the patient's journey doesn't end with a diagnosis. In fact, it begins with a diagnosis. The first thing a diagnosis gives you is prognostic information. Imagine a baby in the NICU, day 2 of life on the ventilator, very, very sick, and you don't know whether they're suffering from a very severe, untreatable metabolic disorder. The conventional diagnostics would take at least 3, 4, 5, even 6 weeks, even 2 months to come to a diagnosis. Would you keep that baby on the ventilator, on the care, the expensive care? Anywhere in the world, it's expensive. But if you were able to do a quick genomic test within 2 or 3 days, get a diagnosis and know exactly this, even though it may not be untreated, there are many, many diseases even today that are quote unquote untreatable. But if you can get prognostic information, you can make sensible decisions which can really ease the pain and the suffering, not only of that baby, of that child, but of the family who get financially and emotionally devastated in that very, very long diagnostic odyssey. So one of our, proud things that we do is we end diagnostic odysseys. That's a very, very important part of what we do. And nowadays, thankfully, with all of the advances that you learn about, we can tailor management. We can actually manage things that were unmanageable before. We can actually treat things that were untreatable before. And you can come up with a surveillance plan. Imagine someone being diagnosed very early on without having a cancer with a BRCA1 or BRCA2 variant that can cause a cancer risk, right? If you know their entire lifelong surveillance can be tailored towards that high-risk situation, and you can actually prevent cancer with prophylactic procedures. And we hope, hasn't happened yet, but we hope even medications that will then be given to these individuals so that they will never develop cancer. So again, if you didn't have the diagnosis, you couldn't even begin that conversation with these families. And that's where the pre-symptomatic people come in, right? You start with the patient, but you identify downstream all of the other people you can help. And I think geneticists take care of families. The genetics team's job is not to take care of the one individual. We take care of the entire family. And that's one thing that I feel that all of the specialist clinicians really appreciate about our teams is that we don't just stop at the patient, and you learn through the day how we do that. And also, because some of these targeted treatments are so expensive, and because the patient's journey is so challenging, even with treatments, the one thing that we have to put our energies on is prevention. And the way to prevent is offer reproductive risk. Dr. Shama Munim is here. She joins us. She has dedicated her life to giving those couples with those very, very challenging situations a way out, a way to prevent this from happening in the future. So let's talk a little bit. We're going to change gears and talk about these exciting new therapies that have come about, because this has really, really, sort of energized the field. And so many young people now are entering the field of genetics because of this. The biotech industry, the pharmaceutical industry has really blossomed because of the advances in genomics. And this is what I call sort of the genetic-based therapies, because not everything is gene therapy, not everything is gene editing. There are nuances, and it's important for everyone to understand, all of the different therapies. So I'm just going to give you a bird's eye view of some of these that are out there. A number of them are already FDA approved. A number of them actually are being given in Pakistan, and that's really a phenomenal achievement. So, the poster child for this is spinal muscular atrophy, or SMA, a devastating disease. In its most severe form, SMA type 0, it's lethal. But the commonest form is SMA type 1, where most babies don't make it to their first birthday. They don't survive because their muscles get so weak, they aspirate, they have recurrent pneumonias. It's a very, very challenging situation. And in Pakistan, because it's an autosomal recessive disease, we see it more because of consanguinity, because of the cousin marriages. And we see multiple children with it, because they don't get appropriate counseling. So we've seen 2, even sometimes 3, deaths in one family, and it's very, very devastating. Other diseases have also developed, there are therapies developed for others, but I'm going to give you some examples of some of these that are happening. One of the biggest advances is designer drugs, or what we call these antisense oligonucleotides, ASOs. And these basically are synthetically created antisense oligonucleotides that target, pre-mRNA, right? So before the messenger RNA can be processed into, the translatory mechanism, it targets it. And what's beautiful about ASOs is you can use them to basically degrade the mRNA so the protein translation does not happen. Or you can actually even go the opposite route and increase protein production. And it's sort of a phenomenal way. You're doing nothing to the genetic background. You're not changing the DNA. You're actually targeting the messenger RNA. And this has really, really changed the field for a number of diseases. So I'll give you the example of spinal muscular atrophy, SMA, which happens due to pathogenic variants in SMN1. The most common is that the SMN1, the critical region, exons 7 and 8 are actually deleted. The critical part of the gene is actually not there. So when the DNA is not there, you can't really correct it, right? So human ingenuity, right? We learned as research progressed that there's an alternative gene called SMN2, which naturally has a variation that makes 90% non-functional protein and only 10% functional protein. Right? And sometimes it's those copies of SMN2, because naturally some people may have 2, 3, or even 4 copies of SMN2. The more copies of SMN2 you have, the milder disease you have, because you're making some protein. So, the scientists who are working on this said, how can we manipulate SMN2 to make more than just 10%? And that's what they did with what they call the splice switching oligonucleotide, or SSO. What this does is that it basically alters the splicing between exon 7 and 8, which is the problem which causes the deletion, and includes exon 7 back into SMN2. So it's almost like correcting the naturally defective gene by splicing, by alternating its splice site. And when you do that, your SMN2 essentially turns into SMN1 and starts making normal protein. And it's become a very good solution. So this is the drug Evrysdi, or risdiplam, which is made by Roche Pharmaceuticals. It's available worldwide. It was very challenging to get in Pakistan, came in as a compassionate use program. But since the last year, we've been working with Roche, and they've created a very, very interesting program to improve patient access, where they subsidize 2/3 of the cost. And the patient has to arrange, or through an agency or organization, 1/3 of the cost. And it's improved access to a number of patients. And we'll hear from, we'll get updates from our Roche colleagues today. But I believe there are more than 50 patients in Pakistan now who are actually on Risdiplam. We're following about more than 15 here at AKU through this program. And there's a Punjab program, there's a KPK program. It's a nationwide program. And it's been a phenomenal help in giving these patients a treatment option. A similar thing is happening with Duchenne muscular dystrophy. Again, a bad disease, not lethal immediately, begins around 3 to 4 years of age. But by the age of 10, most boys are in a wheelchair. Most boys will die by the age of 20 or beyond, because the cardiorespiratory failure that comes with the disease is quite devastating. There is a mild variant of the disease called Becker muscular dystrophy, because it's the difference between in-frame and out-of-frame deletions. In general, very, very simply, in-frame deletion means some protein is still made. Out-of-frame deletion means no protein is made. So the severe disease, Duchenne muscular dystrophy, those patients are making no dystrophin. In Becker muscular dystrophy, they're making an altered protein. Those muscles survive a bit longer. Those individuals can actually live to 40, 50, and sometimes beyond. They have a late onset disease. What can you do? Again, human ingenuity. You cannot change that. It's a huge gene. Gene therapy was very, very difficult because it was a very big gene. You couldn't package it into a viral vector. So, and gene therapy has just been approved a few months ago for Duchenne, but it's a very, very small window, and it's an altered gene therapy. What they did was they asked the question, how about we turn a Duchenne muscular dystrophy patient into a Becker muscular dystrophy patient? And that's exactly what the drug does. So what the drugs do is they take an out-of-frame deletion, splice switch through the antisense oligonucleotide, and get things back into frame. I mean, a phenomenal solution to a conundrum. DMD is something we knew for decades. Everyone, this was a test that even has been present in Pakistan for many, many years. We have patients diagnosed, but we couldn't do anything about it. So what we were able to do is to do this and get into a milder disease spectrum, increase life expectancy, improve quality of life. The problem with the therapy for DMD, though, is and actually same thing with Risdiplam, it is lifelong and it is expensive. And the Duchenne therapy is yet to be given to any Pakistani patient because there is no compassionate use program. There is no access program yet. We've contacted the pharmaceutical company that makes these drugs, but at this point, there is no interest in expanding an access program to Pakistan yet. But that's where advocacy comes in and the group comes in. This concept that David Liu began at MGH is fantastic. If you know your patient's variant, and most patients will have unique variants, you can actually tailor your ASO. You can manufacture the ASO in your lab and then deliver it to the patient. So this is fast-tracking FDA approval, fast-tracking all the research, and giving a chance to patients with devastating diseases. Batten disease, very bad disease, right? And again, daily seizures, minute-to-minute seizures, epileptic encephalopathy. What they were able to do, look at the timeline. The identification of the variant is here, and within about 18 months, you've given a drug to a patient. On average, it takes 15 to 30 years to develop a drug. And here you're doing it in 24 months. It was phenomenal, right? So if this can happen, if this can be democratized, a lot of patients will get a lot of benefits. So it's very, very exciting time for antisense oligonucleotide therapy. Let's move on to gene therapy. Everyone's heard about gene therapy. Zolgensma, the drug for SMA, has been given in Pakistan through the managed access program. Unfortunately, in January of 2023, that program was discontinued for Pakistan. Very, very unfortunate. So we have not been able to give it to more than 5 patients. Those 5 patients who've received it, doing very, very well, surviving, and that's the key. in diseases where you want to change the DNA and correct it, you need a Trojan horse. You need a viral vector to deliver the corrected DNA into the nucleus. The problem with traditional gene therapy has always been that when you try to cut the host DNA and incorporate this DNA that you've, specially flown in through your viral vector, you can turn on or off certain key genes that can cause major problems. And one of the big problems was turning on oncogenes or turning off, tumor suppressor genes. And if you did that, those patients would be susceptible to cancer, and you'd exchange a smaller problem for a very big problem. So that was one of the reasons why it took a long time for it to actually come into, the market. The other thing was, and people who followed the research know this, that there was some unethical research practices that happened at universities, and there was a moratorium on gene therapy research for a long time before things began again. And so very, very careful. Any research that has to be done properly, has to be done under proper research ethical approval and surveillance. The cool thing about newer gene therapies is that they've come up with viral vectors that are non-incorporating. So you can now basically have a viral vector that enters the nucleus, and just the DNA just forms a small circular episome and doesn't mess with the host DNA at all. And so it's considered a much safer approach than your standard gene therapy. And the package load has to be small. So currently it is only used for smaller, packages, smaller genes. But hopefully, as we develop these viral vectors, we can use it more. So Zolgensma was, onasemnogene abiparvovec for SMA is delivered through an AAV9 vector. And this is the vector that is being used now for most new drug development in gene therapy. And then there's this concept of giving gene therapy to the patient in vivo, which is what happens in SMA, versus giving it ex vivo. Because if you're worried about side effects and you're worried about why would I want to change every single cell? If I'm worried about a disease that only affects hematological cells, why don't I just change the hematological cells? And this is the concept that is being used for both sickle cell disease and beta thalassemia, where you basically take the cells out of the patient, you do gene therapy ex vivo, in the Petri dish, and then you deliver an autologous stem cell transplant back to the patient. So it's personalized therapy with the patient's own cells. And this has really taken root. The limiting factor currently is expense, because not only is there the expense of the drug development for the gene therapy, but also the expense of the autologous stem cell transplant and all the machinery required to keep those stem cells healthy and alive while you're doing the transplant. And that has currently been a prohibitive factor in our part of the world. Then comes CAR T cell therapy, and I'm sure you'll hear about it from Dr. Usman as well. This has really changed the face of how we're going to deal with cancer in the future. And it's basically using your own immune system and changing it in a way that it recognizes the antigens on the cancer cells. And again, it's a way of fooling your immune system to only hit your cancer cells and not your regular cells. And again, you take the cells out of the body, you change the DNA such that, they basically make antibodies that will, target the antigens on the tumor, and then you inject them back into the patient so that only the tumor cells get hit by your T cells. It's a phenomenal way that cancer is going to be dealt with. Currently, it is being used as second-line treatment for people who have failed their primary cancer treatment. But as it becomes more ubiquitous and it becomes more accessible, I'm sure it may be used as primary therapy as well. And then comes gene editing. And this really, has made people very, very excited. People know about CRISPR and all its potential, but CRISPR is just the tip of the iceberg. CRISPR is just one of the gene editing tools. There are many, many others that are being developed, and CRISPR was something that basically was present in nature and was, very appropriately studied and manipulated by two fantastic researchers, Dr. Charpentier and Dr. Doudna, who really had the honor of not only just being the two women scientists the first to collectively win the Nobel Prize, but this was one of the shortest journey from experiment to prize in the history of the Nobel prizes, because on average, again, it takes about 20 to 30 years for someone to do the work before the Nobel Committee recognizes it as important and recognizes it as being something that will continue to change, have its impact. These guys got it within a few years because they started the research in 2010 and they got their award, 2012, in fact, and they got their award in 2020. But people have gone even further than that. So now, instead of making the double-stranded DNA, cut, which CRISPR does, people have moved on to base and prime editing, where basically you make a single-stranded nick. It's not even a full cut. So the less you have to manipulate the host DNA, the safer it is, the less off-target effects. Because one of the current problems with CRISPR is that you're trying to cut here and you may cut there, and that will be a huge problem, as you can imagine. So this will really change and revolutionize the field and really help develop newer drugs for treatment. I'm going to end by saying that I've excited all of you with all this fun stuff going on, but what happens when you're sitting in front of the patient? And that is a huge problem. Because to me, if I were to zoom back 5 years ago, if I had a family with SMA, I would tell them that even if you take your child to Europe or the US, they will be able to do nothing. So, we can't offer anything here. This is an untreatable disease. This is a bad disease. I'm sorry, but we will, do palliative care, but we cannot do anything for your patient. Now, because these drugs are not accessible here, I have to tell them, if your child was born in the US or Europe, your child could have gotten this therapy. But because you're born here, we can't give it to you. And obviously, I don't say that, but they come because they know these drugs are out there. They know there are WhatsApp groups of hundreds and hundreds of patients who are wanting these drugs, who are demanding these drugs. And we have to tell them, we don't have those drugs here. So what do we do, right? So the first limiting factor is the cost. Zolgensma became the most expensive drug in the world when it was released, $2.1 million. The beta thalassemia and the sickle cell gene therapy that I showed you, $2.8 million. So it even gets even more expensive when you have to do ex vivo, because it adds the cost of all of the extra stuff that goes in with ex vivo therapy. So this is a huge factor, right? The Spinraza or nusinersen that is used for SMA, that's an ASO. Look at the cost. I mean, unsustainable even in the Western world, right? How will you give it for years and years to patients? Evrysdi or risdiplam, this is the actual cost. The program here subsidizes is two-thirds, but you can do the math. Even 1/3 of this is about 20 to 30 lakhs per year, and for years and years and years. And if you give it for a few months, it's going to do no good. So there are people who sell their homes, who sell their properties, who say, "Ke 2-3 mahine ke liye mil jaye." And I told them, don't do that, because there's no point in doing that when you can't sustain it. So it's just a huge problem, right? The drug for DMD, look at the cost. I mean, huge. And again, these are long-term. So we will have to transition from these long-term therapies to the single one-time therapy that will be happening. The managed access programs, the compassionate use programs, to be very honest, sounds much better on paper than it is in practice for most of our patients, but you got to try. Even if one patient can benefit, you got to try. And, this is how it happens. What happened with hepatitis C and HIV? People tried. And that's how the drugs became free in Africa. That's how hepatitis C drug is free here in Pakistan, because people tried. They didn't give up and said, oh, it's an expensive drug. We can't do it, right? So this is the ethical dilemma that all of us are living and facing, on a regular basis. And this is the challenge in Pakistan specifically, right? That we are one of the worst in spending on healthcare. This is one of the lowest spending on healthcare, annual. This is annual, right? So it was just crazy. This is ridiculous. And yet, we live in a country where gene therapy has been given. We have, we live in a country where there are expensive cars that are imported regularly, by people who can afford them. So this sort of dilemma that we see daily in our society, the extremely rich and the extremely poor, plays out in the medical field. And it's really, 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 very sad. I'm going to skip this and say, what do we need to do? I think we need to collectively strongly advocate. We have transitioned now from being just the diagnosticians. Once diagnostics is democratized, we must think about the downstream flood of patients who will be waiting for these therapies. There's no point in investing, millions of rupees in sequencers and, diagnostics if you do not do something about the downstream effects of what will happen to, when those cancer therapeutic drugs are needed, when those gene therapy drugs are needed for the patient. So we need to do that. And we need to start that conversation today with the pharma industry, with our health ministries, with our philanthropic organizations, with everyone. So, this is the sort of the call to all of you who are, asking, what specialty do I choose, right? What do I do? What do I study? What do I dedicate my life to? This is the time right now. We're sort of at the cusp. The advances in bioinformatics, in artificial analytics have grown so much that biology and machine learning are now meeting each other. And this is the time where we can actually now help most of our patients. The cost of the first human genome to be sequenced was $3 billion. It took 13 years to do that. And today, you can do this within a few hours for a few $100s. And this is just, 20, 30 years of progress. So many, many more years. You can now sequence your genome with a device that is the size of a USB stick. All of the computing happens on the cloud. You just drop a blood on there, take this to the field. Imagine doing this in rural Sindh or rural Balochistan on communities that will never, ever see a hospital, right? You can do that today. So what you really have to do is take your knowledge from the clinics to the communities, from the labs to the communities. You have to unlock the door of this knowledge because that's where all the patients are sitting. Most of the patients who need us can't come to us. So I'll end by saying, this is sort of our call to duty that whatever we do, whatever we pursue must have a high impact, must be of the highest quality. We cannot compromise on quality. It must be relevant to our population. Study our diseases, study our variants, get that knowledge out there. And at the end, do that to improve access to people who need that care. So in summary, medical genetics has evolved rapidly, advanced rapidly over the years. I mean, it's a phenomenal field. So if you're, wanting to partake of an exciting career, this is what you want to do. And you must keep up. Practicing clinicians, the practicing lab people must keep up with what's happening in the world. And currently, the solutions are expensive, but just like 30 years ago, it took an act of God to get an MRI scan on a patient, and now you can get it daily. This will also change, and we must change it together. And at the end of the day, all of these things sound really good. We've given gene therapy, we've given ASOs, but until you work on prevention, this flood will continue to grow, and we have to prevent. And we're going to have a very good conversation later on today on how to prevent these diseases and so forth. So thank you very much, everyone. Thanks for your attention. Thanks to the online audience, and thank you. We, I guess, have a few minutes for questions, or are we doing questions at the end? Okay. You want to just quickly take a look at the schedule and see, or we can move on to the next talk. Okay. So, okay. Does anyone in the audience have any questions? Okay. So Dr. Adnan has a question. Adnan, please speak in the mic. Thank you.
Thank you very much, first of all, for a wonderful presentation. I joined late, but really very, very interesting. I have one ethical question over here.
Yeah.
Of course, there's an immense advance in getting the diagnosis and the treatments, but what do you think of being known for a patient, a full genomic workup on a USB stick and telling him that you have this, and this?
Yeah. So what you're talking about is predictive
Yes.
genomics. And okay, great. Thanks.
How is impacting in America? I heard a few that I want to just give the people a taste of that question.
Right. Thank you. So Dr. Adnan is a neonatologist, and he takes care of our sickest and littlest babies. The question really revolves around if people had their genome on a stick today, how useful would that be? And that's a very good question to ask, because we must be very open to this. Just because the knowledge is there doesn't mean it's always helpful. It may actually harm. You know, there may be information there that may predict something that is quite devastating, that psychologically can be very, very challenging for someone to deal with. So I think it's really, number one, genetic counseling is the key over here. Before you start doing genomes on people, even looking at a single gene on a person, you have to counsel them before. And that's very important. And that really segues very nicely, because Fizza is going to talk about the importance of genetic counseling here. And you know, this information cannot just be digested by a few genetic professionals sitting at a few institutions, right? This information is useless without all the clinicians who will take care of the patient being able to understand it. So I think personally, having the genome on the stick right now is not our priority. Our priority is to educate people about the stuff we can do something about, the stuff that is actionable, right? And as knowledge increases and as awareness increases, that's when you expand that knowledge elsewhere. That's my perspective. It's a very personal perspective. There may be people who say, the knowledge will build it and they will come. First, get the knowledge out there and the knowledge base will increase. And that's an important philosophy too. But I think you have to be very careful with genetic knowledge, because once you know it, you cannot unknow it. So that's very important.
Sir, we have an online question from our online
Sure.
audience.
That'll be the last one before we go to the next talk then. Thank you.
Okay,
Yeah.
Yaya is asking that you talked about therapy in SMA and Duchenne, but what is development in retinitis pigmentosa?
Good question, because retinitis pigmentosa is very common in Pakistan. We see that a lot. So currently there is one drug for one gene. The drug is named Luxturna, and it's for a gene called RPE65, and that causes a condition called Leber congenital amaurosis. There are about 5 or 6 genes for which therapy is just on the cusp. They're in phase 3 trials with good results. And we hope that over the next few months to years, those therapies will come. But for the majority of patients out there, the drugs are still under development. But great question. Thank you. So I think with that, I'll take my leave. And then I think, Maha, you can come in and introduce the next speaker. Thank you very much, everyone.
Thank you so much, Dr. Kirmani, for your enlightening talk. Next, with great pleasure, I'd like to introduce you to our next speaker, Ms. Fizza Akbar. She's a genetic counselor and senior instructor at Aga Khan University, and she has a master's in genomic medicine from University of Manchester. Please join in me welcoming to the stage Ms. Fizza Akbar.
Assalamu alaikum, everyone. I am extremely thrilled and a bit nervous to be speaking at this platform today. And it's never easy to come and speak right after Dr. Salman. So I will try to do justice to the topic that I'll be talking about. I would also like to thank Myla and Aisha for giving us a brief intro to genetic counseling and making my job easy as we go towards my talk. So I've got no disclosures. I'm sorry about my throat, so I might take a second and sip on some water and then we'll proceed. But I'll try to keep it way in the time that I've been allotted. I've got no disclosures, and this is going to be the outline of the talk, and we'll want to go stepwise about understanding what really the process of genetic counseling is. So before we go in, let's just talk briefly about how the term was coined and where did it all begin. It was back in the 20th century when an English biologist actually suggested that the new medical and biological study of heredity we call genetics. It wasn't until 1947 that American geneticist and a biologist coined the term and had published a book on counseling in genetics. The first formal definition of genetic counseling, however, was published by the American Society of Medical Genetics, American Society of Human Genetics back in 1975, and the National Society of Genetic Counselors defined genetic counseling as a process of helping people understand and adapt to the medical, psychological, familial implications of genetic contributions to disease. The process integrates the interpretation of family and medical histories to assess the chance of disease occurrence and recurrence, education about inheritance, testing, management, prevention, resources, and research, and counseling to promote informed decision-making and adaptation to the risk of a condition. However, it's very important for us to understand the importance of being earnest when we talk about genetic counseling. So it's very important for us to be unbiased, truthful, reliable, communicative, accountable, and do our work with utmost honesty. And these really are the guiding principles of ethics in genetic counseling, where we need to understand that the patient or the family has the autonomy. We have to involve them in telling the truth and respecting confidentiality. So they have the right to the disclosure of the information about their diagnosis. We have to make sure that we do good, be kind, and avoid any harm. And in cases where a harm cannot be avoided, to minimize the harm so that the people who are approaching a genetic service can feel protected and reach out to the genetic services. And we need to treat everyone equally and with justice so that they deserve to get the all the information as well as the diagnosis that they deserve to know. So the National Society of Genetic Counseling has a very good resource where the code of ethics can be understood and learned in further detail. There's a link below. I'm not going to go into the details of this, but that's something that the audience might be interested in. So let's see where the genetic counselors are embedded. So they work in interdisciplinary healthcare systems where they work very closely with clinical as well as laboratory geneticists and with non-genetic specialist doctors. So it has to come as a team for all of us to actually convey and give benefits that the field of genomic medicine promises for not only one individual, but the families, as Dr. Salman also said. And these are the different settings for genetic counseling. We talked about how every organ has DNA and every organ deserves a genetic counselor. So that really boils down to the fact that essentially every field could have their own genetic counselor when we talk about genetic counselors in clinical settings. However, they are not only in the clinical setting, but they are also involved in a lot of research, education, and policy making. So genetic counselors are clinical as well as non-clinical genetic scientists and experts who benefit the society in many ways, and they wear many hats. But now as we change the gears back to clinical genetics, we need to understand, and there are many common indications for referral to a genetics clinic. However, I'll just go on to discussing just a few, which would include a family history of hereditary condition, a couple coming in with a previous child with congenital anomalies or birth defects, them also coming in with recurrent pregnancy loss, antenatal anomaly scans, follow for a positive newborn screening test. This is very pertinent to inborn errors of metabolism. And for conditions, for patients when they are diagnosed with a genetic condition or are suspected to having a hereditary syndrome, for example, hereditary cancer syndrome in adults, so on and so forth. And it's very important and very pertinent to the part of the world that we practice in, where consanguinity and a suspicion of a familial condition is a big indication for a genetics referral. So there are many steps that go in a genetic counseling session, and these are just briefly some take-home points about what steps are taken in a genetic counseling session. So Dr. Salman talked about getting a diagnosis, ending a diagnostic odyssey, and that is very essential when a family has been going in and out of different doctors' clinics without knowing what their child has. So years and years of investigations and still not knowing what it is. So establishing and confirming a diagnosis is very important. A lot of times, we might feel that diagnosis is not going to help if there is no treatment, but the closure and that information that they get from that the information that they get is very essential. It's very important for us to actually also offer risk assessment for an inherited condition for other family members in the family, so that that can be prevented, and discussing the options of genetic specific management. For example, qualification for modulator therapies for a certain pathogenic variant that a child with cystic fibrosis has been identified with, or a breast cancer patient who's BRCA positive and can be treated with PARP inhibitors. So that's a very exciting time to be in genetics. And it's very, very important that we effectively communicate and support our patients. And that becomes challenging when we would have patients coming from different socioeconomic classes, different levels of education and literacy. So it's really our job to make sure that everyone deserves the equal amount of information, and we make sure that we go that extra step, that extra mile to help them understand the information that we are trying to convey to them. So the goal really is to enable the patient with all the information and give them the autonomy to make the informed decisions. So they would understand and get a diagnosis to end a diagnostic odyssey, understand the medical diagnosis as well as the implementation and implications in terms of prognosis and possible treatment. And we are living in an exciting time where sometimes something might not be readily available, but there may be clinical trials going on. So the patients can be directed to that without giving them a lot of hope, but keeping it realistic, as well as helping them understand that things may change in future. The mode of inheritance becomes extremely, extremely important to understand, so that it can be, the risk can be prevented in the other people in the family, and so on and so forth. The options available for dealing with the associated risk, and it would be different for different conditions. So for couples coming in with a child with a genetic disorder, giving them the options for prenatal diagnosis or medical surveillance. For example, a child has been diagnosed with NF1. We have a standard protocol for following the child through different medical management and surveillance. So we would offer them all that is relevant in a certain situation. So genetic counseling consultations can be divided across different sessions as per the need of a certain patient and a situation. And at large, they are themed around pre and post genetic test counseling sessions. In the pre-test genetic counseling session, it becomes very, very important for us to take a complete family and medical history, verify the information as possible. A lot of times the patients would come in with certain biases, so we need to really dig in and get the most accurate information out of them, assess risks of disease, mode of inheritance, delayed onset and gene penetrance. It really explain them that what could be the result implications and outcome possibilities. A lot of times families would come to us and say, oh, our primary doctor has told us that you have to come and see the genetics team and get the genetic test done. And we ask them, do you know what is it going to get you? And they're like, no, they just said, go to them, get the test. So we have to really boil it down and break it down for them that sometimes we might not, the information is great, but we might not be able to give your child a treatment based on that information. Or sometimes when they are spending a lot of time and effort in making arrangements for the finances for a test, we might not get an answer altogether, because the knowledge of genetics is ever evolving. We may get a variant of a certain significance which has no clinical implications, so on and so forth. And it's very, very important for us to provide them with the opportunity of asking questions. And in our part of the world, when we say, oh, do you have any questions? Or you are very quiet, I would really like to hear about your thoughts on this. They sometimes get a little, shocked that why are we interested in hearing what they have to say? So we really try to make sure that they ask the questions and understand what we are trying to convey. Because sometimes we would feel that, oh, we are doing a really great job, but it's of no point if we haven't really conveyed what we are trying to convey. And then taking consent for testing and to sharing it with relevant healthcare providers so an optimal healthcare can be provided to not only the patient, but if later to affected or pre-symptomatic family members who test positive for a certain result. So in the post-test counseling, no-brainer there, we give the results and explain what that really means for the patient and their family in the bigger scheme of things. And at present, we provide them with emotional support because it's very hard to be living with a diagnosis that's of a hereditary nature, and it's something that will go with them for a really long time. And this is a conversation that we sometimes have where we say, do you really want to get the test when, you know, we are counseling asymptomatic individuals whose relative is BRCA positive, and they say, oh, it's a simple blood test, there's no harm in it. But then we go on to say that it's not like a diabetes test where, you know, you modify how you eat and you repeat the test and it's better, or a cholesterol test. It's an information that you were born with and you're going to live with it for the rest of your life. So we really need to make them understand what they are signing up for. Offer cascade screening where possible. And most of the time, most of the families are very open to the idea of having their family members get tested. Thus, we have the opportunity to establishing long-term relations with not only our patients, but also their family members. And then we take care of families, direct them to subspecialty doctors who can take care of them in the most optimal fashion. Now I would want to take a few minutes and talk about what really happens inside a genetics consultation. And some of the points that I would want to highlight is there's a lot of recall bias when we are taking a family history or a medical history from a family. They usually assume a diagnosis and have an incomplete knowledge of medical history. So it's very, very important for us to verify the information, ask information for both sides of the family. So there's another interesting fact that culturally, we feel that we belong to our father's side of the family. So that's my family, but my mother's side of the family is her family. So sometimes sometimes I would ask that, oh, is there any certain diseases in the family? So they would say, हमारी फैमिली में तो नहीं है, लेकिन अम्मी के फलाने रिश्तेदार में. And then you kind of go down that socially or culturally you would feel that, oh, since, you know, we take our dad's name and that's the surname that we go by, people would say that our family is the dad side of the family, but genetically it's 50-50. So we have to tell them that, oh, both sides of the family is really important. And the flip side of it is sometimes they would say, oh, there are no cancers in the dad side of the family. And why is it even? There's no family history of cancers in my mother's side of the family, but there are cancers in my dad's side of the family. But why is it important? Because breast cancer risk comes from the mom. So these are really some of the biases that we have to debunk and make sure that we are taking family histories which are most accurately taken from both sides of the family. And while we want to focus on the condition for the primary referral, most of the times we also end up understanding and knowing about other conditions which may be significant or hereditary in nature. So we have to talk about those, but also refocus on the primary discussion points. Again, we know that this is what we would do: state the risk information, be unbiased, be honest, be very objective, take our own biases out of the equation while we counsel the patients, because everyone deserves the right to the information equally. And this is something that I had learned very actively with Dr. Salman when I had started. I walked in a clinic to counsel a patient, and I saw that they seemed to have coming from a rudimentary background, and I didn't do a very detailed counseling with them. And then I came back and told him about the patient, and then he said that, did you do steps? And I said, I thought they wouldn't understand, and I kind of cut it short. And then that was a big learning lesson from me. I do not get to decide who deserves that information. Everyone deserves the same kind of information. For someone who do not have the literacy level as you do, you have to make it easier for them. So it's our job to do that extra step and make sure that they understand it. So this is what we try to do when we see people coming from different cultures, different socioeconomic backgrounds also. While we also want to make sure that the patient can express themselves and be heard, it's very important to also balance the informational value of the content of this counseling session and the emotional content of the session, because we need to understand that genetic counseling is not therapeutic counseling. And if our patients require additional support, we can refer them to psychologists or psychiatry specialists who can help them support with the emotional support that they need after they have received a diagnosis. Again, breaking it down is very, very important. We use different analogies, and as we go along, I would want to share a few analogies that we use, and we help each other while coming with those analogies. And sometimes we make them up on the go as well. It's very important for us to prepare the patient for the possible result outcomes, a positive result, a variant of uncertain significance, or a negative result. And it's very important for the patients to also understand that a variant may reclassify in the future, so we have to see them back and also give them the options of further testing as and once they are available. So very important for us to understand that what have they really gotten out of what we are trying to tell them. Is it all bad news? Or is it, and what does the patient really understand? So I would say that from an oncology genetics clinic standpoint, we have seen people being diagnosed with BRCA1, and then they would start crying as if this is a death sentence. And then we have to take that step back and tell them that, no, this is not what it means. It means that, this is a certain gene which has a disruption. And now you are prone to XYZ cancers. And this is how we are going to make sure that you are getting the optimal treatment and later on surveillance. So we sometimes talk about this being with like a weather forecast, that we are doing a health weather forecast, then what can you develop in future? So this is what, and then obviously giving them the right options, directing them to the subspecialty doctors who can take care of them in a very holistic fashion. So genetics providers would work in multidisciplinary teams so that we can give a holistic care to our patients. And then we need to also support the patient to share the results with the at-risk relatives. They have the full disclosure to the information that they have. In situations where disclosing that information may do some harm to them, they may choose not to do that, and we would support them with their choices. Thus, we are very lucky to make a long-term connection and relationship with our patients and their families, and they would come back to us at different milestones in their lives so that we can integrate the utility of genetics and genomics for not only themselves, but their children. Again, effective communication is the key, and we cannot deny the fact that we do live with some challenges practicing genetics here, but it's equally gratifying when we understand that the people really could gauge what we are trying to convey to them. We have to make sure that genetic counseling is non-directive and non-judgmental. A lot of times when we are taking family histories, a couple with a child with a suspected genetic disorder, we obviously ask about consanguinity, and this is a standard way. We don't frown upon them or have any judgmental way of asking that. But since they have come through a lot of judgments, they would just say, yes, we are cousins, and this is why our child has it. So we have to really give them the support and help them understand why this may have led to an increased risk of disease without being judgmental. And I personally may have some biases, but I cannot bring that in when I'm with the patient in the clinic. So that has to really be taken care of. yes, we want to make them aware of all the options that they have, so that they can make an autonomous decision. And sometimes the patients don't even like that. They're like, we have paid for your consultation fee. We are here for you to tell us what to do. And then, we kind of help them. We do some hand holding because that's what they want. And it's our job to support them also while staying true to the principles of genetic counseling. So we try to make sure that we do things and while staying true to what genetic counseling principles are. And here are really some cultural challenges that we face day in and day out. It's very important sometimes it's very hard for families to understand what a carrier is. If we say that, oh, yes, your child has been diagnosed with a genetic disorder, they would say, how come? Because we have no family history of disease. So it's very important for us to really break it down for them. We live in somewhat a patriarchal society, so a lot of times the blame does come to the woman about giving birth to a child who has a genetic disease. And then that makes us, it makes it very challenging for counseling for X-linked conditions. But we try and do the best because, again, while we are trying to preserve the family integrity, we also have the onus on us to be honest and give all the information that's there. But we do it in a manner where no harm is being done in the families. There's a lot of stigma around carrying the bad genes. I was meeting this family just recently where there were two young women with breast cancer, and they had come with their aunt, and who never got diagnosed with any cancers, but she had brought in her 18 plus daughters to get to have a consultation about cascade screening. And she teared up by the end of the consultation saying that even without a genetic test, people say that, so her sisters had breast cancer, her mom had died of breast cancer, that we are not going to marry in this family because all the women in this family, they end up having cancers. And I was really taken aback because a lot of times you're talking about, asymptomatic women carrying BRCA gene and then telling them that they have the right to the disclosure of their information. But here I was sitting, and this is what they had come up with even before the girls got tested. So this is really challenging, and sometimes we kind of have to really make sure that we are doing our jobs right, and it's a lot of responsibility, really. But then we have to take the burden off them, say that none of us control what our genetic makeup is. And I even told her that, your daughters, when they get married, they don't they continue to do their screening, but they don't have to tell. For example, if I have a family history of type 2 diabetes, I may start taking care of my health, but I'm essentially not telling my partner that, oh, I might develop type 2 diabetes. But when we talk about genetic disorders or hereditary conditions, there's a lot of stigma around it. So we really have to debunk it and make them understand that, it may be seen as just another condition that we are very okay seeing and not even having any trouble with in terms of, talking about that. So when we talk about young couples coming in with having children with a genetic disorder, they really, really want to have a biological child because people are not open to the idea of adoptions in our part of the world. And most of the times when a couple is coming in, they struggle to understand the difference between having a prenatal diagnosis versus a treatment. So they might think that during the consultation, we might identify something in the fetus and treat it so that the child doesn't have, the child that is born does not have the disease. So that is expectation setting has to, again, be reoriented so that they really understand what they are getting out of a test. And most of the times, even after they have gotten the test done in a prenatal setting, they might have reservations around termination of pregnancy. I'm going to not go into details of each point, but here we also need to make sure that we need to really realize that while we're talking about, the governing principles of genetic counseling and how important it is, sometimes the families would understand the need for a test for their child or their family members, but they do not have the financial means to really go and do the test. And that is very saddening, where you know that they see what you're trying to tell them and the utility in what you do, but they just have that financial barrier that they can't really go in and do the test. It's also important for us to know that, again, stigma, we talked about, young women being positive for a hereditary cancer gene. They would feel that there's a lot of stigma for them to get married. So we really need to help them understand that, it is not as bad as it is in their head. We do not have any direct translations for medical terms in Urdu, so that sometimes, a lot of times, we feel that things may be lost in translation. And patients expect for us to give them the direction in a non-directive genetic counseling manner. So this is how the medical system, the patient-provider relationship works in our part of the world, that adds to the challenge of practicing genetic counseling in our part of the world. And we really need to understand also that we need to educate our people, not only the masses, but our medical community to be educating their patient masses in non-judgmental ways, particularly when it comes about the risk of consanguinity and risk of an autosomal recessive disease. But with all the challenges, there are certainly a lot of opportunities. Going that extra mile always helps. And it's heartening to see when the families understand. They would travel and connect through remote areas to get the information. There are couples who have had affected children. They would want their siblings to get tested so that their children would not have a genetic disorder. So people, the collective family system in our family, in our part of the world, sometimes is a barrier, but most of the times is not. And I was very happily surprised when I had understood how the family dynamics really helps. So sometimes when a patient is test positive for a gene, for example, a hereditary cancer gene, and she's going through a lot, but she is accompanied by her sister. So I would say, oh, now with your sister's consent, if she agrees for this information to go to your other siblings and family members, you are now the person who's in charge of conveying this information and bringing the family members back to the clinic. So that we really should also cherish the things that practicing genetic counseling in our part of the world also brings. It's very important for us to take away the stigma and the burden off their shoulders. So the example that I really talked about makes us understand how we make them understand biologically what is going on. Things are not in their control. If I test today, I am maybe carriers for different disorders. So it's something that the biology chooses for us. It's not on us. And take away that burden from them. Drawing chromosomes and showing pictures really helps. Sometimes we would even, give out that drawing to them so that they can, go home and educate their family members or look back at it and understand what we really meant in the clinic. Using analogies is really helpful. I am going to try and wrap up in the next couple of minutes because I'm running out of time. This diagram is very crucial and helps understanding when, the couples would come in who are cousins and would say, how is it that us being couples is increasing the risk of my child with a disorder? So you can go on and say that, oh, as a first cousin, you share 12.5% of your DNA. And that increases the likelihood for the both of you to be the carriers of the same gene. So we have to take away the bias and the judgment and just talk about purely the science of things in a culturally sensitive manner so that they could really understand rather than get aversive from what we are trying to tell them. This again is a good example that we can look at genetics as a cookbook. And if a recipe goes wrong, then the final dish is not what you had wished for. And some analogies is that we can practice is talk about Maurusi Juz or Geneati Kitab, talking about genes as components of a recipe and the final product being our health, which may be disrupted because of the fact that there was a change in the recipe and things did not go the way they were supposed to go. Explaining the concept of carriers and autosomal recessive disorders. So this example actually Aisha helped me with, and I never saw it that way. So she had said that this could be actually, we can use two abstract ideas of two organs that work together. For example, as a carrier, we can think of two sets of eyes. If one eye is not functioning, a sense of sight is still not fully lost. So this is exactly how carriers are. When out of two chromosomes, one has the mutation or a pathogenic variant, they're functioning fine. But if the combination of two faulty copies go into the child, both eyes would stop functioning and this sense of sight is lost, or the function of a particular gene is lost and translating in a disease. So that was a good way of putting it. This is another analogy that we use, explaining pathogenic and likely pathogenic variants against variants of uncertain significance. So this may be a very familiar sight. A lot of people who made their way today crossed through the roads like these in the morning. Here you can think that I am a driver and a gene is a big road. And variations are different puddles, cracks, and bumps in the road. So some puddles or cracks in the road may cause an accident. That's the pathogenic variant, right? And sometimes we don't know if a puddle or a crack will or will not lead to an accident. So that's the variant of uncertain significance. And I get very happy when first I show them the report and then they're like, what are you even talking about? And then I say, think of a gene as a road and then the accident, and then they understand. So that the gratification you get when they understand it is out of this world. And then there's one example that one can use in explaining high, moderate, and low risk genes. So we can think of it as hereditary cancer risk genes. And we can use an example of an earthquake. The epicenter or the location is the organ that's the most impacted. And the high risk gene or a high scale earthquake would do more damage comparing with a low or moderate risk gene. So this is how we could be a way of making your patients understand how a different result in a different gene would impact your management differently. So I would like to really conclude my talk here. And I and Dr. Salman didn't exchange notes on this one. So some of these are repetitions, and which just goes on to say that different minds are thinking alike. So access to genetic counseling is a need and it's not a luxury. We really need to stop thinking that. With challenges, there's a lot of opportunity in practicing, opportunities and gratifications of practicing genetic counseling in our part of the world. We really need to work our way up, and this platform is a very good way of educating our medical community as well as our masses. It's essential for us to build that genetic literacy. Mainstreaming genetics and understanding its need in the patient journey is extremely crucial. And while we are at it, it's a tremendously gratifying field and ever-evolving field of work. So anyone, be a clinician or a scientist, being a part of a genomics workforce is a thrilling and exciting career pathway for you to choose. Thank you very much. I would like to acknowledge PSMG, Dr. Natasha Anwar, and Dr. Salman Kirmani, who had worked as my mentors, my colleagues, patients, and their families. Thank you very much. Oh, perfect.
So first of all, it was a really good talk, Ms. Fizza. My question is, in general, you mentioned the stigma that's often associated with genetic conditions. And so my question is, a sort of extension of that point where with stigma, there can also sometimes be
dangers in terms of, maybe psychological or even physical violence, particularly, and when the role of blame can also come in for a certain parent. So my question was that what can we do in terms of counseling, and also as a physician, um, can we take certain measures to assure the security of not only, let's say, the child, but also their parents from any sort of psychological or even physical abuse that they might encounter in this case? And then just a second part to this is that how often do you see problems like these in your, practice?
Right. Thank you for this very important question. There's the stigma that we talk about a lot of times. They may walk into the genetics clinic with that assumed stigma, and once you have that session with them, you debunk a lot of their concepts, which are misconceptions. So that stigma at that one stage is also taken care of. So that happens. Sometimes, there could be husbands who would say, oh, if I marry someone else, would I have a healthy child? And then you obviously have to say that that's not necessarily the case. And when it comes to psychological stigma, sometimes we have teenagers or children who would have a genetic diagnosis, and we would have summary letters written to their schools for them to understand that this is a certain condition that the child has. However, just for example, if a Marfan syndrome child it has been diagnosed with Marfan syndrome, so they will have to take care when it comes to participating in certain physically stringent physically challenging or rigorous exercises. However, they are generally fine otherwise. So we will have to make that communication go from our end and make sure that the education systems understand. And if there's a lot of psychological stigma, that again, and they need emotional support, we are trying to so they have been a lot of young individuals who are positive for a cancer predisposition gene, and they feel that we need to have support groups for them to actually all come together and talk about what they feel. So that's one thing that we haven't been doing yet, but that's something that we can do. Yes.
My question is that what you highlighted and what we believe in also is more need for genetic counselors and people like you and Salman we want to replicate and clone or whatever you call it. But one of the biggest hurdle we see is that when young graduates looking for that type of career which you have adopted in Pakistan, I don't think there's anyone else. Maybe probably some people do it without proper training, but that's what it is. But how do you get reimbursed, and how can these young people go towards a career where the patients are not supporting it or paying for it? In the United States, for example, genetic counseling is supported by insurances, most of the insurances. But here, insurance is not as often.
Yeah.
So what is your advice? How do they go towards genetic counseling career with an aspect that they can be compensated for their services?
So that's a good question. And I think the institutions and organizations have to become inclusive of people with these degrees, because as a biology graduate, I always wanted to do genetics. I did not know how to do it. But I took the career pathway, things happened for me. But again, a lot of times when we choose a career, we talk about, how we are going to, support myself financially with a degree or a career pathway. So training programs like what AKU and Dr. Salman Kirmani had done for me, and then, we have a genetic counseling trainee, Maruk, I would feel that the organizations and big tertiary healthcare systems will have to come forward and actually advocate for the need, so that if I am going out and, maybe I go back to my alma mater and talk about, how I did it, they might say, oh, what are the job openings? Why would I go into doing something? Because I know for a fact that there are a lot of biology majors who would love to have a career in genetics, but along the path they would deviate because there are no jobs or, there's no career pathway. So we'll have to really advocate for that.
And
And
more answers will come in the panel discussion we will have at the end of the day. He was asking your salary, which you very tactfully did not disclose.
Oh, you did?
Question up there.
Oh,
Okay, so my name is Dr. Rutaba, and I'm currently working as a geneticist at Al Shifa Eye Trust Hospital, Rawalpindi. So I came all the way from Rawalpindi to here to Karachi yesterday. Being a geneticist, I faced a lot of troubles, First thing, the religious beliefs, because I'm working in a very remote area and we are having a lot of bulk of patients from very, very remote areas. So the religious belief, first problem. Second problem about the complex genetic disorders, like glaucoma, which can be inherited as autosomal recessive, autosomal dominant, X-linked. And fourth, the problem, you're talking about truth, being truthful in genetic counseling. How could be you truthful in dealing with X-linked disorders? I have seen divorce cases in that case. So I'm avoiding being completely truthful in X-linked inheritance,
those are really tough ones. It has been a while I've seen an X-linked condition. Dr. Suman, do you have any recent recollections of how you did it?
So I think a very good question. And I think the way you say it is that you go out of your way to say that it is not the woman's fault. Like I do for Down syndrome, right? Now, if you start by saying, okay, 99% of the time it comes from the egg, there's no point in saying that. So you say, okay, the error occurs in the egg, but it could have occurred in the sperm also. So you kind of have to balance it and you have to read the family dynamics. You don't ever want to be in a position where you are not completely honest and truthful, but you can do it in a sensitive way. And I think you have to somehow learn that process over time. It can't be taught in books. But thank you for that point. Last question, I think, before our break.
Thank you very much for an excellent lecture. What I feel over here, that we are talking about the genes, we're talking about the molecules, but we forget sometimes that we are human beings and spiritual beings also. So the spiritual aspect can calm down the situation that if you are getting some mutation or some hereditary disease. Yes, sorry, I'm really sorry.
Yeah. So I want to ask about that. There can be a spiritual aspect that you can calm down the patient. So there should be spiritual support to the patient. For example, if I'm teaching my students, I say to them, we have, in Quran, you have got signs for you within yourself. So I say to them, if you study the normal, say Alhamdulillah. And if you study abnormality, you should say Alhamdulillah, I'm not, Subhanallah for the first thing, and Alhamdulillah that I'm not chosen for this thing, for this type of sign. So all goes back to Creator, and we as a Muslim should have, more support from the spiritual aspect, from the religion, rather than having hurdles, as she was mentioning. So in accepting the science and medicine. So what do you say about that?
And that's a very good point. And we usually do talk about that. Okay, naturally, all of us can be a carrier for a certain thing, that I may be a carrier for an X-linked condition and I don't control it, right? So sometimes we do say that from what we know of science, we can diagnose it right now, but we don't know how it happened, because they would say, why did it happen in the first place, right? So we do bring in that part where, the faith comes in. The other aspect of faith is sometimes they would say, Allah Malik hai, we don't want to get tested, aage ka dekhenge, right? And then you kind of have to make it turn around the faith also, that Allah also has told us that Allah has given us those resources for them to be sitting here and being able to afford all the testing and investigation, right? And Allah wants you to, the knowledge that we get is also what Allah wants us to strive for. So that knowledge is also you being grateful to Allah and then making the way out of it, because Allah has given us,
That's right. I was just talking about where there is, no disease or no outcome, there is no possibility for them to be cured. So that is at that time.
Oh, yes.
In addition to the psychological support, there should be spiritual support.
Yes,
That is more effective.
we do talk about it in a sentence or two, but yes, not officially. We don't refer them to a spiritual healer, I would say.
Okay.
Yeah, and we will have a panel discussion and an interactive discussion on this topic like later on in the session. So we'll take all the questions from our in-person and online participants. And there, it's a time for a very brief 15-minute tea break. So please enjoy the refreshments served outside. Just keep in mind, don't bring them inside. And the restrooms are in the left corner of the quad. The presenters are requested to stay with their posters, and I would encourage the audience to go engage with the presenters and Johanna, enjoy the posters as well. We'll be back at 11:15, so please be Johanna mindful of the time and return promptly so we can resume. Thank you so much.
And thank you to all of you. It's wonderful. There's about 60 people here in the room and about 31 online. So thank you for making this a success, and stay through the day. This was just the tip of the iceberg. The best is yet to come. Thank you.
Ladies and gentlemen, please settle yourself as we are resuming our session again with a virtual talk by an expert in the field. Despite the geographical distance that may separate us, technology allows us to connect and learn from experts around the globe. Our virtual speaker today is Ambreen Khan, a licensed board-certified genetic counselor and worked as an oncology genetic counselor at Hertzman Cancer Institute in Salt Lake City, UT. And her virtual talk will focus on prenatal genetics, providing us with valuable perspectives and knowledge that align with the core themes of our conference. Please.
Saima, the audio is not there. Please check.
Prenatal genetics. I am a genetic counselor. I work at Intermountain Healthcare in Salt Lake City, Utah, in the United States. I do not have any conflicts of interest to declare. And I would like to thank Dr. Ajaz Ahmed as well as genetic counselor Katie Stoll for their help with these slides. So today's learning objectives are to discuss the use of prenatal genetic counseling in pre and post-test settings, as well as identify best practices regarding prenatal genetic screening and diagnostic testing. So what is prenatal counseling? When a couple seeks information regarding the chance of having an abnormal outcome of a pregnancy, whether that's future pregnancy, so preconception, or current pregnancy, which would be post-conception. It is provided by certified genetic counselors when they're available, and there's oversight by medical geneticists as well. Genetic counselors usually work closely with OB/GYN doctors who provide such care, and there's a lot of different reasons why such help is sought out. So pre-test genetic counseling is when genetic counseling is done before the actual test is completed. And pre-test counseling includes information on the types of results a person can anticipate, as well as the risks, limitations, and benefits of testing are provided to the patient before performing that genetic test. Patients at this point should have the option to either decline the testing or accept the testing. And obstetricians and gynecologists and other healthcare providers to determine which test will be offered as a standard in their practice, so that similar testing strategies are made available to all patients. And post-genetic counseling, so if a genetic test reveals that there is a clinically significant mutation with heritable potential, or there is a risk for the patient, then the patient should also be encouraged to share those results with affected or at-risk family members. If OB/GYN or other healthcare providers do not have the necessary knowledge or expertise in genetics to counsel a patient appropriately, then a referral to a genetic counselor or a medical or gynecological oncologist or maternal fetal medicine specialist or any other genetic specialist should be considered. So what are some of the common indications for genetic counseling during pregnancy or during prenatal care? For preconception counseling, so assessing what risk somebody has, it could include things like a family history of a genetic condition, personal history of a condition, teratogenic exposure, so exposure to certain drugs or medications that could increase risk for birth defects, advanced maternal age, advanced paternal age, multiple miscarriages in the history, as well as previous abnormal pregnancies and consanguinity, which is when there's cousin marriages. And then post-conception counseling would include family history, previous abnormal pregnancy, if somebody needs pre-implantation genetic testing, so before the embryo is planted, if they want to test the embryo, if there's a personal history of any condition in either parent, if someone has abnormal prenatal screening results, and then advanced parental age again. So when we think of advanced parental age, specifically thinking about each set of both maternal and paternal age and how that affects pregnancy. So in advanced maternal age, there is aneuploidy risk, so risk for a fetus to be born with an increased risk for trisomy 21 or Down syndrome, and then also other chromosomal abnormalities. And as you can see on this chart, the risk for these chromosomal abnormalities increase as a person gets older. So between the age of 30 to 35, that risk is increased, but then as a person reaches age 40 or 45, the risk is much more higher. So how the age 35 cutoff was decided for aneuploidy risk is the National Institutes of Health based that risk off of two factors: the risk for amniocentesis to cause miscarriage, as well as cost-benefit analysis suggested that the cost of amniocentesis for women 35 or older would be offset by savings accrued as a result of averting the births of infants with Down syndrome. And advanced paternal age is also a risk factor. So if a father's age is over the age of 40, there is a small chance that there is an increased risk during pregnancy for pregnancy loss, for rare birth defects, autism, schizophrenia, as well as an increased risk for childhood acute lymphoblastic leukemia. And then consanguinity, so when cousin marriages or marriages within people related by blood as close to first or second cousins happen, there is an increased risk for birth defects or congenital anomalies. The population risk for congenital anomalies or birth defects is about 3 to 5%. So out of 100 babies born, 3 to 5 of them may have a chance of having a birth defect. If the marriage is between two cousins, first or second cousins or so on, then there is a 1.7 to 2.8% higher risk in addition to that baseline 3 to 5% risk of having a child with some sort of birth defect or congenital anomaly, mostly because of their chance for autosomal recessive conditions. So here's two different articles. One of them is from 1998 and another one is more recent from 2022. And the first one talks about the prevalence and the demographics of consanguineous marriages in Pakistan. This article mentions that there's about 60% of marriages that were consanguineous, and then over 80% of them were first cousins. And then this more recent article talks about how healthcare providers have actually a responsibility to talk to individuals who are married to their first or second cousins about their well-being, as well as what that means in regards to their risk for example, birth defects in family members or any other reproductive health-related risks. So there's standard prenatal screening versus diagnostic testing, and screening tests are tests that don't diagnose a condition, but they can indicate that there's a higher chance for that condition to happen. There is also an increased risk for false positive results as well as false negative results, and these tests are designed to screen the population at risk. So they're population screening usually as well. And then, so when I say population screening, just to take a step back, that would mean like a carrier screening or things like that, which we'll touch on later. And then people who have abnormal screening tests are offered diagnostic testing. Diagnostic testing is used for more definitive diagnoses and has a higher sensitivity with lower false positive rate. And so common screening tests that are offered in the United States are a complete blood count with special attention to mean corpuscular volume or MCV, non-invasive prenatal testing, ultrasound-based nucleotranslucency testing, first and second trimester biochemical screening, as well as ethnic-based testing, sickle cell anemia testing, spinal muscular atrophy testing, as well as cystic fibrosis carrier screening. So preconception usually is when carrier screening will be done, which means before a child is born, one or both parents may go through a blood test or a saliva test looking to see if they have a risk for a recessive condition that could then be passed down to their offspring if both parents have the same carrier risk for the recessive condition. And so information usually about carrier screening should be provided to every pregnant person. That is a recommendation here in the United States. And this testing is optional, and patients can have the opportunity to deny any and all carrier screening. But if a patient is found to be a carrier, then the father of the baby should be tested. And if both members are carriers, then genetic counseling and prenatal diagnosis should be offered. And people who are carriers of certain recessive conditions should also be talked to about implications for their relatives. And only one carrier screening is needed per person in their lifetime. And so, sorry, my slide went back. What is offered in the United States is carrier screening for specific conditions, but also expanded carrier screening, which looks at a big group of conditions that individuals can be carriers of. And this is offered to majority of the couples that choose to want to get this type of screening. And then post-conception, there's biochemical screening, also known as maternal serum screening. And this was recognized as something that could help indicate risks for birth defects. So in the early 1970s, elevated maternal serum alpha fetoprotein was an indicator for neural tube defects. And then in 1985, the American College of Obstetricians and Gynecologists, or ACOG, issued an alert to its members that maternal serum alpha fetoprotein testing should be offered for screening for open neural tube defects for everyone. And so when biochemical screening is done, these are some of the things that are looked at. So alpha fetoprotein is right here. And then if somebody has a risk for neural tube defect, this is usually elevated. In other conditions like Down syndrome or trisomy 18, this is usually low. And so these biochemical markers can be taken during first or second trimester, depending on which markers we're looking at. And then the NT stands for nuchal translucency. And this can help indicate if there's a risk for any of these conditions during pregnancy. So mean corpuscular volume, MCV, is a blood test, and it measures the average volume of red blood corpuscle, and it is a relatively cheap and easy way to screen for thalassemia. The normal range usually is 80 to 100, and if MCV is below 80, then you screen for iron deficiency anemia with ferritin and iron levels. And then if iron deficiency is ruled out, then you screen the partner for MCV. And then if the partner also has low mean corpuscular volume and iron deficiency is ruled out, then there should be a consideration for working up someone for hemoglobinopathies. So why do screening tests? The choice of screening tests is usually influenced by a desire for individuals to have information before the child or the baby is born. If somebody has prior obstetric history of certain conditions or certain issues, then it could be helpful. If there's a family history of certain conditions, then it could be helpful. If there's multiple fetuses, it can also be helpful depending on the age of the mother. Again, screening tests could help determine if there is a chance for an aneuploidy, or what that means is if there's an individual who may be born with, or a baby who was born with chromosome changes, so like Down syndrome. And then there may or may not be reliable nuchal translucency measurement. The cost of the screening usually is relatively affordable. And then options for pregnancy care or termination for an abnormal diagnostic test result can be determined. So screening test could be the first step to lead to the diagnostic test that can then help determine what type of pregnancy care is needed, or if a parent's desire a termination, that they can do that as well. Usually in ultrasound findings, if somebody has enlarged nuchal translucency, which is usually seen in the back of the neck, it is a darker or translucent area behind the neck, and it has a certain thickness. And if it's too thick, it can indicate issues. So if in the ultrasound you see an enlarged nuchal translucency or an obvious anomaly or a cystic hygroma, then the patient should be offered genetic counseling and diagnostic testing for aneuploidy, as well as follow-up ultrasound to look at the fetal structural abnormalities. So fetal ultrasound can be abnormal and can show multiple different things. It can show skeletal defects, it could show neural tube defects, abnormal kidneys or organs. It could also show abnormal abdominal wall defects, as well as heart defects. This is what the nuchal translucency is. This is what I was talking about. Here is the neck and the face of the child, and then this is this area that is translucent. And this is the measurement. So this is 1 millimeter compared to 3 millimeter. This is when it's thickened. And so basically it's an ultrasound measurement of the fluid-filled space at the back of the neck, fetal neck, and usually it's done at 11 weeks to 2 days to 14 weeks, 2 days gestational age. And then increased nuchal translucency can be associated with increased risk for chromosome abnormalities, heart defects, and other birth defects. So this is how the neural tube defects usually could look like. So there can be anencephaly or the missing skull. There could be spina bifida or opening in the back of the spine. And it could be very minimal, so a small gap, or it could be pretty significant. And then abdominal wall defects would look like this. So again, it could be a small opening, or it could be quite a large opening with a lot of organs outside the abdomen. There's also non-invasive prenatal testing that can determine if someone has a chromosome abnormality. And this is a newer test that's offered to women who are at a higher risk for having a baby with chromosome abnormality. It usually uses fetal DNA that's found in the mother's blood, and it's performed at 10 to 24 weeks. 99% detection rate for trisomy 21 or Down syndrome, and there is a trisomy 18 as well as other common aneuploidies for this test. It can also tell us the sex chromosomes of the fetus, and then if there is an abnormal NIPT result, then diagnostic testing like chronospilic sampling or amniocentesis are recommended. So for non-invasive prenatal testing, it's also called cell-free DNA testing. It looks at the cell-free DNA that comprises approximately 3 to 13% of the total cell-free maternal DNA. And it's primarily derived from the trophoblast cells of the placenta. After delivery, cell-free placental DNA is cleared out from the maternal circulation. So this is why this test is specifically done during pregnancy. Here's an article that goes into more detail about the specific test and when it can be used. And then there are diagnostic tests, so procedures geared towards obtaining fetal tissue for analysis, so chorionic villi sampling, CVS, or amniocentesis. Chronic villus sampling usually is done at between 10 to 14 weeks. It is usually after the sample is taken, chromosome analysis is done. It does not detect neural tube defects, which is when we need that blood test, the AFP that we mentioned before. And then the miscarriage risk is similar to amniocentesis. It is important to rule out maternal cell contamination when you're doing chronospilic sampling. And then amniocentesis takes the fluid, the placental fluid, through a syringe, and the timing is usually 15 to 20 weeks. It is also very good at detecting any chromosome abnormalities. It can detect neural tube defects, and it is very targeted DNA testing, which means if a child, if a parent is a carrier of a certain condition, amniocentesis can help detect, sometimes using chromosome analysis and things like that if the child is at risk for a chromosome abnormality. And then the miscarriage risk is less than 1 in 200. And then diagnostic testing when it is done, so a prenatal sample, whether that's chronic villus sample or it's the fluid from the amniocentesis, it is usually done in terms of what is done with the sample is diagnostic testing. So it could be karyotyping of that, it could be microarray testing, it could be targeted DNA testing or biochemical testing, as well as whole exome sequencing as well. So after a diagnosis is made, you usually, as a genetic counselor, provide psychosocial counseling and support. You give information and explain the outcomes as well as medical management for the member, the patient, and then you explain also what options they have available in terms of pregnancy management. The most important thing is to make sure that patient's autonomy and the parental autonomy to make an informed decision is intact. So we do not tell patients what to do, parents what to do in terms of how to manage their pregnancy. That is a decision for them. We also help counsel and provide recurrence risk for future pregnancies, as well as options in terms of testing that may come with future pregnancies. So with that, that is the end of my presentations. If you have any questions or comments, please feel free to email me. My email is ambreen.khan@imail.org. Thank you.
So we're very glad to have Ms. Ambreen online with us to answer questions of our in-person participants and online participants. So please raise if you have any question, and the organizers will get you with the mic.
Ambreen, thank you very much. That was a very good, comprehensive review on prenatal diagnosis. My comment is that we do now nuchal translucency at 12 weeks, and we check, which is called an intracranial translucency, which is as good a screening test and even better than alpha fetoprotein. I know that in the US, alpha fetoprotein is done, but largely eliminated on the continent because the ultrasound is much more superior.
Amreen, this is Dr. Rizwan Naeem. My question is that what is the current status of prenatal screening by ultrasound versus prenatal screening by biochemical genetics, biochemical markers?
Thank you. Assalamu alaikum, everyone. So I believe, in any pregnancy, they are given options. So people are given options to do biochemical screening, and then ultrasound is routine at usually 10 weeks or 20 weeks. That is kind of the routine procedure in most maternal fetal clinics. And then if there is anything on an ultrasound finding or the maternal serum or the biochemical screening, then the patient is escalated to see more of a specialist. But also, if there's anyone else in the audience who is a maternal fetal specialist, please feel free to answer that.
Does anyone else have a question? Okay.
Hi, Amreen. Thank you. This is Ajaz. Thank you for the excellent talk. I have a question regarding NIPT. So at the moment, NIPT is used for detecting aneuploidies in patients. What do you see the future of NIPT as we go forward in terms of detecting more genetic disorders, even either contiguous gene disorders or even single gene disorders? So has there been any studies in that regard? Thank you.
Thank you, Dr. Ajaz. So currently, there is a lot of, so NIPT or non-invasive prenatal testing, if you think of it as an umbrella term, cell-free DNA testing has been done in other areas as well. For example, cancer, there is some, tumor, as your tumor sheds DNA, that can be tested, and that cell-free DNA testing can be done. So I anticipate in the future, as we continue to learn more and more single gene disorders and how we can diagnose them. Currently, I think cell-free DNA testing is being mostly used in cancer settings when the person already has a tumor. But in terms of extrapolating that towards using it in pregnancy, I think there's definitely potential there to be able to diagnose single gene conditions and things like that using cell-free DNA, since it is fetal DNA that is in the maternal blood. I think I do hesitate to think how, if a mother has a single gene disorder, how that would then translate to using cell-free DNA to diagnose it, because we may already know that the parent has it. It seems like Aisha raised her hand as well, but yeah, that is what my opinion is.
So one more question. Again, this is Dr. Rizwan Naeem. We are working towards that extended carrier screening. You mentioned about US extended carrier screening. Are there any guidelines or anything you come across where that extended carrier screening list can be adopted or found out about Pakistan?
Let me, clarify. So do you mean what is on the expanded carrier screening right now in Pakistan, or what is in the US, and then if that can be used in Pakistan?
Are there any recommended genes to be included in extended screening in Pakistan?
Got it. Okay, thank you. So the list is pretty large. And I think any sort of autosomal recessive conditions, especially because newborn screening is not a common practice in Pakistan, I think it would be very important to include conditions, especially metabolic conditions that need, things like diet modifications to prevent like PKU. So those type of conditions, I think, definitely should be on carrier screening. And then additionally, I think any of the conditions basically currently that we test in the US have the potential to significantly limit somebody's ability if they're born with that recessive condition. But there are also a lot of other conditions on the expanded carrier screening in the US that may not really change a huge deal in terms of medical management. So I think if the medical management is significantly impacted, then those should be included if they're autosomal recessive conditions or X-linked conditions.
Can I ask a question and a comment? Yes, very short. Excellent presentation, Ambreen. I'm Dr. Bosina. I just wanted to add that we are screening thalassemia gene right at the moment. Right now, we are doing antenatal testing in Pakistan in different setups, not in the government setups, but in different setups. We are doing the antenatal screening for thalassemia gene. I just wanted to ask that what do you do for sickle cell screening in US, and what do you then advise later on to the families of sickle cell over there? Thank you so much.
So I do believe sickle cell anemia is part of the expanded carrier screening. But in terms of advising the family for testing itself, that is, I think, again, we give them the option, but we don't tell them what they should or shouldn't do. I think most families do choose to get tested. And then there is a recent breakthrough for sickle cell anemia where there is gene therapy available for it. So I think that would definitely change the landscape of testing. Currently, the gene therapy is very new, so we don't know how it will continue in terms of the outcomes. But I think that will significantly change what is being tested and, in terms of like newborns, if they're tested right away for sickle cell, if the parents haven't gotten carrier screening.
Okay, we'll take last comment from our online participant, Miss Aisha Furqan. Can you hear us, Miss Aisha?
Yes, can you guys hear me?
Yeah, please go ahead.
Okay, first of all, thank you, Ambreen, for staying up so late and answering the questions. Everybody, I just want to acknowledge that it's after midnight for Ambreen and she's still awake, so I just wanted to say thank you. And then I wanted to make a comment about a question that was asked by the audience member about NIPT and the single gene disorders. And so there is one lab in the US, so if I'm not going to name names, but if you Google single gene NIPT testing, there's only one that's going to show up. And there are certain very specific diseases that they can test for, and there are limitations, but it is possible for certain conditions. And then for sickle cell, the question was about how we treat those cases in the US. So because sickle cell is more common in a particular community in the United States, everybody in that community gets special attention earlier on during their pregnancy to at least get the point across and provide options for sickle cell carrier testing. And many individuals of those communities are working with faith-based leaders, community-based leaders to increase awareness about carrier screening. They would go to their churches and masjids and just community events to bring awareness. And that's how they spread the word about sickle cell. Great job, Ambreen.
Yeah, and just to add, like if somebody is also playing sports or enrolling in the Army here, usually they are tested for sickle cell, especially if they're practicing at higher elevations, because that will trigger sickle cell trait.
Thank you so much for joining us, Ms. Ambreen, and thank you so much for asking our questions. We're very grateful to have you here and for the opportunity to share your insights with us. Next, we have the most awaited talk, at least for me, of this whole session. Our next speaker is one of the co-founding members of Pakistan Genetic Society, and I would like to call Dr. Syed Ajaz. Dr. Syed Ajaz Ahmed for his talk on clinical genetics. Please welcome him with a round of applause.
And just to add, you're seeing this as a change of schedule, and that's because we've had to delay lunch for a little bit because of the rain outside. So we're going to have lunch now at 1 p.m. and we're going to have Dr. Ajaz do his talk, and then we'll have the interactive discussion on consanguinity right after. So the 1 to 2 will now be the lunch break, and then we'll begin with Dr. Usman's talk at 2 p.m. for lunch. Okay. Yes, you have half an hour. That's okay. Yeah, go for it. Thank you.
Excellent, excellent.
5 would be better. 25 minutes would be better, but that's fine.
Okay, I'll try to do as quickly as possible. You guys are enjoying it? Are you enjoying the session? I can see a lot of interactive questions. So how many medical students here? Well, quite a few. All right, how many doctors? All right, quite a few. All right, excellent. And obviously the rest are laboratorians and science students, so welcome all. I am going to now, from prenatal, the baby is born now. So going from Ambreen's talk to my talk, I'm just going to do a very basic talk just so that you guys can understand what is the basic workup of a child with genetic disorder. You can, wait till the end of the question and answer session, but if there's anything on the slides which you really don't understand and you can't, you can raise your hand and I'll be happy to answer that question real quick. So I don't have any financial disclosure. Oh, sorry, I'm thinking it's going slow. So I will start with some basic concepts which you already know. So for the field of medical genetics, generally, this is not something that has come up so far. If you divide it in broad categories, you have the laboratory genetics and clinical genetics. Within the clinical genetics practice, that's what we are talking about mostly today. We kind of separate them out as pediatric and adult genetics. But cancer and prenatal obviously fall in the adult genetics category, though there are some genetic syndromes that cancer syndromes that pediatrics group has as well. So today I'm going to talk about more of a workup of a child with pediatric genetic disorders. Just to recap, we've already said this many times. We have a cell. Cell has a nucleus. Nucleus has DNA within. In metaphases, they are arranged on chromosomes. So there are basically two sources of DNA in our body. One is the nuclear DNA that is present in the, this is the pointer, Salman? Okay, perfect. So we have the nuclear DNA and we have the mitochondrial DNA. The nuclear DNA obviously comes from both parents, 23 each. So that is where we talk about most of the genetic testing that we do. But there is also this component, the mitochondrial DNA, which is only maternally inherited. And there is separate testing for it. So if you're really, if I'm going to be close. So if you're really wanted to test for mitochondrial DNA, and we'll talk about that, you have to do a separate type of DNA and order a separate test for it. One important concept that I just want to make sure that you guys understand is these two differences, germline versus somatic genetic alterations. So germline means that it's in the egg or the sperm or a very initial stage of the baby when it's being formed. So germline alterations occur in zygote, they're present at birth. Somatic alterations occur in post-zygotic stage or in specific tissues later in life. Alterations in all of the cells of the body. So you test any part of the body, you'll find that alteration. Whereas somatic will only be present in those cells that has that problem. It is inherited from an affected parent or may occur de novo as well. whereas for somatic, not inherited and cannot be passed on the next generation, unless there are exceptions where there are some somatic changes that involve the ovaries and that can be passed on. Investigated by testing blood or other tissues suspected genetic disorders. This is investigated by the testing on only of the affected tissue. So that's an important concept. Underlying cause for some genetic disorders like NF2 and Wiedemann syndrome, which is neurofibromatosis 2. But the most common applications of somatic testing is in cancer genetics. Just sometimes there's a confusion. What is a genetic syndrome? What is a genetic disease? How do you differentiate between them? Basically, they're just very interchangeable. But when you talk about syndrome generally is a group of symptoms, a group of set of symptoms of clinical features associated with a particular disorder. Like you're talking about Marfan syndrome, in which you have a patient whose eyes are affected, their heart is affected, their spine is affected, they have lungs. So it's basically a syndrome. One gene, many different manifestations. When you talk about disease generally, but this is, people use it interchangeably. And for syndrome, you can have, like, for example, hearing loss. Hearing loss can be part of a syndrome where a patient may have multiple problems plus hearing loss, or it could be just hearing loss. We know a lot of people who have hearing loss and they are in general community. So that becomes a non-syndromic hearing loss. For disease, it's usually one part of the body being affected because of that gene causing it. But again, people use it differently. I see it as like cardiomyopathy. There are many channelopathies. There are genes that can affect a particular part of the body. So they may be called, but then people use it differently as well. So just so for that concept. When you are thinking of a genetic disorder, I would like you to think of it in broad categories. So first thing or foremost thing that you should think is the whole chromosome. Is this a full chromosomal problem, like Down syndrome patients that you have seen? Or I'll give you some examples. So you look at this in these broad categories. Is this a chromosomal problem? Generally, there are multiple medical issues associated with chromosomal problems. Or it could be a contiguous gene disorder where there are multiple genes in this DNA that are either missing or extra. If not, then you can go to a single gene disorder. So kind of think of it in those three main categories. That's how I see them. But there are other categories as well, imprinting and all that. I'm just giving you a basic idea at this point. So for pediatric setting, when do you suspect that the child has some problem? In Pakistan, in our experience, the clinic that we have been doing, people suspect a genetic disorder after a couple has 3 affected children. Then they say, oh, you have 3 kids who are affected. There must be a genetic problem. That's really not the case. For all the doctors and pediatric residents and other people, I think that would be a disservice if you see one child and you say, oh, this is just one kid, don't worry about it, just slow or just has special needs and things like that. So you really have to recognize that if you have a child who has multiple congenital anomalies, they have the heart defect or spine defect, and then you kind of know that there is something going on. There's some genetic defect that is causing it. Or the child may just look different. So you pass by the child in the mall and you say, oh, it looks different. I don't know what it is, but he looks different or she looks different. So they may have facial features, drooping eyes, or low set ears, or small head, something about that makes you feel like this is just not normal. So that's something that you have to see. If you see that in the clinic, think of that this kid may have a genetic disorder. Or a child is not growing. You can see a child who has growth problems, short stature, some issues going on, failure to thrive. That's when you suspect that there is genetic disorder. Going forward, a child who's not doing well in school, has developmental delays, has features that are suggestive of autism or regression. In my times when I went to school, there were some kids who would not sit still, and all the teachers would beat them up because they just could not sit still in the class. They would just talk to people and all. Nobody recognized that they may have ADHD or they have ADD. those were not recognized at that time. And they would just say that this kid is just talkative, and they would be called to the parent-teachers meeting and get the meeting, right? So those are the things that are important now. These days, there's more awareness that you have to understand those things. Or if the, like I said, the behavioral disorder or intellectual disability, fairly the child is just not doing well in school, not being able to keep. In US, all kids go through developmental analysis every stage of school, so they can see if the child has delays in some form, so that they would recognize them and put them in special education, that they would need get more help. Over here, some those things are not, are not instituted, and many times the children are just put in the category that they're duffers. They just can't understand anything. They just can't keep up, and that's just not right for that kid. They may have a genetic reason for that, for their intellectual disability. So that needs to be recognized. As doctors, I would want you to really think about that. And or if there's a multi-system involvement where you have heart defect and brain issues or microcephaly and things like that. So those are the things where you have to suspect that there might be something going on in terms of a genetic problem. So once you recognize that, and if you don't have a luxury of having a geneticist or a genetic counselor in Pakistan, there are not too many here. So important information to extract from that family. The medical history is different from what we are taught in medical school. You start with presenting complaint, and then you go to history of presenting complaint, and then you go from there. But for genetic disorders, actually, the medical history starts when the mom was pregnant with that child. You have to ask the questions. Were there any exposures? Were you diabetic? Did you have any teratogenic effects? Here, there's not too much maternal exposures over there. Alcohol is big in, you can have fetal alcohol syndrome, or smoking, or drugs are rampant. So all of that history starts right before the mom was when mom was pregnant, to the time of the birth. What happened during the birth? How long was the labor? Were there any prenatal complications? Was there a possibility that the child had hypoxia or some other issues happening at the time of the birth that may have caused some brain problem? And then you go on forward in terms of when did the child, what were the Apgar scores? When did the child start walking, talking, all of those things? That would give you some idea. If they say the child never, crawled for 16 months and then they started walking, then you know there was something wrong with that child. Or the child had feeding difficulties, then you know that there was feeding difficulties, hypotonia, or something like that. So any clues that you can pick up at that time is very important. So the medical history goes from right from there till the time the child has those problems, and it's a very comprehensive history. Family history, I cannot overemphasize how important family history is. And family history is not what we are taught in medical school: diabetes, hypertension, and the basic history. This history is on both sides of the family and pedigree charting, and I'll briefly talk about that, but that's very important. Then comes the physical examination. The physical examination for a child with pediatric disorder is very different. You really have to observe the child in every way possible. You look at their parameters, and I don't want to get into the details. You have to look what the distance of the eyes is, where the ears are, how the ears look, they're different. Nose, every little details, dysmorphic features are noted. And then you basically pursue, what I do is I start with the head circumference and really go all the way down, looking at every feature and looking at the fingers and there are so many clues in patients who have genetic disorders, sometimes just broad thumbs, sometimes just clinodactyly, which is a small curved pinky. Those little subtle things you have to pick up on physical examination that would give you some idea that there is some genetic problem going on with the child. With that, you come with the provisional diagnosis. You think maybe this is what's going on, and then you do basically a pre-test counseling that Fizza talked about, and then you send out the genetic testing. Important thing is for parents to know what type of testing you are ordering. That's something that you really have to make sure that they understand. And Dr. Rizwan Naeem is going to talk more about it, because sometimes we may find changes that are not relevant to what patient has, and that is important for the patients to know. And then obviously the post-test counseling comes in. So as far as genetic syndromes are concerned, it's not an easy task. Previously we had we had a book of Smiths, and we have some genetic syndromes that we learned, but now these days It's with the whole genome and whole exome testing, there are thousands of genetic syndromes. So there's no way that you can expect that you would know everything. You will know a few, and you will actually find out every day, every week. I see some patient that I've never seen before in my life. And I've been practicing for 20 years. This is where genetics is going. This is so new. So many disorders are being diagnosed. So don't worry about it. Sometimes you can just, just do the regular workup and rely on genetic testing as well. And most of those syndromes have a combination of medical and abnormal physical features. Some are easily recognized, like I said, some are not. And some are very rare and maybe diagnosed just by genetic testing. But many of them remain undiagnosed. That's an important thing. In genetics, it's common just to say, something's wrong. I think the child has, maybe in the future years, we'll have a better testing available that we may be able to find what your child has, but at this time, we think this is what it is. But then you go to the symptomatic problem. In medical genetics, this is our stethoscope. Really, this is our stethoscope. You cannot diagnose a child if you don't have a child or anybody if you don't have a proper pedigree. And in order to have a proper pedigree, you really have to, in our culture, most of the time they say, somebody died, we don't know what they died of, which is fine. You may get some ideas. How old were they when they died? Were they sick before they died? Did they just had a sudden death? Those are the little clues that you can, they say, oh yeah, they died suddenly, or no, they were in the hospital for a long time. So those will make you think that that was a chronic disease or what the problem was. So, and it also tells you if 3 children affected, nobody, you'll think of recessive. If multiple generations on one side, you'll think of autosomal dominant. If only boys are affected, you'll think of X-linked. If all the kids of a woman are affected, you'll think of mitochondrial. So there's little things that will come up, but a family tree is very important. So now I'm just going to quickly go to what we talked about, just a few examples of chromosomal disorders. So chromosomal disorders basically happen when there is disjunction. Everybody in medical school, we hated meiosis 1, meiosis 2. It's it's a problem. I still to this day struggle with it. But anyway, the bottom line is, when you have a diploid cell where you have 2 chromosomes, what is supposed to happen is the eggs should just end up having 1 of that chromosome, right? So basically, it's a division of the chromosomes to have eggs that will have one chromosome. So normally you would have a 1 chromosome segregation, but if there is non-disjunction that occurs at meiosis 1, 2 of these chromosomes will both go into this daughter cell and 1 would be empty. So here you would just have an empty egg where you would have a sperm coming in, and then you would just have 1 of that chromosome. But in meiosis 2, the non-disjunction happens in the second step. So you would have 3 eggs that would 2 eggs normal, and 1 egg would have 2 of those cells. So basically, with non-disjunction, you end up having extra or no chromosome in the egg that would be fertilized by a sperm, and the egg, the chromosome count at the end of the day would be either extra chromosome or less chromosome. The common examples that survive, I always say that survive, are trisomy 13, 18, 21. This is a trick question in MCQs that they would say that these are the only autosomal trisomies. It's not. There are many different ones, but those don't survive. There are a lot of miscarriages, but these are the ones that survive to term, and so you see them. Even then, the 18 and 13 are pretty bad with multiple congenital anomalies, and these children usually don't live much. Some examples of someone living to 8 or 10 years of life, but this is something that you see all the time. We probably know some patients in our family that have a trisomy 18. The sex chromosome abnormalities are also very common, actually. Now with NIPT and all that, we are finding a lot more of these. Turner syndrome is 45X chromosome. Triple X is where there is an extra X chromosome. And we have XYY chromosome, Klinefelter, and triple X. The triple X, the XYY, and the triple X syndrome sorry, this should have been 48XX, which I did not. The Klinefelter and the Turner are the more that we see in the clinics, because the Turner has some phenotypes, and I'll give you some example of those patients. And Klinefelters are boys that have that extra X chromosome, and they're infertile, and then they have other issues as well. So just an example of a normal karyotype here, XY. No, I'm sorry, this is not the one. There is an extra, nobody corrected me on that. There is an extra chromosome 18 here. So this is a trisomy 18 baby. What you see is the child who's dysmorphic, usually low set years, hypertelorism. They may have cleft lip. They have overlapping fingers, rocker bottom feet. So you can see a very severe phenotype. And we had the neonatologist here this morning, and they would call Salman or a geneticist to the NICU to see those patients and counsel the family and what's going on. So I'm just going to give you an example of the Turner syndrome. And Turner syndrome, and the reason why I put this case here is because there was a psychosocial aspect to this case that we discussed earlier. These girls have one X chromosome. There's a phenotype that has been associated with that. They usually have short stature, they have webbed neck, they may have white space nipples. And, there are other little small subtle features that have been described with that. But most of the time, short stature is what brings these patients in, and amenorrhea. So they don't start their period. So, and this is what we have seen in patients in Pakistan and patients in ICH that we have talked about, where we actually had a mom who had this girl who was 18-year-old, very nice girl with Turner syndrome. And we were talking to her about what it is and heart defects and kidney defects and all of that stuff. But her biggest issue was, should I marry her or not? What should I tell the people that she can't have children? And those were the things that bother people over here. So it's very important to understand these issues when you are seeing these patients and counsel them accordingly. And so going from chromosomal disorders, we can talk about contiguous gene deletion syndromes. Basically what happens is there's misalignment. It's just like, think of these like two pieces of Scotch tape. If you have two equal pieces of Scotch tape, they have to like join together in the right way. But if you go up and down, or just the shirt of the button, you put it up and down. So these are non-homologous repeats that are supposed to join together properly. But if they don't, then they may stick to the other similar part of it, and that will result in the deletion or a duplication of the chromosomes. And there are some common areas in the chromosome that it happens on a recurrent basis. And those are recognizable. So those that combination can either happen between two chromosomes, interchromosomes, or within a chromosome where it would flip and stick onto each other. That will result in deletion or duplication. It can happen in intrachromatid, where we will end up having a ring chromosome, or it could happen in the sister chromatid exchange. But bottom line is, at the end of the day, there is either an extra piece of one chromosome or a deleted piece of one chromosome. And that's called unbalanced chromosomal thing, and that can cause disease. When I started, we only had a few that we used to do FISH testing for and try to recognize them with 22q deletion and Williams syndrome and all that. But once this test became available, called microarray, which I'm going to talk to you about, then this area exploded. There was so many different recurrent deletions and duplications that were noted. And like I said, there's a more recurrent ones. The common ones that we see in our clinic are 22q1 deletion. I'm sure medical students know that. D. George, villocardiofascial syndrome. We would quiz each other on those. And then it can be a duplication of the same area as well. You have Smith-Magenis syndrome and Williams syndrome. So the chromosomal microarray test, for those who don't know, it was a game changer for us when it came before this whole exome sequencing and a whole genome sequencing, where basically a patient's DNA is labeled with one dye, the normal control is labeled with another dye. They both are mixed together. Now, if they are equal green and red, you would have a yellow signal. But when there is extra green, then you would know that that extra green means that the patient has extra chromosomal material, so that's duplication. If there is extra red, that means the patient has deletion. So I'm just giving you a very simple term of this test so that you recognize what the CGH is. And this, like I said, was a game changer in identifying many, many, many recurrent deletions and duplication syndromes. Just an example of a child that you may see in the clinic. Does anybody think there's something wrong with the kid? Very subtle, right? You may just pass by this kid and say, looks okay to me. But, as geneticists, we come to recognize these patients that there's something just not right about the child. There's some, periorbital puffing, pouted lips here, and ears are slightly low set, and anteverted nostrils. Those are the things, that subtle features that we know. This is the child that has Williams syndrome. And Williams syndrome, this is an example of a CGH report that I wanted to include, that basically it gives the map where that deletion is, and it says 1.5 megabase pathogenic copy number lost at 7q, and it basically tells you where it is. The deletion can be two, can be 1.5 megabase like we had in my patient, or it can be 1.16 megabase. Basically, this is the gene, ELN gene, that is deleted in these patients, and that causes all the effect that we see in patients with Williams syndrome. They have distinctive facial features that you recognize: short stature, poor growth. They have global developmental delays, mild to moderate intellectual disability. But the fun part is that these kids are the life of the party. They are fun kids to be with. They would, from the nursing station to they will make friends with everybody they meet on their way. They're just delightful kids. They have some medical issues, obviously, hypercalcemia, like I said, they're hypersociability. I'm just giving you one example of each because there's so much that we have to cover. Then, once you pass the contiguous gene deletion, then you come to a single gene. Okay, now this is not some single gene. So you may recognize what the genetic syndrome is, and you can, oops, I'm sorry. So if you recognize the genetic syndrome, then you can basically order that genetic test for that particular gene. But most of the time, we don't know what genetic problem there is. So sometimes we go big. So if it's a patient who has epilepsy, then you can just order the testing for epilepsy gene panel. And that would cover all the genes that have been described with epilepsy. So you're basically putting them in a category. And for that, you can order a multi-gene panel testing. This is available through many labs. Many, many, many panels are available: skeletal dysplasia panel, epilepsy panel, metabolic panel. So if you have a patient that you think of in a certain category, you could just go with that panel rather than going big, because these are cheaper. And you have less chances of finding genetic changes that you have to deal with. But really, if you don't get this answer, then you can go with this next best test, which is whole exome sequencing. And we pretty much order this on a routine basis now because it's become very easy. But it's expensive. From here, it's very expensive. And in the United States, it's covered by insurance. So instead of going that, we just basically go with whole exome sequencing. And the difference between the whole exome and whole genome sequencing is the word exome. With this test, you only are testing all the exomes. Remember, the genes are spliced into, the introns are spliced out and the exons are what's translated. So you're looking at the exon. So you may miss something on here. The whole genome sequencing is a much broader test and covers the intronic regions as well. And it may cover some triplet repeat disorders as well, but not all. This test is basically kind of the mother of all tests. It would cover chromosomes, it will cover microarray, and it will cover single gene. What it does not cover is triplet repeat disorders that you have to understand, and obviously mitochondrial disorders. And if there's methylation or imprinting disorders, that might not be covered by this gene. But it's pretty good. Just a case of, for a single gene disorder, I wanted to include another case for the psychosocial issues oops. So this is a 31-year-old male with a 2-year history of depression, motor twitching and clumsiness, forgetfulness, slurred speech, and but the gentleman is fine. The reason why this is an adult case, and I wanted to include that, this is the family history. You can see very significant family history. The grandmother here is affected, and the grandmother has so many children, and out of that, 1, 2, 3, 4, 5, 6 of them are affected, and their children are affected. So you clearly know it's an autosomal dominant family history. And I'll get to the repeat size later on. But basically, this is a family when you have a person who has chorea and depression and all those features that I told you, they have Huntington disease. And one important phenomena with the Huntington disease is the word anticipation. And that's the reason I put this case here, is anticipation is when the triplet repeats expand in successive generations. In Huntington disease, when it's transferred from the father to their children, the repeat size may expand. So you may see the father unaffected, but they may have borderline CAG repeat, but it expands in the child and then they are affected. So that could be something that will throw you off. These patients have irritability, they have apathy, anxiety, depressed mood, obsessive and compulsive, and some of them are psychotic. There's progressive cognitive decline leading to dementia in these patients. Bigger issue is their motor function, where they have uncontrollable chorea to the point where they move so much that they become sick, and then they can die because of that. In fact, they die because of that. And this is basically a death sentence when you're telling the patient, because there's no treatment for that. So it's very important when you're testing asymptomatic patients. There are patients who know that they may get this disease later on and they test positive. There have been cases where they have committed suicide, that rather than getting this disease and dying of that, they may just kill themselves. So you have to make sure that you're counseling these patients and making sure they're ready for the result, because everybody is hoping for a negative result when they come in, but they may test positive. So that's important. And Salman and I think Dr. Saqib are part of the Huntington Disease Society of Pakistan. I saw your names listed on that. I did not know that this was something that's available. This is a case of a patient from Sachin and NICH that we saw, and I thank Dr. Misbah for sharing this case with me. This was a very interesting case. I just want to highlight what we see here in Pakistan. So this is a kid, a 14-year-old with short stature, bilateral cataracts, diabetes mellitus, sensory neural hearing loss, dysmorphic features, and he was product of the consanguineous marriage. This is the child here. You can see obvious dysmorphic features, long beak-shaped nose, and he's cachexia. He has simple ears, low set ears, and so you can see that he has long hands and long fingers. So to me, this kid looked obviously dysmorphic, but it looked like it had some aging. I was thinking that this is probably an aging disease. And interestingly, he has two other siblings who also have similar issues as he does. So it was clearly an autosomal recessive, consanguineous marriages, and 3 kids affected, like I was telling you. So if I was thinking of aging disease, I thought of maybe this could be Werner syndrome, because that was my provisional diagnosis. And I was pretty sure that this is Werner syndrome, and I thought I nailed it. They ordered the testing for it, and whole exome sequencing was done. And interestingly, like I said, never heard before of this gene, DNAJC3 gene, related combined cerebellar and peripheral ataxia with hearing loss and diabetes mellitus, ACPHD syndrome. So these are the kind of things that we encounter, that you're thinking of something that's common and you end up finding something that you've never heard of. That's what we got. And obviously the child had a frame shift variant, and it was biallelic pathogenic variant that's causing the disease. I'm not going to go over everything on this, but these patients have growth problems. They have short stature. They have sensory neural hearing loss. They have ataxia, which was not reported for these patients. So we were not thinking of that line. But they had diabetes mellitus. That's why they had come to this clinic here. And that's why he has developed cataract, because it was uncontrolled. So this is just kind of an example how complex it can get. For one gene, all these problems can happen. As far as the mitochondria is concerned, Generally, when you think about mitochondrial disorder A, the maternal inheritance, multiple children affected, but mitochondria are basically the lungs of the cells, right? So when the mitochondrial DNA is defective, multiple organs are affected in that person. There are obviously hereditary optic neuropathy and all that. So, but if there's a patient who has so many different things going on, so central nervous system problem, cardiac, endocrine, renal, eye, gastrointestinal, I kind of think of that this could be multiple children affected, all have multiple medical problems or specific, then you can think about mitochondria. But usually most of the time it's a diagnosis of exclusion when you have excluded other condition. So I'll end now. I think I'm on time. Perfect. So the take home message here is do suspect genetic disorder when there are, it happens in my talks, right? So do think of it. All of you are doctors or scientists, or when you see these patients or family members who have certain things, you may talk to their parents and tell them that, you know, we've heard about this. This could be something. You may go and see a geneticist or genetic counselor. So don't let a family just go on saying the child has special needs or child, something's wrong, you know, or intellectual disability or mental retardation. Mental retardation, by the way, is not a term that we use anymore. It's intellectual disability, to be politically correct. But that's what we use here. I used it here when I was here. So those are something that you have to kind of think about these children as they are suffering. Those children are really suffering. So, you know, they need therapy. If you diagnose those conditions in those children, then you may not treat them. You know, somebody would ask you, like, why should I diagnose the condition? And Salman touched on that. It helps the family to understand what their child has. It helps them to get therapies. It brings a closure. It brings information about the recurrence in the family. It brings information about their successive generations. Should the girls be worried that they're going to have children? So all of these things matter. It's just not like if you can't treat a disease, then why bother about it? Things can work out for those families. So think of that way. And so lifespan, medical history, and a complete family history is very important. Always ask history of consanguinity because that's important. 66% consanguinity in Pakistan. So you see that very often. And we all have people in our families. I have people in my family who have cousin marriages. Different genetic testing modalities for the diagnosis. Think of either chromosome, contiguous gene, or you know, the whole genome exome, and also be modest. After all of this workup, you may end up not finding something. So when you're counseling the patient and you're sending $3,000 test or you know, $80,000 test for the patient, make sure you tell them that even after that, there's a possibility we may not find anything. Don't make them believe that, oh yeah, just pay up 80,000 rupees and we'll give you an answer. It may not happen. And we may find variants of uncertain significance. So be cognitive about it because it's somebody's money that is going there. And with that, I will stop. And this is what happens when you're a dysmorphologist. You walk, go on a hike, and you look at a tree, and then you come back and like, there's something wrong with that tree. And this is a tree that actually I passed by in Everglades National Park in Miami, and I felt like, to this day, this tree. Do you see something here? You do, right? It's like, wow, the tree was just looking at me. And I was like, oh, this is cool. So this is what happens when you're this small. And in fact, when my family members have children, you know, some babies born and I go just to see them family, they get scared like, okay, he's going to find something about these babies. So things happen. I'll take questions. I have time. How much time do you have? Few minutes. Thank you. Any questions?
So I have my question related to the overlapping phenotypes. I have came across a lot of syndromes like Mucopolysaccharidosis type 1, type 2, type 3. these are all having quite overlapping phenotypes. So it becomes very difficult to diagnose them on physical examination sometimes.
I completely agree with you. For metabolic disorders, it's a little bit more trickier because you have so many. You kind of can put them in a broad category that this is mucopolysaccharidoses. I mean, you can't always say type 1, type 2. Hunter syndrome is X-linked, so you can kind of go with that. But many times you just have to rely on biochemical testing. I'm sorry, I'm just trying to shield my eyes here. You have to rely on biochemical testing, and there are subtle clinical features differences between those syndromes that you can rely on, but generally most of the time it's more on the other end where you do genetic workup or testing or biochemical testing that would give you a better answer for metabolic disorders.
Yeah, we recruited those patients initially as suspected MPS1 cases, and later on after whole genome sequencing, we found them categorized in Shimkio-immuno-osseous dysplasia, Aguirre-Kucinskis, 3MC. They deviated a lot. So
Yep.
You can say the dilemma that we are having very poor diagnostics related to such disorders.
You know what, biochemical testing surprises me because this is like a mass spec. I don't know, does Alkhan has a mass spec? So, many of these disorders can be diagnosed just by biochemical testing. That's how traditionally they were diagnosed. We didn't need it to do genetic and DNA testing. So I'm sorry, I'm ignorant. I don't know exactly what's available in Pakistan for this type of testing, but that can be done on simple screening tests. And in the US, most of these are even part of the newborn screening that you just
Yes.
screen them beforehand.
But unfortunately, we are not having dry blood spot analysis through tandem mass spectrometry available in Pakistan. I've worked since 2017 in the field of LSD, and there is no such facility available here.
Well, now you are here, so you got to set it up. That's what we are all trying to do. This is not here. We are all getting together to set up this genetic program and things that I can. This is an amazing crowd. So you are an expert and you've worked it. So just do it.
Thank you.
Mohsina, there's.
Oh,
Thank you so much, Dr. Ajaz. It was a really wonderful talk. I really enjoyed it a lot. Thank you for supporting us for the last almost 2 years. Dr. Ajaz is doing free counseling for our patients, online counseling very regularly and very passionately. And sometimes we are unable to, we see so many diagnostic dilemmas almost daily, so many disorders daily in our setup at National Institute of Child Health. And 1 or 2 cases, we were totally stuck, and Dr. Ajaz guided us with his team. And thank you so much for all these efforts for starting genetic counseling free. And our patients are really, this is a luxury for them because they can't afford to even do the test or even to afford the counseling, because the counseling sessions are also very expensive. So in Pakistan, and thank you so much for doing it free also.
Thank you.
एक question मेरा ये है कि we are getting so much VUS in our जो भी हम data भिजवाते हैं. Like even if we are testing for skeletal dysplasia, sometimes we are getting 3 or 4 VUS in the same report. So what should we do then? And Pakistan, do we need to develop our own genomic data in Pakistan also? We are still lacking in it. So what is your opinion? and
Very,
what's-
very good question. And thank you for the question. And thank you for sharing your patience with us. So Dr. Kirmani talked about it in his talk that, the reference sequence is a white person over there, and that's what they're comparing our DNA with. So we have very different variations in the chromosome, and they don't know what those are. They just call it a variant of uncertain significance. So till the time, say, the testing is broadened up to include all different populations, we will end up finding these variants of uncertain significance. But the good thing is that, we can make some clinical correlation with certain genetic disorders. And you've had cases where we have said, no, this is it. This is what's causing the problem. But you have to be very cautious when you're talking about variants of uncertain significance, because that could just be, you just can't rely on them. So that's important to understand. We can talk about it more. I'm sorry, we're running out of time, but I'll be available at lunch if any of you have questions. Thank you.
Thank you, Dr. Ahmed, for your wonderful talk. Next, we have a very exciting interactive discussion on the theme of consanguinity, how to address the issue of cousin marriages. And to take over this discussion, I would like to invite our Dr. Salman Kirmani. Please welcome him.
Thank you very much, everyone. And thank you to our online audience. I mean, it's just so wonderful to see so many people here. And what an amazing talk, Ajaz. I mean, you really, this was a tour de force. I think what a medical genetics resident needs over a 2-year residency, you just collapsed in half an hour. Is fantastic. Thank you very, very much for that. And I'm sure everyone has benefited from what you've told us. So I'm just moderating this discussion here, okay? This is going to be interactive. So this is where we will really need all of you to speak up and to share your thoughts, and we'll have mic runners hopefully around both aisles. So Arif, maybe one person here, and we really want people to interact. Look at this title, right? I mean, it's kind of a biased title in its own right, isn't it? Right? This is some bad thing. We want to make it go away. Quick audience poll. Who wants to make consanguinity go away? Be honest, it's okay. See, you know, like, okay, so it is a problem. All right. So the first part about doing something about a problem is recognize it, right? But you know, it's something that has to be dealt with very, very sensitively. All right. Oh, we have a question right away. Arif, mic runner right here. Okay, just be cognitive. We have to be quick. We have to finish right at 1. All right.
I think consanguinity should not go away. It should be the awareness should spread and the screening should be done.
So the diseases associated with consanguinity
Yes,
should go away,
that
but
should
maybe there is an aspect of
There should be a logic
consanguinity
behind
that is positive. That is very good, because you know that's the first time I'm actually hearing someone verbalize that. Because most medical professionals are like on a jihad against consanguinity. And imagine how it feels to the person who's bringing their child to you, and they are cousins and their parents were cousins and so forth. So that guilt, that burden, you know, how do you deal with it? So this is how I take it, right? And if people who, this is all the younger generation sitting here, I'm going to put the music on. All right, that was the soundtrack to The Good, the Bad, and the Ugly. All right, so we have to acknowledge that society perceives a good in consanguinity. There is a bad side to it. And then there is an ugly side to it. And what is the ugliness? I want to just point it out. And I think the ugliness is the way we sometimes deal with it quite inappropriately. All right. So again, this is not meant to be a controversial discussion, but it's meant to be provocative, right? It's meant to really, get you guys to think. So here's an audience poll. And please, again, we're all friends here. We all want to be honest. How many people feel that there should actually be a law against cousin marriages in Pakistan? We have one person who's honest, the sole person in the audience. Okay, there's one more person. Oh, there's online audience, so please engage. And someone, Arif, maybe you can point out what the online audience is saying. But I'd really, okay, so there are only 2 people. So then we don't need legislation. It shouldn't be regulated by law. That's what most people think of it. Okay, that's very, very interesting. Very, very interesting, okay? Then here's another question. And again, be honest. How many people participating in this conference today are either themselves in a consanguineous union or are the product of a consanguineous union? Right?
So we have an audience.
Many more hands, right? Many more hands, right? And have you ever felt embarrassed about this? Right? Okay, good. That's excellent. But you can imagine, right, that in a medical setting where you're talking about this, it may actually be, you know, these people are trying to, you know, prevent all these diseases and you yourself are participating in the process. You know, what's going on? So it's really, really important, right, to take it a bit personally first and say, okay, how would I feel if I had a child with a recessive disorder and I took them to the doctor? You know, because I've had people who said, my doctor has told me, "Khabardar jo tumne bacche paida kiye. Do not ever have kids." I mean, that's one of the worst things a human being can tell another human being. And these people actually think they're doing something good. You know, so it's really very, very sad when we see stories like that, right? So there is this shame and guilt that our patients come with to us, right? Because they've probably been made to feel bad about it before they've come to us. Or even if they haven't, maybe they're feeling bad themselves when they realize that this is, you know, a situation that arose because of consanguinity. So this is sort of the technical definition of consanguinity, and this is just a setup. We want to discuss this further. That this is sort of the degree of heritability that comes in, and there's this, sort of concept of, how much identical you are by descent to each other, and the closer relative you are, the more identical you are. The diagram that Fizza showed, and then I think Ambreen again showed, was showing the degree of, how many percentage of genes. So the average Pakistani union, which, probably is about at least 50% consanguineous unions, but maybe even touching close to 60-65%. The actual sort of, the sort of the F is actually 0.033, which is closer than a second cousin, So it really tells you that most Pakistani unions are at risk. And, so we see this regularly. We see this in certain closed religious communities where they say, hum kabhi bhi cousins mein shaadi nahi karte. And they're very, very confident about it. But they come from a very closed community, either geographically isolated or religiously isolated. So they only marry within their community. So obviously they will have common ancestors. So they may not be cousins, but they're still related to each other. And that's very important to know. All right. These are the numbers, right? Look at the international thing here. So again, unfortunately, in certain things, Pakistan is always number one. And in consanguinity, we are number one as well. More than Kuwait, more than UAE, more than Saudi Arabia, more than Indonesia, more than any other Muslim country that you will find. All right. And there you are. Those are the numbers. And obviously, the risk of autosomal recessive disorders is high. But one must also, when they're counseling, understand that there are birth defects, multifactorial birth defects, that because of common genes coming from both sides of the family, there may be a higher risk. And people do see a higher incidence of birth defects in consanguineous unions that are non-recessive. They're not recessive, but they still tend to occur higher. All right. So as a society, this is what people say, that the reason why they prefer cousin marriages are all of these listed over here. And, you cannot ignore these. This is very important. It may be very important for a family to be in touch with their daughter. If they marry her, 200 kilometers away in another village, they may never, ever see her again. They may never see her children. Right? So that may be a preference that they have. For us to say, wo to sirf jaidaad ke liye karte hain, or, they only do it for money, or that's not it. There are very, very good reasons why people want to prefer this, and one must acknowledge all of these. This is the bad, so here's an interesting thing, right? That there are some communities that they actually feel that they have good genes and they want to preserve those good genes. So marrying within the family will actually preserve those good genes. And, there are people from animal husbandry background, people who are farmers who breed cows or horses. And to them, it's second nature. Of course, you'd want to marry your cousin, because, your family, your caste has the good traits. So we want to preserve those good traits in the next generation, right? So, I mean, those realities have to be dealt with. You can't just ignore them, right? But here's the bad stuff. And that's, we already kind of know that. So I'm not going to belabor this, because all of you know and you've heard about this, and especially in the recessive disorders, but also the common disorders, etc. All of those come in. There is some data, and again, it's not very good data because it would be very hard to do those studies, but there is some data to suggest that maybe at a community level, there is a slight few drop in IQ points, etc. But again, at an individual level, you cannot counsel that your child will have intellectual disability because you're first cousins, because we don't see that. The people who are in the audience who are products of consanguineous are proof that you're functioning very highly, very well, despite your parents being first cousins. So, again, I'm sorry I highlighted that, but it's important to highlight that because that's the reality. All right. And here's the ugly part, right? And this is how we've dealt with it, right? That, there are times when consanguineous unions happen under pressure, and I think it has to do with a lack of women's empowerment, right? And there's a clear correlation between a woman's level of education and the likelihood that she will marry a cousin. And that probably has to do with just education and awareness, but also maybe just the choice of partners, right? The more the woman is out of the home, the more educated the woman is, the more choice of partners they have and so forth. So it's not cousin marriage by default. In fact, I was just the other day passing by a road, and the matchmaking app Muz had this big ad that said, "Cousin ko chodo, Muz mein dhoondho," something like that, right? So it's interesting that people are actually catching up on that, right? It's all these things that one has to be cognizant of and be careful about. So, here's a case scenario which, happens daily in our clinics, and I'm sure it happens daily in all the other clinics too, that, they've had 3 children who've died. They have 3 healthy girls. And the reason why I highlight the 3 healthy girls is because every family wants to have a boy, right? And it's really unfortunate, but that's the way our society works. And they want to do whatever it takes to have a boy, because to them, that's the desirable outcome. So how do you counsel them, right? And there's many different ways. I've discussed some of those, and we'll discuss, more. But it's, do you tell them 3 is enough? Forget it. Why would you want to take a chance? Or do you say, absolutely, you have every right. Let's do this, 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 and this. Who decides? How do you take it from there, So here are the common responses, when we tell them about the risks. We hear this a lot, right? And people say that, why are you saying that, the cousin marriages increase the risk? We've had so many cousins who married for generations and nothing like this has happened before. And you saw, based on all the knowledge you've learned today, obviously, that can happen. It's just a chance, right? And then the religious aspect comes in, and they say, well, if it was so bad, then Islam would have done something about it. But because Islam not only didn't do anything about it, but in fact, Prophet Muhammad married his first cousin. So that's an example for us, right? But again, it's a matter of perception, right? I mean, there are so many things that the Prophet did which spread Islam in many different ways. And a lot of things that he did was spreading outside the community. All the people who went and traveled to different parts and established things over there was done, for a reason. So it really is a perception. What do you look at? What part of religion do you want to look at, right? And a lot of this comes down to, we see people who get it. We see people who say, we know the 25% risk, but Ya Allah ki marzi hai, we will let it be. And that's fine as long as it's their choice. I think that's very, very important. A lot of lay press has picked up on this. And over the last few years, we've had articles in the press coming like this. And it's good to have the conversation, but it's good for the conversation to be unbiased. It shouldn't be a biased one-way conversation. I think that's very, very important. And here's my dilemma, and I want to really open this up to the audience, right? That in genetics, one of the key principles of medical ethics that we use is autonomy. The couple sitting in front of you, the patient sitting in front of you is free to choose. If they think that a 25% risk is worth taking, they have every right to take it, right? And in that room, in front of that couple, I don't think anyone can argue against that. But let's take a public health approach, all right? And let's broaden the example to seatbelt legislation. If someone sitting in their car chooses not to wear a seatbelt, they are taking their own risk that they might crash through the windshield and die when their car crashes. Their wearing a seatbelt will not save anyone else's life. It is a very personal decision, and they will save only their own lives by wearing a seatbelt, right? But yet, most of the developed countries in the world have legislation about seatbelts. Same thing with helmets, right? A helmet will only save the person who's wearing the helmet, no one else. So can we please mute the mics? Thank you. So should we legislate this? Because as a public health problem, what do we do? So here's the question to the audience. What do people say? Only 2 people before had said we need legislation. And I'm not for legislation by any means. But all I'm saying is, isn't there a compelling case for it if you wear your public health hat? Because we legislated the lockdown for COVID, and we legislated many, many other things. We sent polio workers to face very dangerous situations because we consider it a public health problem. should we think consanguinity is our public health problem or not? So I'd like to really open it to audience. Is there someone who's not spoken yet? Please speak out. There are people obviously in front who've spoken before. Okay, I see hands. Can we have mic runners, please? Mustafa, if you can provide mics. Yeah. Do we have anyone? Otherwise, Maha, you might want to give the mic here, and then I think Dr. Ajaz had his hand up. Okay, well, I can't see over there, so you guys, please. Thank you. G.
Thank you very much. I think the screening should be done and the law should differ from case to case, according to the severity, according to the impact on the population, as well as on the same couple.
But by definition, a law applies to everyone. So you cannot have a law
No,
that
I mean,
changes case to
law,
case.
consanguinity in different cases. I mean, if they are having that, a very severe disease that can have a, very bad impact on the population, then it should be applied. And in some cases where it is less severe, it should be optional.
So very good question. So is hearing loss severe? Yeah. Is cancer severe? Cancer happens at
There
age
can be a
40?
list, yeah, there can be a list of the severe diseases
Who decides the list?
by the medical experts, and then
Medical experts have no clue. I don't know
But
whether
that's
deafness is
that's
as
my
bad as
understanding.
cancer.
That's my understanding,
yeah.
what I can think.
Yeah, no, it's very good. I'm glad. I'm just saying these are questions that people don't have answers to. Yes, we have more. Yeah. Yes. Oh, okay. Lots of people online. Ijaz, why don't? Okay. So can we have the online people?
Yeah.
Yeah. While the online people are coming, maybe Ijaz can make a comment. But how do we show the online people? Please, can we have that?
Do we have it? Okay, I just wanted to say to enact something, you have to lay the groundwork. So you want to make sure that there are some social determinants that are taken care of. Here, women are not able to go out of their houses. They don't meet people. They don't have. So for those girls, their best choice is to marry someone who they have known. So there has to be a groundwork where the society needs to be opened up more before we enact certain law. That's what I think. But I totally agree the problems that are associated with that. But you just can't push the brakes with a screech. You have to bring it to a slow halt.
Absolutely. Very good point. So I think there are some online participants who are saying something. So
I'll ask Miss Aisha Furqan to unmute herself and ask her question or make a comment.
Yeah.
Aisha Furqan, can you hear us?
Yeah, I can hear you now. Great conversation. I just want to say thank you to all the participants in the hall. Such a lovely discussion. My thoughts on this are such that we are coming to this conversation so late in the human evolution, if you want to say, or just medical genetics evolution, if you want to say, or medical ethics evolution, if you want to say. We have had so many examples of countries that are Middle Eastern that practice consanguinity, in Europe that practice consanguinity, in Iceland that practice consanguinity, and there aren't any recognized bans on consanguineous marriages anywhere in the world. In the United States, there are communities that practice consanguinity that are of different faiths, and if we look at the example of populations that are isolated, like Ashkenazi Jews were isolated, we all know that there are no bans against Ashkenazi Jews marrying other Ashkenazi Jews. And what they have done is something that we can copycat. We could take a book from, take a chapter from their books and copy paste it into our laws and ethics, and really not just copy paste, but make it better, make it even learn from their mistakes and not repeat those mistakes, like eugenics, like trying to get rid of marginalized communities, like the hearing impaired and the short stature, or whatever else you have that is considered less than desirable. We're not against that as a medical community or as an ethics community. We're here to learn and we're here to be better. Thank you.
Thank you, Aisha. And I think the Ashkenazi Jewish example is very good, because what they did was the rabbis actually went to the geneticists and said, we see this as a problem in our community. It was about Tay-Sachs disease. What can you do to help? and the geneticists educated the rabbis educated their congregations, and then, registers were kept, screening was done, and then a common decision between, but it was the rabbis who bartered that discussion. And I think that's what we need to do in Pakistan, is that we start with the religious leaders. The problem is that there is a religious leader in every house, in every neighborhood in Pakistan. So who do you approach? Who do you go to? So, I'm not kidding, right? I thought about this, and I think the way to do it is maybe try this out in certain communities that have a certain religious leader where there is commonality. Maybe it doesn't even have to be religious. Sometimes it's just a community leader. So those communities that have a community gathering every week, those that actually gather as a community and make decisions as a community, those that are geographically isolated, in the northern areas of Pakistan, for example, those are the ones to go for. Are there any other comments? So I think people are, maybe people can just read the chat. Any other questions from I think there was an audience question from up here that may be one of the last ones we take, but go ahead.
Look, Usman is a very important issue, but when we have a law, we are talking about a law. Okay. We do have many laws before.
Yeah.
Like for thalassemia screening. So
Yeah.
how they are implemented? No.
Yeah.
So law is not the, I think, option. We need to increase awareness before going for the law. I think that's
Okay.
the.
That's a very good point, and he's right. I think Dr. Usman has a comment. So I think both KPK and Sindh have laws that you cannot do a nikah without looking at thalassemia screening, but no one implements that. Yeah.
So just to add a comment, last night we were having dinner with some friends here in Karachi. They just told me that there are 40 million motorcycles in Karachi, and there is a law that each motorbike should have number plates, but only 18,000 motorbikes have the number plates. So, I mean, the thing is not to have the law, just the awareness. That's the key. And the dialogue. But with whom? So somebody has to start, maybe from this Pakistan Society of Medical Genetics. So there should be some discussion to discuss about the merits and demerits. And based on that, making law would not serve the purpose. Thank you.
Absolutely. Thank you so much, Dr. Usman. I think one of our medical students had a question, so maybe we'll end with that and then we'll give you some logistics about
okay, so I would like to argue, although I'm not a big fan of, the idea that we should legislate not having consanguineous marriages, but the idea is if there is something which increases the risk of, genetic predisposition to multiple diseases, and since you're not the person who's going to be bearing the risks of that, it's the kid, it is ethically superior for you to go for an option where those risks are less. And if you're talking about the entire laws argument, we have a ban on alcohol, and even though while a lot of laws are not followed in Pakistan, the alcohol law is followed in Pakistan, which comes as a result of religion and the awareness itself. So the idea is having a law would work. It would decrease the number of consanguineous marriages. And since the effects of it are being borne by the child and not by you, it is ethically superior for you to choose an alternative which has less risk.
I'm just going to respond a little bit before Dr. Rizwan. So good points, but here's the deal, right? So this is something we deal with in pediatrics a lot, which is a child is suffering, but the parents are not making what you feel is the correct decision for the child, right? So in almost every instance, the parents will be right because the parents have a right to refuse the treatment for their child. In the US, you would have to get a court order to go against the parents' wishes. So in most scenarios, you cannot use the ethical superiority argument that you are doing a favor to the child, because again, that leads to a whole paternalistic downward slope where everything you feel. So you may think that, having a child be a little bit taller is better and, you're doing the favor of child. That's wrong. So yes, Dr. Rizwan.
So I would say why we are talking about consanguinity, but the reason we are talking about is decrease the disease burden in society, right? And that's a public health issue. And I think the point someone made that if as a parent, if I make a decision that my child will be mentally retarded, I still want to follow that decision, my decision, that yes, I want to have a child with mental retardation. But what has that child done to be in the society treated so poorly with all this mental retardation? So we need to think about not about consanguinity, that's may or may not be issue, but screening, counseling, and decreasing the disease burden. California has adopted a very good law, and they have written many papers that how they have decreased the disease burden of autosomal recessive disorder.
Yeah. Thank you very much. Yes, you have something?
I have a comment as
Yeah.
well on this case, since we're talking about what is the child's blame. So a lot of times we see in the gynae clinics when they get the anti testing done and then the prenatal testing done, then the parents have to make the decision that do they need to bring this child into the world or not. Then again the question arises, what has child done? But then there is a contrarian point to that, God has given him a life, and वो उसके लिए जिंदगी लिखी गई है, जितनी भी लिखी गई है, he should be in the world and completed, and it's in God's hand, not our hands, to end the life or not.
So thank you very much, Mahas. That's a very personal opinion, but you cannot transmit that opinion to someone else. And I think as providers, that's one of the key principles of genetic counseling. Your personal opinion should not hinder your ability to counsel them neutrally, as Fizza pointed out, non-directive, right? So I'm going to end with a statement, which is that, this thing about legislating or you have to understand first, why are people making the choices they make? And without understanding that, you won't know. So there was a very, very interesting study done in Bradford in the UK, which has a lot of Pakistanis. They're from a closed community, lots of autosomal recessive disorders. The question was, once you've had one child with a recessive disorder, the NHS is paying for the special needs of all that child. Should you ensure that prenatal testing is done and that termination is done for the next pregnancy, right? That would go completely against all ethics because of lack of autonomy, right? You cannot legislate it. So when they interviewed the parents and they said, and NHS was considering that for the second child, that's fine, the parents can let the child come into this world, but the NHS will not bear the cost. And the parents would themselves have to bear the cost. You know what happened? They said, oh, we're okay with termination. Right? So sometimes what we feel is religious or cultural decision-making is purely financial decision-making. And you cannot separate the two. It's all mixed into one, right? So I think that's the sensitivity we have to show and then do the right thing for the right patient. So I'll end with that. Thank you very much for that very, very vibrant discussion. I think we can have a whole day discussion just on this. Thank you very much, everyone. Thanks to all our online participants. So we're ready for our break, which is going to happen 1 to 2 p.m. We're just 5 minutes over, and we will reconvene here at 2 p.m. with Dr. Usman's fantastic talk on cancer genetics. The lunch is served wherever we had the tea break for namaz. For the males, there is a masjid, which is if you just go to the parking lot, just across the parking lot, you can do prayer over there. And for the females, there is a prayer arrangement in one of the side rooms over here. You can do prayer over here. We have 1 full hour. Please enjoy, interact, ask questions. Thank you very much for everything, everyone. Thank you.
Welcome back, guys. We will be resuming our conference with a very exciting talk, and our next speakers are Dr. Muhammad Usman Rashid and Fatima Me, and they would be sharing their collaborative insights on cancer genetics. I'll just briefly introduce them. Dr. Muhammad Usman Rashid, MBBS, PhD, FRC PATH, UICC Fellow, is a distinguished senior research assistant and head of Basic Science Research Department at SKMCH and RC, with over 20 years of expertise in precision medicine, cancer genetics, and oncology research. He's an alumnus of German Cancer Research Center, and he established the first genetic research lab in Pakistan and has greater than 80 research papers published in top-tier journals. Dr. Rashid is recognized internationally, representing Pakistan in 3 breast cancer consortia, and has received prestigious awards, including the German Cancer Research Center Guest Scientist Fellowship, the European Young Investigators Award, and the Union for International Cancer Control WHO Fellowship. Our next speaker, who will give us a virtual talk, is Fatima Amir. Fatima Amir, MSCGC, is a certified and licensed genetic counselor who graduated from University of Cincinnati and Cincinnati Children's Hospital Medical Center Genetic Counseling Program in 2018. Ms. Fatima works currently at Ambreen Genetic Lab as lab genetic counselor. Previously, she worked as genetic counselor at Genome Medical, a telegenetic company that offers genetic counseling services across United States. She's passionate about expanding access to genetic services to underdeserved populations around the world. I would like to invite Dr. Usman Rashid on the stage.
Assalamu alaikum and good afternoon. So it gives me immense pleasure to be here in Aga Khan. So I'm thankful to the Pakistan Society of Medical Genetics for the kind invitation, and special thanks to Dr. Salman. So cancer is a clinically, genetically, and epidemiologically a heterogeneous disease. It is a global world problem. Identifying individuals who are at increased risk of developing any malignancy or those who have the cancer, but to tailor their treatment strategies are the important aspect of modern oncologic practice. However, one of the challenges faced by clinicians is the appropriate understanding of genetics and the availability of genetic counseling and genetic testing service in Pakistan. So I do not have any financial disclosure. So in this talk, I will walk you through about the basic concepts of cancer genetics. What are the difference of germline versus somatic mutations, genetic testing strategies, and why family history is important, and what is the concept of personalized medicine, with 2 examples from my side about autosomal dominant and 1 example by my colleague, Ms. Aisha, about autosomal recessive genes. Today is the World Cancer Day, about cancer awareness, 4th February, and Pakistan, based on the available data, about 180,000 new cancer cases are being diagnosed each year. And these are the top 5 cancers. Breast cancer is the highest. Second is oral cancer, lip oral cavity, lung and esophageal cancers, and colorectal cancers. So what is a cancer? It occurs when a body cells continue to divide uncontrollably. So every one of us has a breakdown and repair process, but there is an uncontrolled growth which could lead to tumor. It is of 2 types: benign, which would not spread to the other parts, or malignant, it would spread to different parts of the body. So cancer occurs primarily due to genetic changes within the cell, and our cells are made up of billions of cells. Our bodies are made up of billions of cells, and each cell contains the complete genetic print, including information by DNA. So chromosomes are the thread-like structure present in the nucleus, except RBCs, which contains DNA and protein. And from chromosomes, there are DNA which contains instructions for growth and development, and it is packed in chromosomes. that contains all our genes. Genes are the basic unit of heredity. It provides instruction for growth and development. For example, instruction how our eye color would appear. And there are, as Dr. Salman mentioned, only 0.1% differences among individuals, which play a role how our body would respond to certain diseases or develop accordingly. In total, about 20,000 gene pairs in every one of us. One copy comes from the mother's side, second copy comes from the father's side. So here you can see in general, broadly, these are stratified into 3 different groups. Like a car, there are brakes. If brake fails, there are more chances to have accident, like tumor suppressor genes. Or if there is accelerator or genes are switched on, there are more chances for accidents. And mismatch repair genes. So if the repair mechanism is not properly functioning, that could also lead to cancer. The balance of cell growth and division is maintained by these cancer protection genes, just like a well-maintained car. And cancer may occur when these genes mutation result in failure to repair these mistakes. And here we look for spelling mistakes like mutations. Here you see gene spelling. If it would change, then you would detect like with a spell check in the computer program. So we have to look into a spell check throughout the coding sequence of any gene of interest. What are mutations or variant? Mutations are the molecular alteration in the DNA sequence. It is primarily there are 3 different effects: deleterious or disease causative, also called as pathogenic variants, or benign or neutral variants. And the third one is variant of unknown significance. For those variants, nothing is known or little is known to characterize them accordingly. So primarily genetic conditions are caused by these genetic changes. So here you see 2 copies. One allele has an appropriate copy with appropriate gene product is expressed, but the other one is a mutation that could lead to no product, reduced product, or incorrect gene product that would lead to disease. So what are the differences of germline mutations versus somatic mutation? Dr. Ajaz has already discussed about this. Germline mutations leads to hereditary cancer and somatic mutations lead to sporadic cancers. Germline mutations occur when there is a mutation in the germ cell in egg or sperm, and from here all body cells would harbor the same mutations. So that may cause an individual to develop a condition or a disease. Whereas acquired mutations, it occurs during lifetimes, spontaneous or due to some radiation or chemical exposure in somatic cells. Here you see the fertilized egg has the normal body cells, but only in one tissue, in breast, there is a somatic mutation. So disease will only appear in breast tissue. So for germ cell mutations, germline mutation, so every cell will harbor that specific genetic change and it would be transmitted to the next generation. Whereas in somatic cells, only one part of the tissue or the primary will have. Here, for example, it's in ovarian cancer. So only in this area, the disease will appear. It will not be transmitted to the next generations. So every one of us harbors 2 copies of cancer protection genes. For hereditary tumors, so an individual is born with 1 mutated allele, whereas in sporadic tumor, there are no genetic change. And during lifetime, there would be second hit or acquired mutation that would lead to tumor. In the sporadic tumor, 2 hits will be acquired. For that reason, majority of sporadic tumors would appear at a later age as compared to hereditary tumors that would appear at a younger age. That is one key feature of hereditary tumors. Sorry. So having an increased risk of developing cancer compared to general population risk is primarily due to mutations in cancer protection genes. In general, there are 3 different types of groups. Hereditary tumors, it accounts for 5 to 10%. There are several affected members in the family. Second one is familial cancer group, which accounts for about 10 to 20% of all cancers. There are 1 or 2 individuals affected with malignancy. Majority of cancers are sporadic. So these sporadic tumors are presumed to be due to unknown polygenic predisposing gene interaction with environmental factors. So most cancers are not inherited. That is very important to understand. So genetic testing, hereditary versus somatic. Germline testing or hereditary testing and somatic is also called as tumor testing. The purpose of germline testing is to identify mutations that are inherited or harbored from their parents to the next generation. And tumor testing is to identify mutations specifically in the tumor tissue, means somatic cells. So germline testing, DNA could be passed to the next generation. For tumor testing, it not necessarily is passed to the next generation. The target tissue, how can you perform germline testing, is from blood, saliva, or cultured fibroblast for the hematologic malignancies and non-cancer cells. For tumor tissues, these are tumor tissues and liquid biopsies. Applications, there are clear applications for both kinds of testing. For germline testing, one can identify who are at risk, and next generations are also be tested. And it also helps to identify targeted therapies and risk prediction and genetic counseling and other at-risk family members testing. Tumor testing, it identifies the tissue type that a tumor originated from to help determine the appropriate treatment strategies. It could also help to identify alternate therapies and inclusion of potential clinical trials. It predicts prognosis or likelihood of recurrence and monitor the disease progression with liquid biopsies. So early detection is the better outcome. So the sooner you will identify, you can tailor the treatment strategies for those who are already affected or those who haven't developed the disease. So there are certain risk prediction parameters which could be adopted. I will discuss later. Cancer genetic testing is an integral part of modern oncologic practice. Primarily, it could be stratified into 2 different groups: diagnostic testing, mutation search, in which only the mutation is explored in an individual who is affected, and it may take considerable time. Second is predictive genetic testing, in which if a mutation is already found in a patient, then their at-risk healthy members could be targeted and screened for that family-specific mutations. It is very important to identify that genetic testing detects mutation, not disease. An individual has increased lifetime risk to develop a disease, but you are not 100% certain that that individual, the lady or the guy, would certainly develop disease. So it is an important aspect to be discussed in the pre-test counseling. What are the challenges, especially in Pakistan? It requires a very well-trained genetic counselors and health professionals who have the idea about genetics, up-to-date knowledge of cancer genetics. Like Dr. Salman mentioned, nowadays there's new terminologies, so you have to understand and keep you updated. And from the pedigrees, you can also differentiate autosomal dominant versus autosomal recessive inheritance, and the family history and understanding of family structure. So we reported several families in which about 80 individuals were from one village, all relatives died due to cancer. So in the West, certainly you would not observe such kind of large pedigrees. And we have already discussed about consanguinity. Pakistan has one of the high incidence of consanguinity in the world. Genetic testing should always be performed in a healthcare setting, and patient is the center. There needs to be interaction of genetic professionals and primary healthcare providers or oncologists. The purpose is to identify patients who are candidates for genetic testing If an individual developed, for example, breast cancer at age 60, as compared to another individual who have a strong family history and developing breast cancer at age 30, then there are more chances to have a genetic change in a younger patient. And how to effectively use this knowledge to improve disease prevention. I mean, it's like a child who has a opportunity to drive a car. So the car has a limit of 300 kilometer per hour. So you can drive it. So similarly, you have to identify what are the likely benefits for any genetic testing. There are several companies who just want to get to sell their tests. That's all. There is no pre-test counseling, no post-test counseling. So one has to understand, can you identify some actionable mutations? Are you going to help? Because every individual comes with a life and they have a whole family. And especially for females, females are the nucleus of any family. So we have to take into account this thing. So in general, cancer is due to three major reasons: genetics, behaviors, and environment, occupational or environmental hazards. So these three factors play a key role to cause any disease. And family history helps capture these interactions. According to WHO, about 40% of cancers are preventable by just modifying your lifestyle. So I think this is very important to recognize. So we all have a family history of something. The knowledge and understanding and the record of family history is very important. So what is a family history? So every individual, every family have some family members with some diseases. For example, some families have a number of blood relatives who have or had malignancies. Documenting families over several generations is very important. At least 3-generation family history is very important. A family history can be due to chance, because cancer is very common, or common environmental influences shared by all family members. Third, a germline mutation in a cancer protection genes. So you have to take into account 3 generations from your father's side and also mother's side. As Ms. Fizza mentioned, that some people just think that father's side is only the family. This is not the case. So what to ask on both sides? How the individual is related. For example, here, this lady comes to us. So we need to see, after taking this family history, what close relation she has. The type of cancers. Are there specific cancers? Age of cancer and age at death. That would also help us to understand the penetrance. And number of affected individuals in a pedigree, and how is the generation to generation transmission, as Dr. Ajaz Ahmed mentioned. So here you can see in every generation there is something, unfortunately. So this is autosomal dominant pattern. And family history is very dynamic. It's not that you just take the information once and forget about it. At least once in a year, at least you should keep on updating. There is a study from US, about 40% of US physicians do not ask about the family history. And you can imagine in Pakistan, it would be even worse. So genetic testing information is important. Who already had genetic testing? Are there results available, reports available? And based on that, you can offer to their at-risk relatives. So genetic counselors are trained individuals to take a targeted personal and family history and discuss which test is appropriate. What are the clues of hereditary cancer predisposition? It includes, these are the red flags: large number of affected individuals, more than one generation affected, clustering of specific cancers. For example, bilateral breast cancer or breast and ovarian cancer, or colorectal cancer with endometrial cancer. And early onset, as I mentioned earlier, hereditary versus sporadic. Early onset alert you to have some genetic predisposition. Unusual presentation like male breast cancer, and positive genetic testing results already from a family member. And based on that, you could offer And if a somatic testing is performed, then for their at-risk relatives, so you have to first perform germline testing, and then you can further offer predictive genetic testing to their relatives. So genetic testing strategy, which we are offering in Shaukat Khanam over the last 20 years, is to test the index patient. If there is no mutation, then direct testing is not useful. So do not proceed further. We are here to help out the patient and their families. We don't want to add more anxiety to the patient and their families. Second is, if a mutation is identified, then you can offer predictive genetic testing to their at-risk family members. Here you see a family pedigree. The index patient shows a breast cancer, bilateral breast cancer, in a Punjabi patient at age 42 and 49 with IDC grade 3 triple negative breast cancer, and there's a family history of 9 out of 52 individuals have cancers. So in total, 6 sisters were diagnosed with breast cancer below 50 years of age. One sister had uterine or stomach tumor at age 35. There are 2 other cancers, pharyngeal and chronic myeloid leukemia. So we offered genetic testing, and these 5 sisters harbored the same founder mutation in BRCA1. Then we test male just to see the pattern of transmission either from the father's side or mother's side, and the brother of these 6 sisters, because he has 5 daughters, and similarly other at-risk relatives. But we noted that mutation is being transmitted from the maternal side, and she is healthy at the age of 80. So you can appreciate that genetic testing predicts that the person could have mutation, have the mutation, but not give certainty that the person will develop a disease. Here, the brother is also found mutation positive, and 4 out of 5 daughters of that individual were mutation negatives. What does it mean? It means that these girls have the similar risk to develop a disease as any healthy woman sitting in this hall. And those who are mutation positive have an increased lifetime risk to develop cancer. So what are the take-home messages from this case? That genetic testing in this family, it gives info to the patient and family members. Another aspect which is very important to understand, not every individual in the family wants to know the result. So you have to ask whether the relative wants to know the result. If yes, then you can offer, not that just to share the results. And second is informed decision making is very important. Early detection for those who are at high risk harboring the mutation, because there is increased lifetime risk for BRCA1 carriers up to 80% to develop breast cancer and up to 60% to develop ovarian cancer. And for negative ones, imagine in this family, so there are 4 girls, there are 7 girls in this generation. So I have to counsel, I have to meet their other relatives, because for their marriages, they were concerned. So I told that they all have the same risk as any healthy woman living in this society. So they have all relieved anxiety and living a happy life. And follow-up support. This is very important. So for other services, it's entirely different. But for the genetic counseling, so there is as Ms. Fizza mentioned, there's a bond between not only the patient, the whole family. And that should be consolidated with the passage of time. Here you see the second case. This is the endometrial cancer developed in this lady at age 48, and at age 67, she developed breast cancer of Pathan origin, IDC grade 3, triple negative breast cancer. IHC was negative for MSH2 and MSH6, and a strong family history. 25 out of 58 individuals in this pedigree had cancers. So there are multiple affected, like her sister developed breast cancer, another sister developed breast cancer, one stomach, multiple breast and ovarian cancer. We suspected that this is HBOC family. So we offered BRCA1, 2, CHEK2, and 51C, all were tested negative. So here you can appreciate that multi-gene panel testing is very important in such cases. And there is a phenotypic overlap of HBOC and Lynch syndrome. What we found, an MSH2 germline mutation in this index patient, and another sister also harbored the same mutation, and her daughter, who developed ovarian cancer at age 43, also harbored the same mutation, and index patient daughter with endometrial cancer also harbored the same mutation. And one individual with prostate cancer was tested negative. And we also reported this earlier. So systemic treatment for cancer patients with mutations, as Dr. Salman has mentioned, there are several therapies available which are very expensive. But there are also strategies for those genetic patients available. Like this is one study published in Journal of Clinical Oncology. So several neoadjuvant chemotherapies were offered, but cisplatin, you can appreciate the response rate is 83% versus all other patients. So if you do not have any PARP inhibitor facility, or as it is very expensive, at least such patients could get benefit from platinum-based chemotherapy. So this is a question, will our new knowledge of genomics helpful? Would it revolutionize? Yes. Increased understanding of genomics raises hope that healthcare can become more personalized, hope that personalized medicine can improve the quality and decrease the cost. So Dr. Salman has already mentioned this one trial and error. So this is a reactive medical care. You diagnose cancer patients and keep on switching the medicine until patient dies or lost to follow-up. The new concept is right treatment for the right person at the right time. Right timing is also very important. And this is an efficient medical care. Health management, genetic testing, and tailoring the treatment strategies. What are the potential targeted therapies? It targets cancer on a molecular level, like somatic testing via NGS can help identify therapies based on their mutations. And multi-gene panel testing, it varies, primarily depends on the oncologist. So somatic gene testing includes like genes in mismatch repair and homologous recombination repair pathways. So one novel drug of homologous recombination pathway is PARP inhibitors. How it works? If there is a DNA damage, there would be single strand break. So PARP inhibitors interact with PARP present in the body, and that would lead to double strand break. And if there is in normal cells without any BRCA1 mutation, there would be a survival, apoptosis, and cell would survive. So these are highly effective and highly specific for cancer cells and non-toxic to healthy tissues. Whereas if HR deficient cells with BRCA1 mutations, that would lead to apoptosis or cell death. So the treatment strategies could be tailored based on genetic background. There are several targeted therapies based on NGS panels. So here you can appreciate several approved drugs which are being used with the application of NGS. There is another recent paper for those individuals who were identified using NGS for unknown primary cancers, for unknown primary malignancies. So that is really challenging for the oncologist. So which kind of treatment should be offered? But with NGS, so what they have done, 36% of patients got the targeted therapies, about 19% received immunotherapies, and still half of them, no treatment was available. So in conclusion, genetic testing is increasingly being used in the management of hereditary cancer worldwide, and genetic changes may contribute to a substantial proportion of hereditary cancer in Pakistan, understanding of cancer genetic testing and non-directive genetic counseling is prerequisite. So non-directive, so you can offer what are the risks and benefits for any kind of genetic test. You cannot force the patient that you must follow this thing. And with genetic testing, we need to have psychosocial support. This is very important. So you need to have some psychologist in your team who could also address issues which are quite serious in our population. So one of the patient came to me. So I always shared the genetic test result, but for one family, I did not share my results because as Dr. Salman just shared, that you have to feel the dynamics and the pulse of the emotions. So the husband was with that lady, and I felt if I would just share, he may just leave the girl immediately. So I did not share. I have to share with the primary physician those results. And she then, the oncologist, share with her and tailor the treatment strategies. So genetic testing for cancer susceptibility in Pakistan, it may allow physicians to identify individuals who are at risk and thereby allow them to tailor their preventive and therapeutic modalities based on their mutations. For BRCA1/2 carriers, so there are different options available, like prophylactic mastectomy, oophorectomy is available. Once an individual has completed the family, so then prophylactic oophorectomy or removal of ovaries could be offered in our healthcare setup. And predictive genetic testing to all at-risk relatives is important to make significant medical decisions and to avoid any kind of unnecessary intervention. As I have shown in one pedigree in which 7 girls were tested negative. So they do not need to undergo intensive surveillance or medical management. So both outcomes yield to benefit to the individuals and to the society. The increased access to genetic testing facilities is required. For this purpose, I think Pakistan Society of Medical Genetics is established, and we need to have a nationwide partnerships. At the end, I'm thankful to the study participants, my colleagues, clinicians, clinical research office, cancer registry, and my team in basic sciences. And from Pomeranian Medical University, they have the world's biggest BRCA database. So they have a nationwide genetic counseling service. And German Cancer Research Center, Professor Ute Hamann, and obviously the Union for Cancer International Control, Geneva. Thank you very much.
To the Pakistani Society of Medical Genetics and to Aga Khan University for having me speak today. I'll be continuing on the topic of hereditary cancer predisposition as presented to you by Dr. Rashid earlier. I am a salaried employee of Ambreen Genetics. So in continuation of the objectives for this particular talk, I'll be illustrating a risk assessment using a different case example for an autosomal recessive hereditary cancer predisposition syndrome, in particular, what's called constitutional mismatch repair deficiency, or for short CMMRD. And then we'll also just go ahead and review some psychosocial implications of hereditary cancer genetic testing to keep in mind. Okay, so to get right into it, let's just do a quick overview of what CMMRD is. CMMRD, or the long form is constitutional mismatch repair deficiency, is a rare childhood cancer predisposition syndrome. If you think those genes look familiar to you, that is because they do. These are some of the same genes that are also involved with Lynch syndrome that was presented to you earlier by Dr. Rashid. But what really sets this apart from Lynch syndrome is that CMMRD happens when there is biallelic mutations, or it's an autosomal recessive inheritance pattern. So what I mean by that is if you've got 2 copies of each of these genes, to have CMMRD, you must have a mutation on both copies of the particular gene that you inherited from each parent. So, for example, and we'll run through an example of a family history in just a bit, but MLH1, for example, we have 2 copies of it. 1 copy we get from each of our parents. So to have CMMRD, both of your parents must have had Lynch syndrome because one of their copies of the genes would have had a mutation. And so then to have CMMRD, you would have inherited both of the copies of the gene from each parent that had the mutation on it. Now, what you can imagine then, because this is a childhood cancer predisposition syndrome, it's a little bit more severe than Lynch syndrome, is you're going to see some of these cancer risks occur earlier on in life. So that's part of why we call it a childhood cancer predisposition syndrome. These cancer risks can include, and this is by no means an exhaustive list, but they can include things like colorectal cancer, certain types of brain tumors, blood cancers, particularly lymphomas and leukemias, cancers of the small intestine, urinary tract cancers, endometrial cancers. And just like in Lynch syndrome, how the risks look different depending on the gene, we may see some of that risk look a little bit different depending on what gene the CMMRD is related to, although I don't think that those have really been stratified at this time. One of the kind of hallmark features, if you may, is what's called these cafe au lait spots that we may identify on children. And these are just these kind of like patches on the skin with a little bit of a darker pigmentation, as you can see in this image here. These are by no means cancerous. They are just benign findings. And if you are familiar with neurofibromatosis type 1 or NF1, this is a kind of a hallmark feature you can see in those patients as well. But so often this can actually cause a little bit of confusion, and occasionally a child may be misdiagnosed with NF1 instead of CMMRD. But this is something that can also be seen just as the spectrum of what you may see with this condition. Now, let's kind of run through an example family history here for CMMRD. So, and I'm going to pull my laser pointer up here. So let's just see if I can get that to show. There we go. So this child right here, so she has a glioblastoma, and she was diagnosed at 9 years of age. So this child here, in this case, is kind of a course of concern. And one of the things I look at in her family history is that her father had a colon cancer at a young age, at the age of 45. And then the other things I note here is on her mother's side of the family, you've got both a colon and an endometrial cancer. So, you know, whoever would be assessing this family history and knows about Lynch syndrome would recognize these patterns and kind of see that that is of concern and potentially then link these things. Now, I also just kind of included an example in here is if you do actually get genetic testing performed. Let's see here. So here in this example now, you've got your MSH-associated CMMRD family, and I put kind of their testing status of here, just as an example to help you illustrate how this kind of can play out. So here, for example, like I said, you've got this child with glioblastoma in childhood, and so if we're looking at MSH6-associated CMMRD, we would have then expected, you know, if we had her genetic testing, that she had a mutation on both copies of her MSH6 gene that she inherited, both from her mother and her father. So if we were to test her parents, you would expect that they have each one of those mutations. So as you can see, both of their copies of their MSH6 gene, they've got a plus and a minus, indicating that there's a mutation on one copy and, you know, just kind of your wild type or your normal allele on the other. And So then two would then have, as you know, with MSH6, if you've got one copy of the mutation, you have Lynch syndrome. So these two parents then have Lynch syndrome, and their child, unfortunately, then has inherited both copies of the mutation. That is generally a 25% chance of happening if two people have a carry a particular mutation like this. It would be a 25% chance for the child to inherit both of those copies and end up having CMMRD, or the autosomal recessive condition. Now, just kind of to elaborate on this a little bit more, but one of the things I kind of put in this example pedigree is this double kind of dashed line, well, not dashed, but double solid line here. And this is basically an indication. It's a standard kind of notation on family histories for what's called consanguinity, meaning when you may have a relationship within the family. So what I mean by that is, these two people actually are related by blood. And so you can see that indicated here by this line here. So this person's mother was a sibling to this person's father. So they are first cousins. And this is something, this is the reason I kind of wanted to include this is because, of course, we can run into the clinic with this in the US as well, with certain populations a little bit more so than others. But in Pakistan, especially, if you're going to be practicing there, this is something to keep an eye out for, because you may see some more autosomal recessive conditions or kind of when you see some of these consanguineous families, especially if we have a known mutation in the family. So that is just indicated here by that double line. And so, yeah, so this is just a good example here of what that could look like. While we're looking at this pedigree, I also just like to point out a couple other things that can be helpful. I know Dr. Rashid already covered the things to look at in a family history, but just a couple other things to mention. In this family history, for example, if I was taking this in the clinic, some other things to keep in mind. So you've got this lung cancer here at 70. This is more just like a red herring. This is not something I'm concerned about when I'm doing risk assessment. We know, first of all, with lung cancer that for the most part, they are not because of an inherited predisposition. And, at age 70, it's not something by itself that is of concern. There's no syndromes that connect both lung cancer and colon cancer, so not something generally to be concerned about. And then, again, here we also see that this person right here, the grandmother of our patient on this side, she would have had Lynch syndrome had we done genetic testing on her. But you don't always have these testing statuses, right? So in this case, though, one thing to note is that, she did not have cancer, but she also passed away at age 35 of an accident. So sometimes having young deaths in the family, or when people haven't lived long enough, that can also mask the manifestations of certain syndromes. Now, with that being said, with Lynch syndrome, the risks are not 100%. I used to love telling my patients this when I was in the clinic. Sometimes people think that if they test positive, that's a guarantee to develop cancer. That is not the case. So this person could have very well lived to be much older and not have developed cancer either. But just kind of for the purpose of understanding how risk assessment can happen, these things get taken into account by someone who is trained to take these family histories. But not least, I just want to quickly cover some psychosocial implications of genetic testing with cancer risk. So one of the main things, of course, talking about cancer risk can be emotional, and it can bring out a variety of feelings. I like to remind people that every person's experience with cancer is different. Now think about this, right? You're talking to a family about cancer risk. Probably one of the reasons they are even seeing you to begin with may be because maybe their mother passed away of cancer, or they have a loved one that has passed away from cancer, or someone who is currently battling a cancer diagnosis. And so they may have some really raw feelings around talking about cancer, and it takes a really big step for them to even be coming and talking to you about that. So that's something always, worth noting and acknowledging in a session like this. It's also worth noting that cancer can be a taboo topic in many cultures. There's lots of times where people did not like to talk about their medical histories with other relatives. You know, we see this a lot in the United States, especially with older generations, and, you know, with Pakistani families as well. You know, I come myself from a Pakistani family. I know that it varies, of course, from family to family, but there can be families where maybe medical histories and things are just not things that people like to talk about. It's not information people like to share. People like to be secretive about it. And so that is another kind of consideration to have psychosocially when you're seeing these patients and talking to them about doing genetic testing for cancer risk. And then, of course, financial implications are also something to consider. So, you know, here in the US, we've got health insurance coverage for the most part for a lot of people, not everybody. But, you know, similarly in Pakistan, it would be worth considering, like your patient that you are seeing, do they have health insurance through their employer or through, you know, is it a kind of a national plan or is it institutional? You would kind of have to know some of these things as well, because at the end of the day, there may be a cost associated with the testing, and that can absolutely be a barrier to a lot of people. Or maybe it is sponsored testing and something that they may not have a cost about. But I will tell you that even as someone who's seen patients in the United States who have had health insurance coverage, if they may be going through a really difficult time, say they themselves have a cancer diagnosis and they're undergoing treatments and paying for a lot of that out of pocket as well, it can be really difficult for them maybe to consider paying for a genetic test in addition to that, even if they may not know whether they'll have a shared cost in that or not. So that's something to consider. Of course, there can also be implications of the results for other types of insurance and employment. Now, I don't know what this would look like in Pakistan specifically, but I know that here in the US, there are certain types of insurance plans like life insurance that may ask about whether you've had genetic testing before. And if you are positive, they may not offer you the same type of plans. They can discriminate based on that information. There are some laws in place that protect against discrimination from other entities like health insurance and other types of certain types of employment, not all of them, but those are not you know, completely covering everything. So things like that may be taken into consideration. And then I just wanted to highlight here that genetic counselors are trained to address some of these psychosocial needs that can arise in a session with the patient. You know, your patient is more than just their genetic test, they're more than just their diagnosis. And genetic counselors have that special training to look at the psychosocial and kind of their whole person a little bit more. But with that being said, the genetic counselors are not by any means a replacement of appropriate needs for social work or psychology to step in. So, you know, there may absolutely be times where a formal referral to social worker or psychologist may be needed instead, because maybe the needs of the patient may not be able to be met by the brief psychosocial counseling that can be done in a session. And that, again, would be something that typically genetic counselors are trained to kind of know where their boundaries are with that. All right, and so that's all that I had today. I look forward to seeing you in the question and answer. Thank you.
So since since Ms. Fatima is also here for Q&A, I would also like to invite Dr. Rashid back to answer some questions. And please raise your hand if you have any questions. We'll get to you with your mics.
Can you go back to the pedigree?
Which one?
Yeah, this one.
This one.
Yeah.
So, Miss Fatma, could you? Yeah, just a second. Yeah, it's here. Yeah. Can we also see the pedigree?
Yeah, can you show the presentation?
Maybe you can ask a question.
Amir, show the slide one option.
Yeah, in the pedigree, you showed that the grandparents have passed away, but still, and they were not diseased, even this one, yes. The grandmother is not diseased, but still she's shown as a carrier. So how can we confirm like she's heterozygous or carrier? How can you label her?
Great question. And this is for the CMMR deep pedigree, is that correct?
So in this situation, so the patient, the lady has passed away, but before that she was tested and she carried one mutated allele.
She was tested in the
Yeah,
last.
yeah, she was tested. Based on the results, it's mentioned here.
Okay.
Correct, Fatima?
Yeah, and actually, I kind of showed the testing status. This is not a real pedigree. This is a made up one. Most often, if that's so long ago, we're likely not going to have had genetic testing done on them. But this was just to show as an example, to show that there was a consanguineous family and who we would expect to be positive. But generally speaking, you're right. We wouldn't have information for grandparents who've passed away so young. But also part of showing that was that it's not uncommon for us to sometimes maybe see someone in the family who's passed away at a young age, and that can sometimes mask the presence of a variant if they say may not have had a cancer. But with that being said, kind of like Dr. Rashid mentioned as well and showed that real pedigree, having a genetic mutation is also not a guarantee of cancer either. So.
And I would like to add that in this family pedigree, so if you see the colon cancer, 2 colon cancer were diagnosed at age 45 and 59, and 1 endometrial cancer at age 53. So above 45 years of age, all these cancers were diagnosed. And this lady, the grandmom, passed away at age 35. So as Ms. Fatma mentioned, so that was just masked because she unfortunately had some road traffic accident and she passed away. She might have developed cancer, may or may not be. Can you please show the pedigree so that is there anyone just to share the pedigree? Can you show from here? Sorry?
While we are setting up the pedigree, Dr. Usman, okay, let's clarify this.
So you can appreciate that for the family, comprehensive information is important. This is a 3-generation family pedigree, and you can see which cancers has appeared. Colon cancer at 59, endometrial 53, colon 45. So she passed away. This is theoretical pedigree, but this is also not uncommon. So she passed away at age 35, and she was tested, and she carried 1 allele. For autosomal recessive, you have to both copies need to be mutated to develop a constitutional mismatch repair deficiency syndrome. Like this girl developed glioblastoma at age 9. I hope that answers the question. Just focus on the concept at the moment. So, yeah, please.
Thank you for the presentation. Very interesting and very elaborate. एक आध बड़े basic से questions हैं कि because we are not cancer specialists, so we are not really familiar. But I want to know that what number of conditions are now being treated with targeted therapy in Pakistan, and what kind of a progress in the next 10 years do you see that happening, and what's the cost difference in terms of a kind of conventional chemotherapy versus a targeted therapy?
Very good question. Like for the conventional chemotherapy and the targeted platinum-based chemotherapy, this is as cheaper as other conventional therapies. And for BRCA1/2 mutation carriers, those BRCA-associated tumors are more responsive to platinum-based chemotherapies. Like from this JCO paper from Polish group, they reported 80% response rate in neoadjuvant setting. But the other targeted therapies are quite expensive. Few of them, the cost Dr. Salman has mentioned. But we are hoping that it would reduce the cost with time. For PARP inhibitors, those therapies, so you don't need to be admitted on the day basis just to get the chemotherapy. It's on the oral tablet form, so you can take the tablet. This is being used for the metastatic cancers, second line for breast cancer, ovarian cancer, and prostate cancers. And several other trials are ongoing. And for some Gefitinib or some other targeted therapies are available, but to know exactly the cost, so I mean, I cannot comment on that. These are relatively expensive, but we are hoping that with time, it would reduce.
Thank you, Dr. Usman. It was a wonderful talk. I have just a small question, short question, that there are several environmental factors which are going around us, like there are endocrine disruptors, and there are certain chemicals which are causing but they're thought to cause some cancers. So how do you relate with the genetic things? Are the environmental factors are causing these mutations, or what is going on?
So for genetic things,
Otherwise, cancer is has become so common.
yeah, cancer is common, and there are three major reasons, like environmental factors, lifestyle, and third is the genetic. There is a close interaction. For hereditary tumors, a person is born with with an inactivated allele. And for others, there are some, for example, radiation exposure, some contaminants in air, water, or food, dietary habits, lack of physical activity, obesity, hormonal therapies like age at menarche, menopause, history of parity, breastfeeding. So these all factors contribute to develop in the inactivation of the second allele. So we, on germline testing, we focus on the blood sample and we check that one. In the tumor tissue, there is inactivation of that second allele. With those cases who have the germline mutation, they have the increased lifetime risk, but we are not certain that individual will develop. For those individuals, if there is a cancer already in the pedigree, so we can tell that all cancers developed at or after age 40. So according to European guidelines, one can offer intensive surveillance strategies for healthy at-risk individuals who were found tested positive. So at least 10 years earlier, they could undergo surveillance strategies. And once they complete the family, then there are surgical options available. Bilateral prophylactic mastectomy, for example, which would reduce up to 95% not to develop breast cancer, or prophylactic oophorectomy, which would reduce up to 95% not to develop ovarian cancer, and up to 50% not to develop breast cancer. And there are other oral contraceptives and other therapies available.
Okay, so we'll take one last question from the audience and then we'll move on. Yeah, please go ahead.
Assalamu alaikum. Sir, as a student, my question is there any technique by which I can compare, if my father is a carrier, okay, so I can compare my DNA with him, so I can determine either I'm a carrier or not.
So how would you?
Is there any technique? My question is then.
So there are several techniques.
Yes.
Like यहाँ आगा खान auditorium में आने के different approaches हैं. सिर्फ एक रास्ता नहीं है. So you have to first be sure कि आपके जो father को है, he is really positive for a specific genetic change, which could be associated with the disease. Every one of us, अगर आप किसी का भी sample लेंगे, there would be genetic changes, but not every genetic change could be pathogenic. Even pathogenic variants not necessarily would lead to cancer or any other disease. पहले आपने ये देखना है, you have to be certain that your father have harbored a genetic change, number one. Number two, then based on that, your sample could be screened for that specific change. It doesn't mean अगर आप में नहीं है तो आपके किसी भाई में नहीं होगा या बहन में नहीं होगा. So all those individuals also needs to be tested accordingly. But there are several techniques which could be utilized, like Sanger sequencing, for example. Thank you very much.
So thank you so much, Dr. Rashid and Dr. Fatima, for this enriching talk. So without any further ado, I would now like to invite Dr. Rizwan Naeem. Just a brief introduction though. He's a professor of pathology at the Albert Einstein College of Medicine, and he serves as a founding program director for the Laboratory Genetics Fellowship Program. He's a graduate of the Sindh Medical College here in Karachi, and he's an executive director of Sindh Medical College alumni. Currently, Dr. Naeem is serving as president-elect for the Counseling of Program Directors at the Association of Molecular Pathologists. Over to you, sir.
Thank you. Again, once again, it's really heartwarming to see so many students and so many people interested in the mission of PSMG, that how can we increase awareness and do what we can to decrease the disease burden using genetic testing. So let's start to it. I have no financial interest or no disclosure material or financial interest that related to this talk. So genomic medicine, we have heard a lot about it today. And what I'm going to do is to just give you a basic concept. So what it is, that why and how we do it. I mean, that there are many different technologies, Dr. Saab was saying a few minutes ago, that for testing a known genetic mutation versus a new genetic mutation, there are many different ways you can do the testing. How do we decide what is right test and methodology? And you have heard so many methodology. Dr. Ajaz talked about RACGH and so on and so on. But who decides that? What is the right methodology to do the testing? And that's what we're going to talk. And then the benefit of testing, individual benefits and the community-wide. And can we use genetic as a preventive medicine? So these are the topics we'll be covering. I hope this lecture will provide an overview of genetic testing as a tool that can serve many individualized research and clinical needs, basic understanding of what is a much more complex topic. So again, genetic testing is a very complex topic, but what I'm trying to do is to simplify it. The intentionally glosses over finer details in favor of simplicity. Often, cartoonishly simple understanding of concept. What this presentation will not cover: a comprehensive review of all the application of genetic testing and understanding of actual steps and effort need to perform genetic testing in the laboratory. So one concept is that genetic testing is very uncommon or uncommon. So I will show you why genetic testing is not uncommon and how common it is, and with some of the data. So about 200 pediatrics diseases are being identified due to extra or missing chromosome, which Dr. Ajaz very finely introduced, that Down syndrome or Prader-Willi syndrome or DiGeorge syndrome or Turner syndrome and many, many others, at least 200 and maybe more. Hallmarks are multiple major or minor anomalies, usually appearance with the mental disability. So extra chromosomal material or loss of chromosomal material, one thing that has common is mental disability or syndromic finding. Point mutation and exon level alteration can cause single gene disorders or syndromic finding also, which was also discussed, that one gene, one protein concept, which was developed many years ago, is totally gone now. One gene can be syndromic or multiple genes can be single type of disorder. So this is a little old data, but nearly 10,000 conditions can be identified, medical condition can be identified using genetic test. And this data is continued to increase. This is a data from number of kits sold in United States for genetic testing, right? It is not infectious disease testing. This is genetic testing, and genetic testing, as you can see, that really keep going up, and that is because of human genome cloning. And now we know the human genome a little more. We can develop different approaches for testing different things and use different type of kits to do the testing. So how the genetic testing impact disease, right? Salman talked about that sometime it's just very important to know a closure for our family that this is why your family has certain disorder or certain disorder, the impact. So this graph, if you look at it carefully, what it's really telling you that there are new treatments implemented, right? So once you develop a diagnosis, especially in cancer and now in some non-cancer cases also, you know you can use a certain disease, medication or treatment to help make a diagnosis, something called co-diagnostic test that different mutation can cause, can be used for different testing. It also help you refer to a specialized referral. If you don't have a genetic testing and you have just common syndromic finding, you don't know which specialty or consultant that patient need to go. Once you identify the variant, then you can divert that. Yes, this is a cardiac variant, for example, and you go and see a cardiac geneticist to see how things are going. Similar thing is neurological, similar thing is skeletal dysplasia, or so on and so on. So identifying a variant which is relevant to disease helps you divert patient to that particular specialty. Then the very important part, as we were discussing today, that prenatal diagnosis, right? Unless once you know the variant you have find in a proband or there are many diseases where you can use in a prenatal diagnosis, simple tests using amniocentesis or CVS, for example. Thalassemia is a very common example in Pakistan, as you know, but many other chromosomal and syndromic disorders can be diagnosed before baby is born. And at least you give that option to a family to decide what they decide in case they have a child. They can be told that this is the issue on site. So, for example, I was talking today from the Down Syndrome Society, and I was very surprised that 21,000 families have been registered in Down Syndrome Society. So what that means, I don't know how many of those women even offered prenatal testing or any type of genetic testing, right? Would that have changed the decision if they knew that their child will be a different type of phenotype? Down syndromes are very good kids, and now they're they need specialized assistant, but that should be the choice of that family, not to just have them in a society without any discussions or without any informed consent about what's going to happen. So we also discussed that some of the predictive testing, which is becoming more and more common, the cancer genetic is one example that where you can do some of the predictive thing, and that predictive testing can help decrease the disease society, or at least monitor that person, that how before they develop severe disease, they can be interrupted, or disease can be provide some counseling or some options for that case. So about 60%, 61%, 61.13% genetic disorders are we are talking about now variants. So if you are, this is the data from genome sequencing, or sorry, exome sequencing, and about 18% cases still have no diagnosis, which we have seen at PSMG. Many of the patients, we come back and have a severe phenotype or sometime, but there is no variant at present identified which can be used, because we are not testing intronic variant at present, right? So many of the speakers have discussed what is the difference between exomic sequences, or functional DNA variants, and intronic variants, which we usually think that they are non-relevant. but many of them have shown that they are relevant in one way or the other. Then about 20% of the cases are variant of unknown significance. We don't know enough because we are not testing that many people to know if this variant has been seen in a disease, as was explained in a cancer genetic testing, that even if you have a variant, that will take some time for certain diseases to show up as a disease-causing variant. But likely pathogenic and pathogenic, 60% of the variant identified so far are likely pathogenic or pathogenic. That's a pretty good yield. If you, out of 100 tests you have sent, and there are 60 of them are showing you results which can be used in one way or the other, different ways, treatment implementation, referral, and prenatal diagnosis or predictive testing, that's a pretty good yield for the genetic testing. So there's another concept that I cannot do genetic testing because it's very expensive, right? So yes, it is expensive in certain cases, but Salman talked about how $100 genome, and that day is coming where you can just do one genomic test. And think about that. The expensive is not that particular test. If you do that test once, that's a lifetime test, right? So you don't have to do that test again. Once the baby is born, you do that testing, and that data, she or he can carry it in a thumb drive or maybe in a chip in her hand or somewhere inserted in a body, and you scan it and you will see that what variants you have. And by that time, hopefully we'll have much less variants of unknown significance versus variants of, we know what these variants are doing and how they are pretending. So this was an interesting concept, as someone has also spoken, that if first time human cloning cost $3 billion, right? $2.7 billion. So that's a very expensive ordeal. Now the next generation technology, a human genome can be sequenced for less than 10,000, and the foreseeable future, the cost should be reached 1,000 and hopefully less than $100. Actually, I was talking to someone yesterday that Chinese company somewhere is offering a syndromic type of or panel type of testing in $150. So that's cost is keep coming down. That graph is just showing the cost per raw megabase of DNA sequence, right? And see how, and this is again a very little old data, but you can just appreciate the cost is coming down. So it's not an expensive testing. So let's talk about Pakistan, right? This was when I last visited Pakistan. I was looking at Dawn, that was the Dawn newspaper, and there was a genetic conference for the awareness of genetic diseases among children. So this is a Dawn report saying that the genetic disease is in most cases serious, chronic, debilitating, and life-threatening, often requiring long and specialized treatment. Dr. Huma, who is also our partner in this syndromic type of testing, has done a lot of good work in Lahore. That conference was, that was her statement. Then the genetic diseases are responsible for about 50% of death of children before the age of 1 year in Pakistan, right? So that is a very alarming thing. Almost 50% of the kids you let you die because they cannot do genetic testing or because of genetic diseases. So we need to be a little more aware of these facts. Responsible for a significant handicap in the older children who have muscular CNS disorder. So there are much less issues with the accident or broken bone or so on and so on. The genetic cause is a very important cause of muscular or CNS diseases. And these are, they were making awareness that after infectious diseases, again, this is another interesting fact, that after infectious diseases and malnutrition, genetic diseases are the biggest cause of morbidity and mortality in children. A very high prevalence of inherited or genetic disease in children. This includes diseases of the liver, heart, kidney, and brain. Physicians caring for children many times do not have the required knowledge or expertise to guide families with such disorder. And that's the point I think geneticists are making again and again, that once you have a genetic testing, we need more people who understand genetics. We need more physicians to understand genetics. We need more genetic counselors to understand these diseases and guide these patients to an appropriate treatment or follow-up. So genetic diseases affect 5 to 10% of the children's, and that's again data from United States. And this graph is basically showing as number of diseases, for example, aggregate frequency, for example, shortened lifespan, or major handicap. So all these diseases, chromosomal disorder, Mendelian disorder, multifactorial disorder, metabolic errors, syndromes, and isolated anomalies, they all contribute to a major handicap and shortened lifespan and aggregate frequency. So these things can be reduced, right? These things can be reduced by genetic testing. And my objective today is to convince you that doing genetic testing will reduce disease burden in society on many different ways. Of course, death, of course, if you have one family with muscular disorder, that family is suffered a lot already, and that changes their perspective of future life. And of course, their SIBs, who may be very, very normal, also suffer because of that family member. So what different type of genetic testings are done? For example, diagnostic testing. There's a different diagnostic test, as Fizza has said, that it's different than a screening test, right? A screening test gives you high risk or low risk category, that you are at high risk. Now, if you are at high risk, now you go for confirmatory testing or diagnostic testing. Then there is a pre-symptomatic and predictive testing, right? That is also available in many of the diseases and help as to physician or a genetic counselor to at least offer that information to families and to patients. Infertility, multiple miscarriages, that's very common in Pakistan, as you must have seen. And one of the common reason for that is also consanguineous marriages, right? So what happens that if you have a consanguineous marriages and two parents knowing or unknowingly carrying autosomal recessive disorder, some of these disorders are lethal. And if they are lethal, they will abort no matter what, and usually first trimester, second trimester. So and who suffers through that? Women, right? Because they don't know that they are carrying autosomal recessive disorder, and no one knows that the partner is also carrying an autosomal recessive disorder. So if we can make this awareness, you will have much better women healthcare also, that why women has to go through this abortion or termination or unknowingly spontaneous even abortion, and then you do a testing before they get conceived, so you can decide what your options are. Pharmacogenomics, as Salman said very well, that these, if you carry your genomic sequence with you, you will also know which medicine might work better for you or which medicine may not work better for you. And that's coming by the medical students who are there. When I was at medical school, there was no concept about many of these things. Genetic was just, it was a subject. Only thing we knew that, yes, in CML, there's a small chromosome called Philadelphia chromosome. And that also we remember because it's the name of a city why it was diagnosed. So now things that within our lifetime, things have changed dramatically. And I can assure you, all the medical students here, that within their lifetime, things will exponentially increase. And this knowledge base is very, very important. As Myla and Aisha has mentioned, that we did a survey of medical schools students to see what is the awareness of genetic testing, genetic conditions in medical school. So we are going to talk today about curriculum change. Unless you need to train and teach these students now so they are ready for tomorrow's diagnostic work. And then prenatal testing, and then screening, prenatal, newborn, blood transfusion, pre-implantation testing, and cancer diagnosis, prognosis, and treatment. So all these things are possible due to genetic testing. So now, I love this slide, and I've been using it for many years, and it just tells you that how the genetic testing is done and what you are looking for. The conceptual wise, when I was a student, really, when I was looking at this is the chromosome, right? So you count the chromosome, and if there are 45 and there is no Y chromosome, you said this is Turner syndrome. If you count the chromosome as 27 and there's an extra small chromosome, we used to call it, it's a Down syndrome. And if there are 3X chromosome, we call it 3X chromosome. There's no name for that for now. So and Klinefelter syndrome. So the major changes of chromosomes we used to do by just counting the chromosome. Then I was a fellow actually when we started using high resolution chromosomes. Right? What high resolution, this was accidental discovery that someone in a lab used trypsin on a culture really. And when they dropped the slide and stained with Ginza, which we used to do, they said, oh my God, what is this happening that there are bands now? There are light bands and dark bands. So what they realized that this material is made of protein. Right? The genes are packed within proteins, and proteins, if you treat it with trypsin or any enzyme, digestive enzyme, it dissolve or make it light or dark. So dark regions are actually are highly packed proteins, so they don't digest that well. And light regions are protein also, but covered less with the protein, so they lose some of the protein, and that's why these bands are discovered. And then due to these bands, many syndromic chromosomal finding came to life. For example, Prader-Willi syndrome and Angelman syndrome, which was a very confusing thing, and that there is the same type of small deletion in chromosome 15, and sometimes phenotype as Angelman syndrome, a happy kid walking around, and sometimes same deletion is seen by high resolution chromosome banding, and the patient is Prader-Willi syndrome with much bulkier and eating disorders and so on and so on. So this was a revolution, as a matter of fact. There are many papers came out, wow, this will change things, and now we can look at chromosome at much higher state. Then we we started digesting chromosome. Right? Well, DNA, we extract DNA and we digest them and break them apart and run on a gel. So by running on a gel, and different digestive enzyme gives you different segment sizes of chromosome. And by just running the segment sizes on gel, you can say that, yes, this protein, this DNA is this size, and it is cutting DNA at the certain size, and that's how we diagnose sickle cell anemia, for example, was diagnosed because of this. And still you can use southern blots, used to be a technology which no one is using anymore, but that was a revolution. Now we are looking at genome level, but the real revolution really happened. And then back clone and yak clones. What clones are really that when you digest pieces of DNA, when you digest the pieces of DNA, for example, right, you can put these pieces of DNA into BACs, bacterial artificial chromosome, or yak, yeast artificial chromosome, and then they grow. So you have a higher concentration of small pieces of DNA, and then you can digest that BAC or yak shell, and then you can probe with fish. Fish probe actually initially started with these BAC clones and yak clones. So you can look at very specific segment of DNA for the presence or absence. And I'll show you some example. But the real revolution really happens when human genome was cloned. So we said, this is the DNA code, and now you can go look at it and interpret it as you wish. You can cut them into pieces. You can amplify certain region. You can do much better studies using the database we have for CGH, and that's called sequencing. And now that is called next gen sequencing. So it is the example I gave that it's like looking at moon when humans went to moon, right? You can see the moon from here, and that's, you know, sometime you see someone जो है वो चरखी चला रही है, कोई खातून बैठी हुई है, कुछ भी हो रहा है. But you know, that's like looking at chromosome. But when you land on at moon, and now looking at rocks, individual rocks and individual things, that's like sequencing. The same thing you can look at very different ways that how, and that's what genetic testing is. Someone need to decide what is the right test. And that someone is people like Fizza and Ajaz and all the other geneticists who can tell you that what it is. And this is an example of evolution again, that chromosomes are like this, then fish came into life, and then segment analysis came into life, and then sequencing, Sanger sequencing, real-time PCR, and so on and so on. These are all good technologies. We use these technologies still in our lab for a particular reason. But the real, and this is how the chromosomes are done. So it is still 20 years ago, someone said that chromosomes are done. We don't need chromosome analysis because there is no use for it. Now we have DNA sequencing. No, that's not true. This still is very commonly used and one of the common tests done, but it's a very cumbersome test. You start your culture, you harvest your culture, you drop your slide, you stain your slide, digest your slide, you spread your slide onto glass slide, and then you start looking at it in the microscope, and then you align these chromosomes. So it takes weeks. Okay, so this is what chromosomes are done. But when chromosomes we are testing, sometimes we come across this type of scenario where you have a 47 chromosome, but there's a chromosome. Anyone can identify this chromosome? No one can. I cannot. I've been doing it for 30 years, right? That's why it is called marker chromosome, and they're placed nowhere. So what do we do? Maybe we can do FISH. FISH is a very simple technology. What it is that you have a double-stranded DNA, you denature it, you spread them apart, then you have a probe which you hybridize and label it with a dye, and this probe will match at a very specific site on DNA strand, and then you wash away the rest of the probe, and you will see the signal, presence or absence of signal. So that's a technology. Array CGH also using the similar technology, but we can diagnose many deletions, amplifications, translocation using FISH, and these are many different variants of FISH. And the one which was very popular was this Chi karyotyping, where different chromosome can be stained with different colors, but there is no use for it. So coming back to the same, coming back to the same, this marker chromosome, what do we do? And it's a prenatal case, and that's the case we published also. It was when I was at Baylor, that a patient is pregnant 12 or 13 weeks, and then we need to see what impact this marker can have. So that's when the array CGH came into life, right? So what array CGH basically do is you extract DNA from all these chromosomes and label, use a test slide also. And just because and I'm not going to go in detail, Ajaz already talked about it. Basically, it's a whole genome testing, but at a very, very high resolution, right? And that's what it is. And it works on the same technology as FISH. You use the genomic DNA, you don't culture that DNA, and you label by different colors, you can identify this. And so the same chromosome, remember this chromosome? So this chromosome actually a combination of chromosome 15 and 16, and array CGH tells you that who would have thought, right? There's no concept that there'll be a trisomy of combination of chromosome 15 and 16. And yes, that's what it was. So the family was informed in timely manner because of RACGH, and then they make their decision, whatever there is. But that's why these things are so important. So chromosomal microarray, multiple congenital anomalies, it's very, very good in multiple congenital anomaly, dysmorphic feature, intellectual disabilities, development, and autism spectrum disorder. Once science has start using RACGH, the diagnostic yield of these type of patients increased 20%, right? That's a very good thing. And this is, DNA alteration, for example, different type of DNA alteration can identify point mutation, deletion, insertions, and repeat alterations type of thing. And Sanger sequencing. Sanger sequencing advantage, there are many good advantages and disadvantages, but Sanger sequencing is a very good test for identifying, confirming the variant you see in next-gen sequencing. So what is next-gen sequencing, right? That's a simplified concept. And how many of you are doing next-gen sequencing? I know 1, 2, 3, 4, right? So for those who are not aware of this, it's a very simple process also. You do your DNA extraction, you do your library prep. What library prep is that you fragment this DNA into small pieces and then amplify with known ligation on both ends to amplify these regions. And once you are done that, then you start sequencing, and sequencing gives you FAST-Q file, which are digital imaging file. Then you convert them into BAM file, and then VCF files where you start looking at variants. So how do we compare Sanger sequencing and next-gen sequencing? Next-gen sequencing, multiple region of multiple sample at the same time. So the advantage of next-gen sequencing is you can pool 10, 20, 16, 30 samples at some time and sequence them all together because you ligated your address at the end of the segment. So each patient has his own identity and you can separate them as you move on. But, so let's look at the separate reads produced for each molecule of DNA sequence, very quantitative, highly complex, faster and less expensive when sequencing large region of DNA analysis, much more automated. Because these analyses are the most difficult part, but they are software who help that. Sanger sequencing, region of one sample at a time, one sequence read produced, an average of all DNA molecule, not very quantitative, less complex, faster and less expensive for sequencing. It is good for small stuff and right. And then there are different type of DNA sequencing and next-gen sequencing, intronic, exonic, panel sequencing. Changing cost of DNA sequencing also, you can see that. So I'm not going to end. My time is cutting short. The question was, why can't just we do DNA sequencing then? Because DNA sequence is not a simplified test. It's like a hammer. For example, if you're using a hammer, you don't know which type of hammer you're used for what work you're doing. That's why the geneticist and genetic counselor are important to help you to use the right hammer. So we also talked about somatic versus germline. Not going to do that, but that is an important concept to remember. And the balancing act, I think that's what geneticists are, to see what test is offered and how it is offered. In the end, pros and cons. Application is also next-gen sequence application, and microbial diagnostic and next-gen sequencing is very important, and especially the COVID sequencing is important. So we knew that sequencing changes can change the phenotype or genotype of that chromosome. So in the end, last slide, take-home lesson really is biology and a very foundation of genomics teaches us that successful organisms are successful. If we call ourselves organism, the successful organisms are successful because they do one thing very common, very well, they adopt. So we as a diagnostician need to adopt, and that adoption will help us survive. Thank you so much for the time.
So thank you so much, Dr. Naeem, for that insightful discussion on laboratory genetics. We'll be opening the floor to any questions from the audience now. Is there anyone? Yep, just one. Would we have any online questions then?
Oh, yes.
Yep.
Thank you.
Thank you.
So just a little bit of change in schedule, if you all will allow us. I know that there's a tea break scheduled right now, but if, in the interest of everyone's time, what we could do is we could carry on with the schedule, and we can do the abstract presentations right now, go on to the panel discussion, and then we can have an early tea, and then everyone can get to go home early. How does that sound? Yes. Okay, good, wonderful. Thank you so much for that support. So we will begin here.
So we'll be starting with the oral abstract presentations now. So previously we had Dr. Kulsum joining us, but because of logistic issues, she wouldn't be able to join us. In her stead, we have Dr. Rutaba Gul from Al Shifa Trust, who will be presenting now. Just an update before we begin. So just so it's out there, every presenter will have 7 minutes to present, and then 3 minutes for question and answers. So please be mindful of your time. Thank you.
Thank you so much. Thank you so much, Al Khan University and Pakistan Society of Medical Genetics for providing such an marvelous opportunity. Today, I'm going to present one of my paper that is actually identifying the genetic causes of phenotypically diagnosed Pakistani mucopolysaccharidosis patients by whole genome sequencing. So everybody knows that lysosomes are firstly discovered by Christian Duduve as the disucide bags of the cell. And there are many proteins that are destined for lysosome. They are tagged with mannose 6-phosphate, and then they just transported to the lysosome. I'm not going in much depth. So lysosome has a number of different enzymes encoded by genes to maintain the internal homeostasis. Disruption of lysosomal enzymes result in accumulation of substrate, storage in the lumen. So there are some non-enzymatic proteins as well in the lysosomes. There are integral membrane protein, trafficking protein, and regulatory proteins. If any of the lysosomal protein, either enzymatic or non-enzymatic, is malfunctioned due to some mutational changes in the genome, then they are collectively known as lysosomal storage disorders. LSDs are included in the broad category of inborn errors of metabolism. LSDs are very rare. They are affecting 1 in 7,000 to 1 in 8,000 live births. Depending upon the number of enzyme, transporter, and regulator, there are more than 60 subtypes of lysosomal storage disorders reported so far. Most LSDs are inherited as autosomal recessive, except for Fabry, Danon, and Hunter syndrome that shows X-linked inheritance pattern. There are some common symptoms in LSDs, including delay in achieving milestone, bone deformities, neurological manifestation, and multiple organ failures. LSDs are diagnosed initially through, especially those ones in which there are the mutations in lysosomal protein. They are diagnosed mostly from the leukocyte measurements of enzyme levels from leukocytes, bone marrow testing, and genetic analysis. And treatment options include enzyme replacement therapy, bone marrow transplant, hematopoietic stem cell transplant, pharmacological chaperone therapy, substrate reduction therapy, and gene therapy. They are in their clinical trials. In Pakistan, we have limitation in diagnosis and treatment. So the methodology includes ethical approval from the institute, then affected families recruitment and samplings, their DNA extraction through phenylchloroform method. Remaining families were subjected, some of the families were subjected in for Sanger sequencing. And then rest, they were all performed at Qaid-e-Azam University, Islamabad. And then remaining families were subjected to whole genome sequencing at Novo Nordisk Foundation Center for Basic Metabolic Research, Copenhagen, Denmark, and then data compilation and write-up. So initially, we categorized all subtypes according to their number, mucopolysaccharidosis type 1 and type 2. They are both included in this paper. Mucopolysaccharidosis type 1 is actually autosomal recessive. And here we have the deficiency of enzyme alpha iduronidase. This is located on gene IDUA. And actually, that enzyme is involved in the degradation of heparin sulfate and dermatan sulfate. This is very rare, affecting 1 in out of 100,000 live births. And there are 3 phenotypes: Hurler, the most severe form, Hurler-She, the intermediate one, and the She mildest for all. But they can only be categorized after their genotype analysis. So these are the collected families, recruited families. I'm not going to show every detail in such a time shortage. So these are 12 families. You can see here the overlapping phenotypes, as I mentioned, delay in achieving milestones, their abdominal distension, hernia, and many more. So these are the variants that we identified after whole genome sequencing. You can see in the first, we recruited the family as MPS 1, but it has shown mutation in GALNS gene, which is actually involved in causing mucopolysaccharidosis type 4. Then IDUA, this gene is actually causing MPS 1. Then in the third family, we see polygenic inheritance. In this family, the first variant in homozygous form is in SGSH gene, which is actually causing Sanfilippo syndrome, and then GAA next. Then in the fourth family, this is ABCA5 gene. That is actually the novel gene we identified here. Formerly in 2005, ABCA5 gene is reported as the gene that is involved in causing hypertrichosis. But in 2005, Cuba et al. Reported in the mice model that ABCA5 gene is actually causing LSD-like phenotype. So this is the first human case in the world that we reported here causing LSD-like phenotype in our patient. Then IDUA gene. Sixth family also LDUA gene. One of the things that I want to mention here is you can see L490P variant here. This is the prevalent variant we found in mucopolysaccharidosis type 1 patients in Pakistan. One of my papers soon going to be online. You can see out of 10 families, we identified this variant in 8 families. Then IDS, this gene is actually causing Hunter syndrome. LDOP, the 8th family, and then in the 9th family, you can again see multigenic inheritance, and 10th family is also showing multigenic inheritance. 11 and 12, both families are showing multigenic inheritances. So these are the pedigrees of all 12 families showing consanguinity in all of them, you can see. So the reason in spreading autosomal recessive disorders in our population, the main culprit. MPS 2, second category that we included in this paper is mucopolysaccharidosis type 2, that is X-linked, and that is caused by the deficiency of enzyme iduronate 2 sulfatase. Again, this enzyme is involved in the degradation of heparin sulfate and dermatan sulfate. It is caused by mutation in IDS gene. This is also very rare, affecting 1 in out of 100,000 live births in male. It has 2 subtypes, early onset and late onset. So these are the 2 recruited families, initially recruited on the basis of their phenotypes, because we are not having accurate clinical diagnosis here. So the 2 variants that we identified confirmed that, yes, they are actually the Hunter syndrome patients, but both variants are novel. So these are the pedigrees. You can say there is no consanguinity in both. Okay. Yeah, this map is just showing that we recruited family from diverse areas of Pakistan. So this is actually the ancestral analysis that we performed on our families. You can see most of our families here in purple color are overlapping with the ancestral analysis of our South Asian population, except for one, that small purple dot that deviated slightly toward African region. So in present study, we presented 14 families initially diagnosed as MPS, whole genome sequencing to identify potentially pathogenic variant. Among these, 12 families were clinically characterized as MPS 1, while 2 were clinically diagnosed as MPS 2. We identified potential novel candidate gene ABCA5, that is a breach, and other chapter in the monogenic disorders, being complex in highly consanguineous population like Pakistan. There are limited resources in Pakistan for diagnosing and treating complex genetic disorders like LSDs. That's why our recruited cases show diversion in initial clinical diagnosis and genetic diagnosis, especially in MPS 1, where we have seen 7 deviations out of 12. So our study increases the knowledge of genetic factors involved in rare diseases. For this purpose, Pakistani population is unique due to high number of families with consanguineous marriages and high frequency of large pedigrees. Whole genome sequencing is a very, crucial thing now to diagnose such complex disorders. I really appreciate the participation of the families. Thank you.
Thank you so much, Dr. Rutaba, for that wonderful presentation. We'll be taking any questions from the audience if there are any now.
अस्सलाम वालेकुम मेरा मेन क्वेश्चन ये था कि जैसे आपने अपने पेपर के अंदर भी बताया कि MPS टाइप 1 के लिए जो मेन कल्प्रिट था वो कजन मैरिजेस है और पाकिस्तान में इतनी अवेयरनेस भी नहीं है जेनेटिक टेस्टिंग और स्क्रीनिंग वगैरह के लिए भी तो व्हाट स्टेप्स शुड वी टेक अवेयरनेस क्रिएट करने के लिए गवर्नमेंट को अदर देन कोई लॉ इम्पोज करने के लिए क्योंकि वो उनकी मर्जी है कि वो उसको फॉलो करते हैं या नहीं करते तो व्हाट स्टेप्स कैन वी टेक टू क्रिएट अवेयरनेस कजन मैरिजेस से रिलेटेड या फिर जेनेटिक स्क्रीनिंग से रिलेटेड?
अच्छा, इस तरह से है कि अवेयरनेस, क्योंकि मैंने तो जस्ट अपनी रिसर्च स्टडी 30 ओके सर, जस्ट अपनी रिसर्च बेस्ड स्टडी की थी और उस दौरान जितनी फैमिलीज रिक्रूट की हैं मैं ऑन द स्पॉट उनको जितनी गाइडेंस कर सकती थी करती थी। बाकी ये एक नेशनल लेवल प्रोग्राम होना चाहिए फॉर क्रिएटिंग अवेयरनेस।
So next, I would like to call upon stage Dr. Misbah Hanif from the Sindh Institute of Child Health and Neonatology. The floor is all yours.
Assalamu alaikum, everyone. Thank you for being here. My name is Misbah Hanif, and I feel honored to be the student of Pakistani Society of Medical Genetics. So today I'm presenting summary and insight from the first genetic services clinic in public sector pediatric healthcare in Sindh, Pakistan, a joint collaboration. So let's talk about the discoveries and the journey we have made so far to improve the pediatric population of our public sector hospital. Genetic disorders affecting 1 in 2,000 globally have a significant impact on affected families. Despite of having a large population and high consanguinity rate, resources are very limited for the people who deserve genetic services. Obstacles include lack of trained professionals, limited availability of genetic testing. To address these issues, Pakistani Society of Medical Genetics initiated a partnership with National Institute of Child Health in collaboration. This unique initiative, first of its kind, provided free of cost genetic counseling services to the patient from the public sector hospital. Here in the slide, I put the logos from Pakistani Society of Medical Genetics, National Institute of Child Health, and Sindh Institute of Child Health and Neonatology. which reflects and representing a crucial partnership which was highly needed in public sector hospital. These are the objectives of my presentation. First is to explain the our clinic experience and data that we have got in our clinic. And second is to explain the role of the consanguinity and its prevalence in our cohort. In the journey of Pakistani Society of Medical Genetics, they took a crucial partnership and with National Institute of Child Health, and at the same time, they announced a position for the trainee genetic counselor at 2020. A significant achievement was occurred when National Institute of Child Health and PSMG collaborated and signed an MEU with the Invitae Laboratory to provide a subsidized costing for the genetic testing. Moving into the 2022, PSMG contributed in genetic research by presenting a novel case of WFS1 spectrum disorder at ASHG 2022. Transitioning to 2023, Genetic Services has initiated at Sin Institute of Child Health and Neonatology, a public sector hospital developed to provide cutting edge services to our people and pediatric population of Sin. Let's dive into our methodology of telegenetic clinic, which was established at National Institute of Child Health at 2020. Most of the referral were from the National Institute of Child Health and Sindh Institute of Child Health. We provided genetic counseling services for both of the institution and other institute as well. They get tested through the partnership of a whole through the partnership from the institute Exeter UK and Invitae Laboratory. Pakistani Society of Medical Genetics, excuse me, whose phone is this? It's disturbing me. Okay. PSMG focused on the training and educating the next generation to empower them in genetic counseling services. Sorry, I need to mention that our team comprises of the pediatrician and the trainee genetic counselor from National Institute of Child Health and the geneticist and genetic counselor from PSMG. We provided pre-test genetic counseling to 138 patients in our cohort. Post-test genetic counseling was provided after testing results, and it is in this session families receive result disclosure. It is actually provided to address any question or concern they may have. For the research purpose, we use the clinical research form to collect important data like history, socioeconomic status, consanguinity. For that purpose, exit survey was also conducted to get their feedback. This is the demographic data of our cohort. Here we can see significant discrimination, especially in context of consanguinity, which we have been discussing since morning. Here we can see a high number of patients were visited us who had a positive consanguinity. So what does a genetic counselor do in a typical session? We, genetic counselor, gather comprehensive family history and the health history, which can be associated with the genetic diagnosis. By using special nomenclature and symbols, we draw a family tree across 3 to 4 generations. Genetic counselor pay close attention to the information which can be missing or need further verification for correct or proper assessment of the risk in the family. Genetic counselor plays a very crucial role in explaining and help people understand why the test is required, and they explain them the method with all the benefits and the limitations. Oh, sorry. This is the typical genetic session. This is our typical genetic session in which we start. We begin with the warm greeting and gathering the information which make the foundation of our session. Then we move into the educational component of the genetics, in which we provide insight to genetics and inheritance pattern. Then we we calculate and we try to assess the risk of the disease in the family. And we always make sure that the informed consent is taken before explaining the logistic and practicalities. If any testing is offered, then we provide emotional support, and a dedicated question answer session is also provided, and then we conclude our session with follow-up plans. These are our findings, in which we can see that approximately 138 patients were tested, and out of them, approximately 50% had positive family history. And unfortunately, 90% of them were consanguineous or the product of consanguineous marriages. In our cohort, we found a high yield of findings. We got approximately 68% pathogenic variant, including pathogenic and likely pathogenic variant. So, which is a high yield considering our small sample size. This is reflecting our good approach and efficiency of panel genetic testing. In this slide, I tried to emphasize the role of the consanguinity, but I guess it's not needed now. This is the slide from our endocrine clinic. Here we can see a various spectrum of disorders were seen, and the highest number of the patients came with osteogenesis imperfecta and disorder of sexual development. And yes, this is the findings from our metabolic clinic. Here, like Dr. Rutaba, we found most of the patients were diagnosed with MPS. This is our neuroclinic findings. But I guess panel testing was not efficient in neurological disorder as in other domains. Approximately 400 patients with suspected genetic diagnosis. Oh, I'm sorry. Okay. So we we make a registry at Sin Institute of Child Health, and try to register them because we are more frequently seeing as we have seen in our this cohort. So the challenges we faced in our clinic was emotional impact in pathological findings with no treatments, psychological stress, multiple affected children, as we Dr. Kirmani and Fizza has discussed, religious and cultural sensitivity, feeling of being. However, we tried to stick to the non-directive counseling approach. These were the founding of our genetic counseling exit survey. And we found 100% of approximately 100% satisfaction of our patient. Thank you so much. It was very short of time, but I like to thank you, my mentors, especially Dr. Jamal Raza and Professor Mohsina Noor, for giving me this incredible opportunity to learn from PSMG. And I just want to thank Dr. Ajaz for his continuous support and believe in me that shaped me who I am. Thank you so much.
Thank you so much, Dr. Hanif, for that wonderful presentation. We are a bit short of time. We could take one question if there's any. Are there any questions as of now? So no questions. Thank you so much, ma'am. Thank you. So next, we would like to invite on stage Dr. Ameema Asad here from AKU. The floor is all yours, ma'am.
Assalamu alaikum, and thank you to the organizers for choosing our work for an oral presentation. Without any further delays, I would like to start. So I have no disclosures to make. Cardiomyopathies are defined as structural and functional abnormalities of the ventricular myocardium, which remain unexplained by coronary artery disease or abnormal loading conditions. Amongst the primary cardiomyopathies, hypertrophic cardiomyopathy is the most common one, and it is caused by abnormalities in various genes and has an estimated prevalence of 1 in 500 people. The most devastating outcome which is associated with HCM is sudden cardiac deaths, specifically in young individuals. With improvements in healthcare and improved diagnostics, we have over 1,500 pathogenic and likely pathogenic variants being identified in 26 genes which are associated with HCM. These include 9 sarcomeric and non-sarcomeric genes. So sarcomeric genes are essentially the ones which encode the muscle protein of the heart, the myocardium. Genetic testing yields important diagnostic and prognostic information, but variants of uncertain significance make the interpretations of results difficult, as we have discussed earlier also. But most of the genetic data comes from the individuals of European descent. In our work, we report the results of the genetic analysis of Pakistani patients with HCM. So our methodology was a mixture of retrospective and prospective recruitment of patients. For our retrospective arm, we reviewed patient records who were tested for next generation sequencing based targeted panel at Invitae Genetics in the USA. This covered a study period from June 2019 to May 2023, and these were a total of 19 patients. For our prospective recruitment, we recruited patients who had cardiac imaging consistent with HCM at our institution. And these patients underwent whole exome sequencing at 3 billion in South Korea. This covered a 2-year study period from February 2021 to February 2023, and these were 48 patients. So combining both the retrospective and the prospective patients, we had a total of 67 subjects. Of these, 17 were females, 50 were males, and 10 were children under the age of 18. All of these patients had an echocardiogram and or a cardiac MRI, which was consistent with morphological features consistent with HCM. So when the results of genetic testing arrived and we compiled our findings, so we found out that basically the bigger pie chart that we see here, it was 25 patients who had positive findings, which positive results, which was that they had 1 or more pathogenic or likely pathogenic variants. And these were around 37%. Then 33% were negative results, while 20 over 67, almost a 30% patients had VUSs. So now moving on, what we did was that we used some genomic tools and did reverse phenotyping, which was basically the correlation of phenotypic and genotypic findings, and we resolved these VUSs. So this pie chart basically shows that 6 out of 20, almost a 9% were reclassified as likely pathogenic, while 14, almost a 21% remained VUS. So moving on, this slide is basically a summary of what we discussed previously. As we can see that this 37% here was our initial diagnostic yield. but after resolution, it increased to 46%, which is consistent with previously published literature also in other populations. So the spectrum of genes which was identified in our positive patients was around 10 genes. So the most common one that we can see here, the biggest chunk is with the MYBPC3, which was found in almost 43% of our positive patients. The second most common was MYH7, which was found in almost 15%. Then we had TNNI3 as the third most common gene. The other common genes were TNNT2, GAA, GLA, MIB1, ALPK3, and SCN5A. Now to elaborate our results further, from our total patients which were positive, these were 31 patients. 8 of these, 26% had biallelic variants, meaning that these patients had autosomal recessive disease. Now 5 out of 8 of these were pediatric patients. So when we further divide our positive patients based on their age, so from the 6 pediatric patients who had positive results, 5 of these had autosomal recessive disease, which was 83%. This also included 2 neonatal deaths because of severe neonatal cardiomyopathy. From the 25 adult patients who had positive results, just 12% had autosomal recessive disease, which is around 3/25. So this is the first ever study, which is from Pakistan, which uses NGS targeted panel and WES to study deeply phenotyped patients with HCM. By our findings of the study, we can also conclude that the diagnostic yield which we achieved and the most common gene, which was MYBPC3, which was found, is consistent with other populations. Severe autosomal recessive MYBPC3 related cardiomyopathy was seen because of high rates of consanguinity. And definitely, it is crucial that an underrepresented population like ours needs to have access to genetic testing for tailored management of patients. And appropriate genetic counseling is definitely needed to prevent the occurrence in future generations of inherited hypertrophic cardiomyopathy. It is crucial as novel targeted therapies are being developed, which are variant specific. So this was the list of references that we had used. To finish off my presentation, I would like to thank my team members, Dr. Salman Kirmani, Ms. Fizza Akbar, Dr. Asra Wahid, Dr. Yavar Syed, Dr. Fateh Ali Tipu, and Mr. Arif Mehboob Ali, and of course, our patients and their family members for their support in making this project a success. Thank you. Do we have questions?
Yes, so thank you so much. Do we have any questions before we move on to the panel discussion?
Thank you for the wonderful talk. My question is about the autosomal recessive cardiomyopathies. You mentioned that there was a very severe phenotype. Did you see that consistently across the board for all the genes, or it was just for the MYBPC3 gene?
Just for the MYBPC3 in 2 of our pediatric patients.
Sorry, I've got a question. You're mostly adult patients, but negative for the any genetic variation. That is true. And they were mostly viral, or what was the cause?
So from our total 10 of the pediatric patients, we had 6 which were positive. 60% was the positivity rate. And then the leftover, which was around 57, we had adult patients. So 25 were positive in them.
And rest were negative.
Yes,
For any
yes.
genetic mutation.
Yes, for any genetic mutation.
Any more questions from anyone? I think that is about it. Thank you so much, ma'am, for your presentation. So without any further ado, I would just now like to call upon stage Dr. Salman Kirmani to initiate the panel discussion.
Thank you very much, everyone. And thanks to all the oral presenters, really appreciate it. And a special thanks to Dr. Rutaba Gul, who made her presentation and did it at the last minute. So thank you very much for that. I'm just very excited to be hosting this panel discussion with our PSMG colleagues. And we've got a really, really esteemed panel who, despite the rain, despite all the challenges, despite many other commitments that they had today, I know, in fact, there were multiple commitments even today, they've all come because they're very committed to furthering the cause of genetics in Pakistan. I think each one individually has a lot to contribute. So I'm going to just briefly introduce them as they come up, and then we'll sit and have a discussion. And then, this is again going to be an interactive discussion with our panel, and we'll encourage questions from the audience as well. So first, I'd like to invite Professor Syed Qureshi to come and join us. So Professor Syed Qureshi needs no introduction. He's the Vice Chancellor of Dow University of Health Sciences. That is not only my alma mater, but an alma mater for many people out here, and many people have served the university before joining us. So thank you so much, Professor Syed, for being with us today. Next, I'd like to invite Professor Tazeen Ali, who is our Dean at the School of Nursing and Midwifery. And she's been a fantastic colleague and a fantastic advocate for genetics in nursing. In fact, we're very proud to say that we have Pakistan's first genetic nurse in Zohra, who is sitting with us. Zohra Hassan has been serving in the capacity of a perinatal genetics nurse with us for almost a year. And we hope to train her further and go beyond. And she's connected with the global network of nurses. So a very, very important discussion from your end to do. Next, I'll call Professor Shahid Shamim, who is our Director of Graduate Studies. Thank you so much, Dr. Shahid, for being here. And Professor Shahid is, oversees all of our graduate programs, and we would like really his perspective on how can we strengthen the graduate studies and so forth. And then we have Dr. Khalid Ashrafi, who's actually ended up on the other side of the campus. So he is being escorted over here. He is our representative from the CPSP. And he's going to talk about the role of the CPSP in advocating for genetics training for our physicians and dentists. So I'm going to begin, and we have given you a little bit of perspective. We had a wonderful conference, and our panel here is going to round off the discussion. And we talked a lot about the different capacity building that's needed in different areas. And that capacity building was not only in specialized centers, in labs and in clinics, but also in terms of raising awareness about consanguinity and about the public health perspective on how we can prevent these diseases. So I'll begin with you, Professor Qureshi, in terms of the undergraduate curriculum. One question that keeps coming up is that we know we have so many medical colleges all over Pakistan, and the 5-year course is, traditionally based in 2 years preclinical, 3 years clinical, although a lot of that is now being revised, and, clinical teaching is being introduced earlier and earlier. But how do we introduce, Dr. Khalid Ashrafi, thank you so much for coming, and thank you for being here. So I just did the introduction for you. So Dr. Khalid Ashrafi is representing our CPSP representative, and we must congratulate him. His daughter got married yesterday, and despite having family commitments, he is still here. So thank you very much, Dr. Khalid. Professor Syed Qureshi, how do we better integrate genetics into that medical college curriculum, those 5 very precious years after which our doctors will fly off into the world and do good for our country? How do we do that better than what is happening today?
I think there are various aspects to that. First of all, the PMDC needs to be on board because they will regulate, although in isolation, various medical colleges may start off adding a lot of genetics into their curriculum until and unless the PMDC issues a diktat of what is to be done for the smaller medical colleges, and especially what we call the private medical colleges. This is not going to happen. Secondly, you need to have more people. Faculty is not available to teach genetics in Pakistan, and that needs to be increased. The numbers need to be increased. And for that, possibly initially a master's program in genetics needs to be started. But again, where is the faculty going to come from who will be teaching that course? At the moment, as far as the Dow curriculum is concerned, genetics is taught by either biochemists or pathologists. And we have a a week long module or a week long part of the module in the second year. And then in the spiral, we take another week in the fourth year. But again, all of this, even in the fourth year, it is taught by basic scientists, not by clinicians. So this aspect needs to be improved drastically. And until and unless, possibly you will have, until and unless you have a pure genetics department, it's unlikely to happen because people from biochemistry and pathology are not going to pay that much attention. They have not been trained to do it. They are not going to pay that much attention to teaching clinical, especially theoretical genetics is anybody can possibly teach it. But clinical genetics is going to be a problem.
Great. Thank you so much for that very insightful perspective, Professor Qureshi, because I think that's something that all of us have seen play out. And I think it's not just a problem here. Even in developed countries, there is a shortage of clinical geneticists. And there's a way that we must, even our basic scientists who are teaching in medical colleges have to be connected with the clinical, issues at hand. And you raised a very, very good point. And I think you pointed out the role of PMDC within that. I think as we look at these curricula, it's very, very important to see what other countries who are developing these capacities, maybe, how's Malaysia doing it? How are other countries who are closer to our context? How are they evolving? Because, those are the kind of models that we would need to follow. Thank you so much for that perspective. I'd like to then move on and ask.
Can I
Gigi,
add
please.
on to what
Of course.
Professor Qureshi said? I think another angle that I would like to add on to it, whatever he said was absolutely true. But one thing is putting something into the curriculum, whether PMDC says about it or the university decides or the basic scientists decide that, no, we need to put this in curriculum. Anything that students do not relate with to their clinical years, they don't understand it or learn it as a subject or as a topic which they could retain, right? And we have all been through the undergraduate years. For example, one of the things is bioethics. It is in the curriculum, there are classes going on, but because nobody talks about it in the clinical years, students just forget about it. Or the senior ones tell the junior ones, oh, just go through it and pass the exam for it, and that's all you need to do. We had, I graduated in 1994. Okay, we had one chapter in pathology book of genetics, which everybody left. Nobody read it. The only words I understood in this whole presentation was probably none. Okay, I don't know what it means, chromosomes and chromosomal things and all that thing. But yes, that was when I graduated, and the way I was taught. Now, because it's a necessity, genetics is such an important subject that every doctor should have some awareness of it. But the students will only take it with them if the clinical faculty has awareness of it. Most of the clinical faculty we have right now in more than 150 medical and dental colleges in the country are product of an era where genetics was not taught. So I think with all those things which Dr. Qureshi said, Professor Qureshi said, I think we also need to make the clinical faculty aware of how genetics can play a role in their practice, so that they can give that to the student, and students coming from the basic sciences, knowing about something about genetics, can take it forward to the clinical sciences and understand and remember it, I think.
Right. Now, that's a very good point. And I think this is the right time, because if you were to attend a neurology clinic, for example, a lot of the patients have genetic disorders. So the student will immediately relate the importance of that and why. So, and I keep stressing to people, genetics is not just a pediatric thing anymore. There are more and more adult onset disorders, more and more adult onset cancers, and we need to, sort of move because, even taking a family history, pediatricians are probably the best at drawing those circles and squares, but the adult physicians and the adult care providers need to do that too. So thank you very much for that perspective. I think the other thing that Dr. Qureshi very insightfully said was establishing a department of medical genetics. That's where, all of the major universities have a department of genetics, but to actually label it medical genetics connects the genetics professionals automatically to patient care. And I think that's the step. So I'm going to bring that back to my dean and make sure that we do that. Thank you very much for that. That's great. So since we're talking about medical education, I'm going to stick with that for now. And Dr. Khalid Ashrafi, I'm going to ask you, what is CPSP's role in not regulating, but ensuring that all the residency programs that CPSP oversees has appropriate genetics content?
First of all, thank you very much for joining me here in this forum. Medical genetics is blueprint of life. And I think that we should give awareness about it at all levels before we start including it in the curricula, so that everybody should take it very palatable, asani ke saath. At all levels means, I think I agree with my friend, right from medical students to our basic practitioners to our specialists. And then we will be in a position to develop faculty, and that faculty would decide at what level should we introduce it. Now, this can be started in a hospital, teaching hospital, but College of Physician Surgeons Pakistan has always taken initiative for all the innovations. Let me tell you that when this college came into being, since its inception, only 5 disciplines that we were having for fellowship program and some for the membership. And now we have 82 programs, more than 82, and some are still in the pipeline. This can be started as a membership program for 2 years, for a full fellowship, or as a second fellowship. That's what the faculty would recommend us. At what stage, at what level, what you want from us. We as College of Physician Surgeons Pakistan full-heartedly support this in Pakistan for our education, for promotion, because this is what is needed. This is what is needed. Ye future bhi hai, present bhi hai. This would help us in many ways. And this is the awareness that we have to give. It's not going to replace the traditional medicine. कुछ लोग, I suppose they would feel frightened कि probably ये कोई हमारी fellowship के साथ कुछ और चीजें नहीं. I think it is going to help in diagnosis, early detection and treatment. So इसके लिए awareness का पहले बहुत सारा एहतमाम करें, programs करें. Let people feel that this is the time that we have to know about it. We have to implement it in our curricula and then do it. So college will definitely help in developing faculties, in organizing workshops, in fellowship programs or a second fellowship, whatever the faculty would give us, we will wholeheartedly support those faculties. But before forming faculties, you have to give awareness on a larger scale to develop all this. Thank you so much.
Thank you so much, Dr. Ashrafi. So here's a dilemma that we're facing as we're planning this, right? So if you take the model of what happens in the United States, medical genetics is a standalone residency. It has a requirement that you have to do at least 2 years of training. Most people finish some primary training, either pediatrics or family medicine or neurology or obstetrics gynecology, and then they do a standalone training in genetics, which includes both pediatric and adult experience. And here, as we see traditionally, as I mentioned, that most people in pediatrics are more familiar with genetics compared to other specialties. And it's very tempting to say, let's add a second fellowship to pediatrics and let's make genetics a second fellowship to your primary FCPS in pediatrics. But that would basically, you would have to train that pediatrician on a number of adult oriented things. And would they actually come back after that and practice on adults? Or do you start from scratch and say, no, we'll follow that model of you start from scratch and you teach people both adult and pediatric medicine, give them a hybrid residency where they get exposure, even a little bit of OBGYN, and then, start the genetics curriculum. What do you think is the best model there?
I think this should be decided by the faculties, faculty of internal medicine, faculty of pediatrics, pathologists. They should sit together. They should decide at what level we should introduce this medical genetics in our program. And at a 2-year program or a primary fellowship, this is something that needs a lot of discussion and deliberations. And as a result, whatever the conclusion would be derived from those, I assure you, we will take it to our faculties. We will take this message from here to our faculties. I would write it to the deans of internal medicine, pathology, pediatrics, gynecology, that let's sit together and have a discussion at what level and how should we introduce it, and what is the best way that we can educate our new fellows or fresh graduates that they are inducted in the program, and how we can improve all about it. So this cannot be decided in just a meeting
Sure.
of few minutes. That needs
Yeah.
some time and that needs discussions. And they will look after each and every corner. It's a multifaceted. So we have to look after it. Thank you.
Thank you, Dr. Ashrafi. I think Dr. Shahid Shamim has something to add.
Yes, I'm sorry, I cannot stop myself from intervening here because as an educationist, I would suggest, I was thinking of what experience do we already have which can reflect on how genetics can be brought into the mainstream postgraduate medical education. One example is of emergency medicine. Now, for something which does not have an infrastructure of departments at present in many institutes, you would not be able to provide a career ladder to a pure geneticist, somebody who does a MBBS and goes into medical genetics and does a fellowship of medical genetics would not have a place to go for a job.
Well, we hope they will, but yeah, that's a good point that a lot of places may not yet.
And then to establish departments, you need experts, you need specialists. So it's kind of a chicken and the egg thing. Like before you have a department, you need a specialist, but because you don't have department, you cannot make a specialist, right? So the process for that, from an educationist point of view, is you start off with as a second fellowship, right? So people who are already fellows in particular, for example, in adult or pediatric departments, can do a fellowship of medical genetics. And in that fellowship, they probably would go through whole of the curriculum with their more focus on their own specialty from where they're coming. So within a very limited time, you can have a group of physicians who are genetics trained, who are already in place as faculties in some departments, and you can just gather them together in one place and start a medical genetics department. And then move on to to having a primary fellowship in. So I think that could work out for, and as Dr. Saab said, that definitely it's just coming up from above our heads. It needs a lot of discussion and deliberation and how things would process, and the specialists, genetics specialists need to sit, the educationists need to sit, the CPSP people need to sit with it and decide how it should be taken. But,
thank you very much. And I think that alternative model is very interesting. I think Dr. Rizwan has a comment. If someone can hand him the mic. Oh, you have the mic. Go ahead.
So thank you so much for a very lively discussion. So one thing I wanted to, I'm a laboratory geneticist, so I wanted to emphasize that many of the institute, AFIP, Dow, Khan, they all have sequencers now and sequencing machine, but the productivity of the test results are not there. They are somehow lacking the expertise in interpretation part of laboratory test. So when you'll discuss medical genetics, please don't forget laboratory genetics, because they are the one who will provide you results. Before any results, you will not be able to help manage that patient. And so what we do in the United States, for example, after pathology residency, there is a second fellowship or first fellowship. You do MGP, molecular genetics pathology. And that fellowship trained that pathologist who already know what the labs run and how the labs are done. He become specialized in genetic testing also. Similar thing we do for OBGYN or pediatricians or PhDs in medical genetics. We do 2 years of fellowship, which is called LGG, Lab Genetics and Genomics, which I did also. At that time, it was different. But that lab genetics and genomics are the clinicians and PhD trained people who interpret test results in genetics. And those are, I don't think there is any one in Pakistan at present on those, all of these specialties. Yes, medical genetics is good, but medical genetics, I think I agree with what others have said, that it should not be a primary training program, at least at present, but it should be a secondary program for pediatricians or for internal medicine. Thank you.
I think you are very right. This discussion of medical genetics would not be complete without involvement of a pathologist. That's what I said, that we need to have discussion between physicians, gynecologists, pediatricians, and pathologists. And pathologists have a pivotal role in this medical genetics. And what we think that without your, I think, sorry, decisions and whatever, it's very important. And we need to have that. When we will start this whole process, we would invite pathologists, the deans of various pathologies, histopathology and molecular and whatever, along with physicians, along with pediatricians, along with gynecologists. And I think this is a long way to go. It will take some time. It's not a matter of few minutes or hours or days that we will decide at what level and how we are going to start it. But definitely, this is the time that we should think about it. This is the time when you should start working about it. And this is the time that we should be with the rest of the world, because as you said, we don't have a department even in Pakistan. How we are going to start this whole process, this business here, we have to think about it.
Thank you very much. I think there's one more question from the audience and we'll come back to the panel. So Professor Nick Brown is here with us. He's a visiting faculty member at AKU, practices in the UK and now in Sweden. Yes, Nick.
Yeah, just a small observation. Interesting listening to the ideas that have been raised so far, none of which are mutually exclusive. So I want to add one more spoke to this as a potential. So it's kind of a philosophical one that I think part of the reason that genetics has maybe taken off more slowly over the last 40 years or so than some other subspecialties is because it's been treated as almost like a separate entity on its own. And I think one way around that would be to make everyone a geneticist, starting from, I'm being serious, starting from medical students upwards, that any lecture should, and this will obviously take some time to indoctrinate, should include a comment as to what are the genetic implications, why have they got this disease, right from the very first year, through later training to postgrad training. And that will take some time to permeate. Just reflecting on something that happened to us as medical students in Leicester. This was in the mid 80s. We had this guy called Alec Jeffries, who was a lecturer for us. He was a guy who was on the verge of discovering DNA fingerprinting. And he was so relaxed and cool, and he'd come in and say, yeah, I just had an interesting discussion with some colleagues in the US. And literally, this was over that time period, he made the discovery, which essentially changed crime investigation, and was so modest about it. And
you're right.
It was there in front of us, and we didn't seize the opportunity.
Yeah. No, thank you very much for bringing up that point. In fact, Johns Hopkins about 5 or 10 years ago named their medical school curriculum From Genes to Society, because they wanted every single medical student graduating from their program to know that genes relate to society. So I think that that's a very, very illuminating point. Thank you for that.
Yes, that is so correct. And I'll reiterate it, which we started off with the undergrad, the importance of teaching genetics in the undergraduate is absolutely phenomenal. You cannot start off a postgraduate program without putting it into undergrad curriculum, and not just in biochemistry or pathology, also in the clinical side. And for that, we need to make the clinical physicians aware of the importance of genetics in their practice, so that they can do that with the students.
Great. Thank you very much. So Professor Tazeen, I'd like to ask your perspective. So, this is the age now where we've realized, unfortunately in Pakistan quite late, that we really haven't empowered our nurses, and we've really not used them to their full potential. So your perspective on, the nursing curriculum, and how we can have a genetics nursing workforce.
So thank you, Salman. Being the only female panelist, I'm going to take a privilege to talk about 4 things here.
Okay.
The first thing is I really wanted to congratulate you for having this seminar, this conference. And I am really surprised to see that today was such a beautiful weather, and yet you guys are sitting inside and attending this. So I can understand how important it is. And now, exactly, I do buy your point that yes, now it looks really beautiful over here because we are discussing something where the entire world is moving. So this is very important. Second, I just wanted to share that what Dr. Syed was trying to say, that we do have some curriculum in PMDC, but still we need to dig out how it should be taught and how it should be monitored, what Dr. Shahid is trying to say. I was about to say him Shahid. His teachers used to say that, right? In Australia. So I was about to say that. I said, no, I need to stop here. So we have a pretty similar situation. We do have in our curriculum, our first year undergrad students and second year do study genetics in their pathophysiological courses. And our master students, we are having, Aga Khan University is the only university at moment at Pakistan who is having Masters of Nursing program, having 2 different tracks. One is a clinical track and another one is an education track. So those students who are getting done their clinical track are the ones learning about genetics in relation to specific diseases like diabetes, mellitus, asthma, different allergies, and also congenital abnormalities. And they are also having some kind of exposure with the pediatric department. They do that. But again, what Dr. Syed was trying to say, we do have curriculum available. Pakistan Nursing Council is really putting it as a blueprint. However, we do not have very good tutors who could really teach it. And then you have to invite the medical colleagues. By the way, medical colleagues and nursing colleagues are genetically together.
Yes.
So, so we need to work together to be able to do that. So then we do have, but we have to invite medical colleagues to be able to teach. But it's important that we should have people within the nursing community who should have advanced education within the area of genetics. Like, I'm really glad to see that Zora, you are already a part of it. Probably you can become our first advanced practice nurse who could really practice genetics in the future and teach and come and join us to School of Nursing and try to teach over there to our students. It's important. Now, there are many areas I was just thinking about in the area of practice where we can really practice it. I think already a lot of nurses who are conducting the clinic with their diabetologist, and they are trying to give counseling and everything to the people around. But I think it's important that whoever nurses who are specialized and dealing with the genital diseases or any diseases which you really cannot get rid of it, nurses should get the kind of education on it, so then we can have integrated system. So it's not only specialized field, but field within the integration of current pathology and different diseases and all. It's very much important. Aga Khan University is trying to think about initiating of advanced practice nurses. It's already there in Sweden. It's already there in Australia and America. But in Pakistan, we really would like to initiate that. India and Bangladesh have already initiated it. Pakistan need to start that, and we would like to start with the acute nursing care. Second, we are thinking about public health, and I think third should be genetics.
Genetics should
Advanced-
always be first.
Exactly.
Yeah.
So that has already been started, but let's also take
Okay.
this up as a,
Great.
and I 100% agree to you. Why I 100% agree to you that all my studies, I'm also an epidemiologist and statistician. In my all the studies, I have, been calculating relative risk or the attributable risk. So what do we do always? Modifiable factors and non-modifiable factors, isn't it? We always say that smoking is attributable, contributing 30% of the risk, but genetics contributing 30% of the risk, and sometimes we call it unknown. So we are really, kind of novice in calling it, rather than genetic factors, we call it, unknown factors, which is not at all appropriate. So it's important to understand what are those unknown factors and why do we have to call it non-modifiable factors? We should be calling it modifiable factors. Today, we could really run different kind of researches, genetic researches, to be able to change the sequence of it. Why can't we do that? We can do that today. So then I really do not want to call it, as a researcher and as a nurse, that I do not want to call it that they are not modifiable factors. They are modifiable factors. And thank you very much for inviting. And we really have to work more and more as nurses, rather than two only nurses sitting within the entire audience. It's important that we should sit together hand in hand to be able to work to get rid of all those diseases which are really giving us hard time.
Wonderful. Thank you so much, Professor Tazeen. I think Dr. Ajaz has a question from the audience.
Assalamu alaikum. I'm Dr. Ajaz, I'm a clinical geneticist. Shahid and I graduated together in '94. And like he said, we left the chapter of, genetics, and now here I am, a clinical geneticist. I think the important issue is all on the stage and all in the audience feel that it's important that we need to bring genetics to Pakistan. We will talk about it today. We will say, yes, we need to do it. And tomorrow we'll go and do our stuff. The important question is, it has to be a multifaceted approach. It cannot be just a fellowship, a residency, or a nursing, or medical student. So we can see it. We need to do it at all levels rather than just one approach. So the big question would be, is there either a provincial or a federal or a CPSP body, a working group that could incorporate geneticists and other specialties that would come up with a genetics policy moving forward in the next 5-10 years, have markers as to how we are going to implement this? And that is my question. And Dr. Jamal Raza is here. I would like to hear from him as well as to who would be the body, or which body can we formulate that would make it go forward.
Thank you, Ajaz. I think that's a very important and pertinent question, that what do we do in real life to improve the situation? And, as you said, we'll talk about it and then move on with our lives until we have a solid plan. I think the role of public sector and the governments is very important in this. Dr. Syed is sitting here, and really he is at a helm of the affairs, like heading a large medical university. And I think the easiest, sorry, Dr. Syed, putting you on the spot, but I think the easiest, the easiest way forward would be for a university to take a major step. And since you are in the public sector, you can talk to the government and establish not just a department of medical genetics, but also a department of genetic testing. You see, one of the reasons that genetics was not addressed, because we couldn't make 2 and 2 of it. We did not have genetic testing available for our patients. So there was no genetic diagnosis. Since we have addressed the issue of genetic testing, whether, we are sending it away to INVITE or कहीं भी हम भेज रहे हैं, अब हमारे patients जब reports लेके आ रहे हैं genetic testing, तो by force now all of us as clinicians are forced to learn, to talk, to address the issue of medical genetics. So I think this goes hand in hand. On one hand, Department of Education that will create the, Dr. Syed ने जैसे कहा कि जी teachers ही नहीं है, तो कहीं से आपको teachers पहले तो बाहर से शायद लाने पड़ेंगे, और फिर locally produce करने पड़ेंगे so that they can teach the subject of medical genetics. And also we need a center which can do the testing in Pakistan. We cannot rely on test, sending to USA and Germany and all that. So kahin na kahin, one center of excellence which can do testing, maybe at a public sector, at a cheaper rate. So once you start doing genetic testing more and more for your patients, that will on one hand improve the genetic information for the physician, for the people in general, and on the other hand will produce and improve the department of medical genetics and create more teachers.
As far as this point is concerned, our university now has a center which has been brought about by the funding from the Higher Education Commission and also fairly substantial funding from alumni in the United States. So some of the money has been spent, the US money has been spent, some of it is still available, and some I mentioned, I think, to you that we are still progressing on that front. We have started doing some of the testing, but this is not being reported here. This is being reported again by one of our, or will be reported by one of our alumni in the United States. So we are taking it from there and we will go on and see how things evolve. I can't say that we are going to be a success story, but at least we are trying.
Thank you so much, Professor Rashid. Could you share a little, can I ask him this? You had this vision of starting, this is something that's taken probably the last 5 to 7 years to establish, right?
Yes, 5 years.
Can you just maybe,
COVID messed it up.
yeah, and then this is during COVID also. So could you maybe just give us a 2-minute summary of how did you prioritize? What was the capacity building effort? And what is the output you're expecting?
I will start with the last. The output, I think, would be huge because the need is huge. So obviously the output is going to be there. How did we start off? I was offered some money by the higher education. I had it in my mind. I was offered by the head of development at the HEC that some money had been left over from a project and this could be made available if I could do it quickly. So we did it quickly. We had already worked on setting up a genetic center. I had visited Chicago actually to see how to go about it. And from there, we have procured the equipment. Dr. Rizwan is going to visit us tomorrow and then he's going to possibly point out some of the shortcomings that there may be. From there, we have started testing mainly of cancer material. I'm not sure what is the exact cancer material, but I was told by Asif Qureshi, who is heading the unit at the moment, that 40 samples have been pushed through. So let's wait. After these 40 samples have been analyzed and have been reported on properly, then we will be able to say what is going to be. We are in the process of getting another software which will help us in the reporting. And as work increases, Dr. Rizwan last night mentioned to me that we'll probably have to go get a robot, which does a lot of the preparation work. So we are getting there slowly. But at the moment, medical genetics, we don't have anybody. And
yeah, and we have talked
although
about
I would like to point out that again, one of our alumni who deals with IRD, inherited retinal diseases, and there is a lot of inherited retinal disease, especially in Sindh province because of consanguineous marriages. And we have started work on that. And that also is in an evolving phase.
Great. Thank you very much.
Just to add on to that, I have worked with people, Asif Qureshi and his predecessors in the Dow University Genetics Lab, and it's a fantastic setup with a lot of equipment which I don't understand, but it looks very nice, and they are doing a lot of good work, especially from on the research side of it. And if I'm not mistaken, there are some of these labs in Karachi University and University in Islamabad, which are research labs, but are not connected with the clinicians or medical laboratory people, pathologists. And maybe that can be another way of going about, where if these can be connected with the healthcare people, maybe something can come up.
So that was exactly my question for you, is how do we make that connection? Because these are all, graduate schools, right? They're graduating PhDs, MPhils, master students, and then they continue to work in the lab side without access to actually the medical side of things. And then sometimes there's a sample load that comes in from some collaboration. The collaborations are very sporadic, and it's never really done in the realm of patient care. The samples that come in, the patient may never even hear the result back. So how do you connect those two worlds?
So these are people who are coming from nursing or biotechnology or medical or dental backgrounds and going into PhDs into genetic research through genetic research. I mean, they need to be roped into medical education, undergraduate and postgraduate. However, as I said earlier, we need to have a career ladder for them within the medical institutes. As you said, there should be departments or at least some place where they can they can launch their themselves and start working from there. And they can be a very good resource for teaching undergraduate and postgraduate students.
And then I also would like to highlight over a role of a nurse as a public health nurse, specialist.
Right.
So public health nurses could also play this role. It's not only picking up the report and telling the community, but it's important that someone need to advocate them. Someone need to guide them how it should be done, that cousins and cousin marriages shouldn't be taking place and all. And other than that, I was also thinking that when we talked about genetic testing, I think nurses play a big role over here in consent taking. It's important to have a consent and telling them how the test will be done and what could be the possibility later on after the test and all. So I do buy what he said, that it's important that nurses, doctors, basic sciences, everyone need to work together to be able to have the outcome.
Thank you.
I
Yes,
think
Dr.
there was a time when we did not have any department of medical education in Pakistan. Then in the various universities, I remember Dhaka University has taken a lead in medical education also. College of Physicians Surgeons Pakistan, they developed Department of Medical Education, then Directorate of Medical Education, started with diploma, then membership, and now we are moving on to fellowship. Then advanced skills labs. Initially, we had basic life support workshops and basic surgical skills. Then we moved on to ATLS. Then we moved on to ACLS, PALS, whatever, in all the for gynecologists, for pediatricians, for surgeons, for physicians, for cardiologists. Now, the first thing is awareness. पहली बात लोगों में एहसास दिलाया कि इसकी ज़रूरत है, and then it came up. I think today is a great day when we are doing this great job of our awareness. Medical genetics is blueprint of life. We have to work on it, and inshallah, very soon we will see that we have departments in various universities, because university has the main role in research. Universities do research and give us guidelines. College of Physician Surgeons perhaps is more in postgraduate teaching and training. Guidelines, they come from universities. And I'm sure with the presence of this great man, Professor Syed, we would be having a research lab or a department in Dow University very soon, which he is already involved in. He is a visionary person, and I'm sure he will do it, and others would follow him. So very soon I see a good future of medical genetics in Pakistan, and inshallah we'll achieve it. Thank you.
Thank you very much. So I think we are, going to end. There's one last question.
Thank you.
Go ahead.
I'm Dr. Kulsum. I'm professor of biochemistry from Bahria University. We have been teaching genetics in biochemistry and partially in pathology. But the thing is, when it comes to the lab work and then interpretation, as sir was saying, that is lacking for the medical college. Okay. So I think there should be a change in curriculum, as sir was saying, and along with that, offering some postgraduate programs. And along with that, the government should take the lead in awareness as well as the, screening programs, so that it should be, all together. So it should be the labs and interpretation should be available for the developing doctors.
Thank you very much. I think Zora has her hand up, so I'm going to give you the final word, Zora. Yes.
I am involved in a group internationally. I have a privilege as a reference group member for a global nursing network for rare diseases. And they're working on the curriculum, where they're working on the modules, they're working on the techniques to introduce things into the curriculum, because all over the world, throughout the nursing curriculum, they don't have such modules that can introduce or make nurses more, have their hands-on to the genetic knowledge or practice.
Yeah, thanks for bringing that point. So I think Dr. Jamal, yes.
I think, as pointed out by Dr. Shameem, there are a number of other labs across the country in Faisalabad, in Islamabad, Lahore, which are doing genetic works. A lot of them are associated with the agricultural side and not the clinical side. So I would request Salman, you to be the champion and try and rope them in together on the clinical side, because we need one guy who can pull all this together. And I think you at the helm of the fair, being at the AKU, can take that lead.
It's never going to be one guy. And I'm going to say it's never going to be one. It's going to be, and it's never going to be a guy. It's usually going to be a woman who does the work. So I think, on both counts, I say that. But anyway, I'd like to thank our esteemed panel. Thank you so much for adding to this very rich discussion. And you've given us some action points to take over. I think Dr. Jamal has put the pressure, not only just me, but I think everyone who participated today, because I think it's a collective responsibility. Each in their own circle, in their own way, must have that positive influence to go forward. And I really appreciated Dr. Ashrafi's very optimistic ending to the panel discussion that, it will happen. So hum dekhenge, inshallah. So with that, I'd just like, before people get up, I've gotten the toughest part of the day, which is to thank people and to, give a vote of thanks. And I will forget someone and I will be very upset about it. So the best way to do it is to not name anyone and just say thank you. So first of all, I think PSMG deserves a huge hand of applause and credit. Now I'm formally associated with PSMG, but this is not about me. This is really a group of individuals who, had the expertise, had the desire to contribute, made the right local connections, remained sincere throughout the process, remained patient, and, magic happens when good people get together. So thank you to everyone. I think we have Ajaz over here, Dr. Rizwan, Dr. Jamal, Myla, and Aisha have been online. Dr. Saqib, unfortunately, could not join us. Really thankful to PSMG. And I think all of our other partner institutions who took part, SICHN, NICH, and Noor University. I think the AKU team that pulled this off together really deserves a big hand of applause. Really amazing work. And despite all the challenges and everything, so Arif and Khalida here, who are my right and my right hand. So both of them really made it work. And the students from PFL who, emceed so well and organized things. Our IT team, our conference secretariat, who has been working relentlessly just to get all the online stuff going and everything, really fantastic. Our industry sponsors, who were fantastic from the get-go. From the moment I talked to them about the conference, they said, yes, we will support it. It's going to happen. So Roche Pharmaceuticals really deserve a big hand of applause because they're standing up for rare diseases, and again, their drug, whatever they're doing with their rare diseases program here, is never going to make money, but they're doing it because it's the right thing to do. So they really deserve appreciation. And I think you guys are a model for other pharmaceutical companies to do something similar. So thank you very much for all your support. And again, the rest of our team, I'm going to forget someone, so please, I apologize if I'm forgetting you. But you mean a lot. The participants, you guys have been fantastic. I mean, truly remarkable from the beginning. You've been engaged, amazing questions, excellent all the way through. Our online participants, I apologize to them that we haven't been able to read their comments, but I'm going to archive them and we'll share them. And there is going to be a video that has been recorded from the morning. And every registrant will get a link to the video once it's finalized. It's going to take about 5 or 7 days, but you will have that. And, you know, this is not patented. This is not, this is to spread the love of genetics far and wide. So please go ahead and use it for your students, for yourselves, for your education. Thank you very much, everyone. Thanks for a wonderful, wonderful day. Thanks. Yeah. So can we have a group photograph?
We have a group photograph, so I would request everyone to please come in the center. Yeah, just come in front. People who are sitting behind, please come at the front and together in the center of this era. While we settle down, there's one announcement. Hopefully you all have received a feedback link from me. So once you fill out that feedback form, then your request will be catered for the CME certificate. And in a week time, our certificate team will going to contact you about the process to get the certificate. So do fill the feedback in a week time, okay? At the earliest. Thank you.
Please take a seat. We're going to take a picture right here. Please come in the middle so everyone comes. And a special thank you to Dr. Usman Rashid, who traveled specifically from Lahore. He's traveling to Europe tomorrow, but he adjusted his schedule so that he could attend the conference. He's been sitting from the beginning and contributed really, really well. So thank you so much, Dr. Usman. We've learned a tremendous amount from you, and thank you for being here for our conference.
Yeah, please come in between center. थर्ड रो से आप लोग सेकंड में आ जाए प्लीज यस हाँ Thank you, everyone. Thank you. So T is in the same area where we have morning.